Good afternoon, everybody. And I just want to set the tone. With us, there are a lot of people who are joining virtual, but majority are with us physically. And I'm very happy to say that patron of my hospital, Dr. Rath, is here with us. And so basic theme of this conference is adaptive immunotherapy, cellular therapy, and basic focus on CAR T cells, because not because there is a lot of interest. The interest was always there, but there are a lot of issues related to this, and a lot of patient demand, a lot of industry demand, a lot of regulatory demand. So the whole idea of this concept of this meeting was we all sit together, the people who are using, the people who are manufacturing, and probably regulatory, so that we can understand each other, understand each other's problems, and maybe come up with some better and a faster solutions. What solution I want as a clinician is, I will explain very simply. I want a solution of CAR T cell availability as good as availability of COVID vaccine. So there is COVID, there is no vaccine. All of a sudden you have vaccines. All of a sudden all trials are over, and all of a sudden more than 1 billion population is vaccinated. So that's the speed we need. And if at all we want to really do something in CAR T cells, we will have to follow this speed in all the areas, more so from regulatory purposes. So my plea is we will be able to generate enough interest. I can assure you that the agencies have been sensitized about this meeting. They are aware about this meeting, and I'm sure the message will go that why this meeting is being held and what is the aim behind. So the aim is to make the drug accessible and the research accessible as fast as possible, so that we don't be dependent on someone else. So like the government's today's theme is Atmanirbhar Bharat. We have to be really Atmanirbhar, and I wish I don't have to import it from other countries. So that's the basic theme. And we deliberately designed it in such a way that first half an hour we just do an internal discussion so that we know each other, and then we go ahead with presentations. We could locate 6 companies who are in some stages of development or launch of the product. So all are represented here. Probably this is one of the forums where everybody is there. So you will get a time to interact with all of them. And maybe because some of you may be researchers, some of you may be investigators, some of you may be end users. So that's the whole idea, that bringing everybody together. So the first part is about half an hour discussion on current challenges and opportunities in cell therapies in India. So we have with us here some people physically present, whom I would like to invite to the dais. Dr. Gaurav Kharia, please. Dr. Siva Prakash, Dr. Tanveer, I think Dr. Ranjit Sahu is not there. Dr. Shaswati, she would be joining virtually. And we are virtually joining Dr. Dharma Chaudhary, Dr. Rahul Purwar, Dr. Dinesh Burani, and Dr. Shaswati Basak from Ahmedabad. She is a basic researcher from INTAS. Before I start, my humble request, because it's really a very small audience, so I really request everybody to introduce themselves. Start from Tanveer, please.
I'm Dr. Tanveer, currently working as assistant professor in Germany. My PhD from CSIR IGL, whereas I'm working on cell-based therapies. Then I moved to States, where I did my postdoc at NIH. At NIH, I was actually working on CAR T cells, and I was also part of the team who developed Yescarta. I was working with the Stephens Group. And then in 2018, I came back to India. Here I joined Jamia Millia Islamia as assistant professor. So my lab is currently working on next generation CAR T cells. Besides, we are also exploring CAR NK cells. And I'm also working with Cellogen Therapeutics as a consultant. And that's about me.
Shiva, please.
My name is Shiva Prakash Ramalingam. I'm from CSIR Institute of Genomics and Integrative Biology. I'm a principal scientist there. I did my PhD from University of Madras, and then I went to Johns Hopkins as a postdoctoral fellow. Spent there for almost 7 years, working in gene editing nucleases, induced pluripotent stem cells, especially for cell and gene therapy. Came back in 2017, joined in IGIB. So my lab is focused on two aspects. One is genome editing based cell and gene therapy, and the second one is on CAR T cells for the hematological malignancies. We work closely, my lab work closely with Tanvir. So we also have some collaboration with the Cellogen Therapeutics for the CAR T cells.
So before Dr. Gaurav, I would like to introduce Dr. Brenjens Rainier. He is our star speaker for today. Not only star speaker, he is an original researcher on CAR T cells. If you don't know him, probably all of you know him, and I'm sure all of you have read him. More than 27 years into CAR T cells. So the whole history of CAR T cells is associated with him. And if any one of you is actually interested into digging into the history of CAR T, there is really a big history, because you did not know how to get T cells, how to enrich T cells, how to duplicate T cells, how much cells to use, whether you want to use 1 T cell or a 10 T cell or 100 or a million, all these issues. Probably he has been a contributor in the research of all these queries. What drugs to use, what conditioning to use, and he has done basic research on every aspect of CAR T cells. So I'm very happy to present to you Dr. Ranges, who will be here with us. And over the day, I would request you definitely to interact with him. Dr. Gaurav, please.
Yeah, thank you, sir. So I'm Gaurav Kharia. I work as a clinical lead at Center for Bone Marrow Transplant and Cell Therapy. So we have a center, one of the high volume centers doing transplants. Apart from this clinical domain, my other area of interest is cell and gene therapy. So I'm also the founder and director of one of the startups, cell and gene therapy startups called Cellogen Therapeutics. And we have been working to come up with indigenous CGT solutions for various, malignancies to start with, and also for hemoglobinopathies, which are need of the hour for this country. And we have collaborations with Jamia, with IGIB, with DBT, who have been helping us extensively all through this journey. And some of the work will be presented by Dr. Tanvi today. Thank you very much.
Good evening to you, ladies and gentlemen. I am Dr. Colonel H.S. Ratti. I represent MedTherapy Biotechnology. It's a cell and gene therapy unit based in US, Boston. And we are setting up a manufacturing facility here in Noida, translating into aspects of what he wanted, that all the I have my team here. We'll be presenting our what we intend doing at this facility, and coming up with availability of what he's been talking about is Atmanirbhar facilities once India opens up. Thank you.
Hello, everyone. Myself is Dr. Niyaj Mishra. I'm working as an Assistant General Manager in MedTherapy.
Hello, everyone. My name is Rashid Hassan. I am Senior Manager in Vector Development in the MedTherapy. Thank you.
Hello. My name is Dr. G.K. Rath. I've been working in AIMS in cancer for about 50 years now. I established the first ever translational cancer research center in a place called Jhajjar in Haryana. It's a 300-acre campus. If you have not seen, then you have not seen Taj Mahal or Qutub Minar. So right now I'm associated with Dr. Dinesh in Sarbada as the advisor. Thank you.
Hi, my name is Sunil Bhatt. I'm a pediatric hematologist oncologist from Narayana Health City, Bangalore, and I'm one of the principal investigators for the CAR T trial, phase 2 CAR T trial, which we're running at Narayana Health City with Immunil.
My name is Dr. Samir Thirupure, and I'm a hematologist and a bone marrow transplanter from Mumbai. And you can probably categorize me as an end user who would use all the science and use it in the patients. Thank you.
Hi, afternoon. I'm Dr. Anil Kamath. I'm a hematologist. I had clinical development at Immunil, and I'll be happy to share some data later in the day. Thank you.
Hi, everyone. I'm Akhil. I'm colleague of Dr. Anil at Immunil Therapeutics. Thank you.
Hi, good afternoon. I'm Dr. Randeep. I'm heading the Department of Medical Oncology at NH Gurugram. I think I'm one of the only medical oncologists in this group and an end user.
Hi, everyone. I'm Dr. Tejaswini. Yes, of course. I count as one more. I'm Dr. Tejaswini Ved. I'm a hemato-oncologist and I've recently joined Medanta. Thank you.
Hi, I'm Dr. Garima. I'm a pediatric hemato-oncologist and I work at Indraprastha Apollo Hospital.
Hi, good afternoon, everyone. I'm Bhavna Gupta, working in regulatory affairs with Dr. Reddy's Laboratories.
Good afternoon, everyone. Myself, Sanjay Garg. We represent Intas Pharmaceutical Oncology.
Good afternoon, everyone. Myself, Dheeraj Sajdeva, representing Intas Oncology.
Hello, everyone. My name is Pawan, and I am an Associate Director in Med Therapy Biotechnology. Thank you.
Hi, hello, everyone. I'm Dr. Juhi Verma, and I'm representing Med Therapy Biotechnology.
Good afternoon, everyone. I'm Vasundhara and representing Med Therapy here.
Hi, I'm Aditya. I'm also a medical oncologist and an end user. We're part of the team which moved to Apollo Hospitals Bangalore and hope to set up cellular therapy unit towards Q2 of this year. I do run the adult transplant program over there for now. Thanks.
Hello, everyone. I'm Yash Dave. I'm part of the Dr. Reddy's commercial team.
Yeah, hi, everybody. I'm Dr. Vineet Govinda Gupta. I'm a medical oncologist and end user. Thank you.
So thank you very much, everybody. And I just leave the floor forum now to, sorry, I have to introduce my panelist on the virtual. Dr. Dinesh, can you hear me?
Yes, sir, I can hear you. I'm Dinesh Burani. I'm an end user in Rajiv Gandhi Cancer Institute.
Rahul?
Good afternoon, everybody. My name is Rahul Purwar. I am faculty member at IIT Bombay and also currently serving as CEO of ImmunoAct, one of the startup in the CAR-T therapy area. Thank you.
Dr. Dharma?
Hi, good afternoon. I'm Dr. Dharma Chaudhary, and I am working as a Director Blood and Marrow Transplant, Sanar International Hospital, Gurgaon, and I'm one of the end users for CAR-T cell therapy.
So I'm really happy to again welcome you all to this forum. And I can tell you, you have seen with the presence that this appears to be one of the really powerful forums of the country, which can move the country. So I would now just request Gaurav to take over and do some preliminary discussion. We do have presentations, but I still want, before we go to presentation, I would want you to come up with some challenges which we are facing in CAR-T space. Please.
I mean, the first and foremost challenge which we face as a country is that we are quite sluggish as far as the growth of this particular modality is concerned. So, I mean, as we can see, and as you also highlighted, there are just a sort of handful of companies or startups working in this particular domain. To count them, not more than 5 or 6. And if you look at our I would not say competitor country, but yes, they are competitors when it comes to medical technologies, such futuristic technologies. There are close to 2,500 to 3,000 trials ongoing, and I would say another 500,000 other small or big companies working in CGT domain across the globe. So the first and foremost challenge is that we have been quite late in whatever beginnings we have made in this particular domain. Subsequently, even after the late beginning, usually we have this tendency to bounce back very fast, but to sort of fructify any idea about cell and gene therapy in this country is still sort of very nascent. The receptiveness for this particular thing is not as expected, okay, from various sectors, I would say, from government sectors and from end users also, I would say. So these are, just few of the challenges which I can say to start with. There are other challenges also which we face on a day-to-day basis as far as the developmental process of any particular product is concerned, which is quite a rigorous task. And we have been through this, and some of my other colleagues, Rahul is also here. So we have been through this rigorous process of 2.5 years, 3 years, but each challenge we face, we come up with solutions, and we are happy to come up with some indigenous. So Rahul already has an indigenous product. Subsequently, we are also trying very actively to come up with a indigenous product for the Indian market, because that is how we can bring a state-of-art product at an affordable cost, which is the core need of our country. So these are some of my thoughts.
Shiva, Shiva, you have any thoughts?
So I am from academic institute, actually. I'm a basic research scientist. So what we are facing is the funding for this research. So the government has to encourage more. We are looking for more support for the next generation cell and gene therapy thing to take it forward, especially in the basic research as well as manufacturing and other things, taking it to the next level. So that is what the research side we are looking for.
Dr. Tanveer?
Absolutely, I agree with Shiva. There has to be more funding from the government agencies. But besides that, I think we also need to come up with more innovative approach, like people have developed and they are clinically being tested allogenic CAR T cells, which genome editing based allogenic CAR T cells. And then there are also approaches where people are making these CAR Ts in vivo. I think once we have those technologies validated, so then it may be accessible to the wider patients. Like we can have products made at different places in the world, and then they can be distributed also across globe when we have allogenic or in vivo CAR T cells. As of now, with autologous CAR T cells, I think it is more kind of a centralized product where these products have to be created at a particular place, and the patient has to be also within reach. So once you freeze thaw these products, they lose their viability. So I think coming up with new next generation products, allogenic and in vivo CAR Ts is what I feel we need to invest more, both in terms of research, innovations, as well as support from industry and government sector.
Dr. Dinesh, Dr. Gurani.
I agree with all the presenters there. I was just thinking whether we will have I think some later you will have it the update from all the companies. Where are we at the moment, and
Yes,
how far we are away from the end users?
So we have practically all the companies here. So you will really hear from them.
Yeah, and Gaurav was saying the end user is slow. The end user is not slow. We are waiting. We are eagerly waiting for.
No, sir. Don't
end user is not slow.
So you are one of the end users. The end user is actually the patient. So the receptiveness in the patients is also not as expected. The awareness about the overall things. So about we as clinicians. So I'm on the both. I'm on both the sides actually. So a developer as well as end user. So
we are eagerly
from the
waiting.
clinician's point of view, I think we all are just waiting to make this modality or therapeutic modality available to our patients in need.
Yeah. So Dr. Dharma, please.
Yeah, I think I agree with Dinesh. What we are looking right now is a readily available CAR. and which can be, results of that company can be reproducible to the internal standard. So the story goes same like the story in the transplant in India, that 20 years back, there are hardly takers for transplant. The cost effective, accessibility, the faith in the system. Now the car comes, this question will be there initially. But I think this country is vast and even 1% of the country can afford the CAR T cell. We will have a huge number of the waiting list to get the CAR T cell therapy for those patients. So I think we need to have more and more companies who can provide those autologous CAR at least as of now and the allo CAR in future. So, and we will hear the updates from the companies where we are and how can we support, because once the demand comes up, maybe we are not ready to support all the demand.
Yes, Dr. Rahul.
No, I think all the panelists have elaborated very well all the challenges. And that's good to hear there's a lot of demand and clinicians and end users are waiting. And so are we. We are also waiting for the regulatory nod and finishing of our clinical trial. But unlike vaccine, these trials take little time. So we have to be patient because there is some process to be followed. And I think India will soon have, I think we will hear from other companies as well as us, that I think soon India will have the end users will have access to the CAR T cells. So I think we will have a little bit more update in the presentations.
So I think we have really enlisted the major challenges, whatever are here. And the major challenge is, there are a lot of challenges: the development challenge, cost challenge, accessibility challenge, availability challenge. And it's really nice to hear that we are all aware of these challenges and trying to mitigate them. So after this panel discussion session, we will have some individual presentations. So the flow is, we'll have few individual presentations and then which go till evening. And in the meantime, is my humble request, anybody is allowed to intervene during any presentation, because we want this forum to be a discussion, and we want this forum should come out with something, at least some thought process, and our thought process of all of us become a common thought process, so that we can actually do some advocacy at a state level, advocacy at a patient education level, and a professional education level. But for that, all of us should try to at least get some thoughts which are common to all of us. It will be easy for us to grow. So I would like to thank all my panelists, and thank you very much. And for my next session, so we kept some small presentations, and I'm sure you would get insight into what is actually happening in the CAR T space. Some data may be a duplication between the presentations because nobody knew what other person is speaking. So please forgive for that, forgive the speaker, because it's quite possible that he may duplicate some slides or some science. It's all right. Let there be a revision. There is no harm. So our first presentation is by Dr. Tanveer, who is working at Jamia Millia Islamia on a CAR T space. They have a huge lab in Jamia working for many, many years. And he would like to tell us something on next generation multi-antigen targeted CAR T and CAR NK cells. Please.
So good afternoon, everyone. I'll be talking about next generation of multi-antigen targeted CAR T and CAR NK cells. So this is a relatively new approach where we target multiple antigens present on either homogeneous or heterogeneous cancer cells. As a significant development in the field of cancer immunotherapy, this approach allows us to effectively target these cancer cells, and it also helps in treatment outcomes for patients. So multi-antigen targeted CAR T and CAR NK cells has seen recent advancements in clinical trials, and the results so far have been encouraging with these multi-antigen targeting CAR T cells. So the CAR T cell therapy has made significant progress over years, and the first two CAR T cell products which were FDA approved for commercial use were in 2017, Kimria and Yescarta. I was at NIH doing my postdoc when the phase 2 clinical trials were ongoing for Yescarta, and then I came back to India. But as you can see, in last 3 years, we have 4 commercially available CAR T cell products. So, and by 2023, we are going to see more of these commercially available approved CAR T cell products. So all the 6 CAR T cell therapies which are FDA approved uses second generation design. And I'll be talking more about these designs in subsequent slides. And what we know so far is there is clinical data available from more than 10,000 patients as of Feb 2023. And most of these trials have been conducted with second generation CARs. And then we have also newer generation, third generation, fourth generation, fifth generation CARs, which have also been tested in clinical trials. So what defines the generation of these CAR T cells makes a lot of difference. First generation CARs, which were developed in 1993-4 and then tested subsequently in preclinical models in early 2000, they did not show the expected anti-tumor activity and proliferation. And then we had second generation CARs. The second generation CARs were developed at UPenn as well as at NIH by Carl June at UPenn and NIH Steven Rosenberg's group. So second generation CARs were the ones which showed better anti-tumor activity as well as longer persistence, and the trial started in 2008. So the difference between Yescarta and Kimria in terms of second generation is the extracellular domain is same, that is CD19 targeted scFp, while the intracellular domain, it is either CD28 based domain or 4-1BB. So Yescarta utilizes CD28 domain and Kimria has 4-1BB. So that is the difference between the 2 second generation CARs, and they have shown promising results in clinical trials. And then we have third generation. The third generation CAR has one extra intracellular domain. So it can be a combination of CD28 as well as 4-1BB. And there are other such domains which people have utilized. And then comes the fourth generation. Fourth generation becomes a little more complex because in this case, in addition to having these co-stimulatory domains like 4-1BB or CD28, the fourth generation CARs also have an extra domain. So that extra domain, as in this example slide, you can see, that can be an IL-2 secreting molecule. And what these cytokines, intracellular cytokines do, they help in long-term persistence and proliferation of these CARs. So you do not need to give these cytokines from the outside exogenously. So that is the fourth generation CAR. And these CARs are also called as armored CARs, or people call them as trucks. And then we move to fifth generation CARs. So here the design becomes more complex and ambiguous. Some groups have listed fifth generation CARs based on the extra domains which can impair the signaling pathway of JAK-STAT, or there can be other domains associated which can enhance the persistence of these cells. And then other group also mentioned fifth generation CARs as the CARs which are allogenic, genomic, after doing genomic manipulation editing, like TRAC gene editing to make them allogenic, or PD-1 gene deletion to make them exhaustion resistant so that they can survive for a longer period of time. So the fifth generation is a little more ambiguous. And then people also say fifth generation CARs are the ones where you simultaneously target 2 different cancer antigens. So the design can be a bicistronic CAR, where you can have extracellular scFEs targeting 2 antigens, for example, CD19 and CD20, which are expressed on the B cells, either as a bicistronic form or a tandem CAR. And then B cells, which are known to express 3 prominent cancer antigens, CD19, CD20, and CD22. So those can come under the category of fifth generation CARs. So irrespective of the generation of these CARs, what they need to do is they should have a better anti-tumor activity. And in addition to that, these CAR T cells should persist for a much longer period of time. That is the whole idea. when we talk about the allogenic CAR T cells. So now, as I was mentioning, the data that we have so far from more than 10,000 patients and what we have learned with the second generation CARs. And again, the second generation CARs, mostly the SCFE domain, which is the extracellular domain, is FMC63, which is a mouse clone, the mouse antibody clone for CD19. And then people have done all these different modifications, made them humanized, or then now we also have human CD19 SCFEs. But irrespective of the SCFE, the second generation CARs, so far we have seen, there is a relapse of more than 50% in the patients. And then this relapse is majorly because of lack of persistence of these T cells when they go into the body of the patient. And second prominent factor here is antigen escape. So the antigen, which is CD19 expressed on these B cells, eventually these B cells become resistant. They shut out this CD19 antigen. So the CD19 directed second generation CARs are not effective in eliminating this B cell tumor. And of course, there are other challenges like CRS, which is manageable. Insertional oncogenesis. This is a case with lentiviral and retroviral vectors, but has been seen in therapies where these vectors are used, but we have yet to see this insertional oncogenesis happening in CAR T cells. And then manufacturing cost, whether we are using automated or semi-automated systems, and autologous or allogenic CAR T therapy. So what I'll be focusing in the rest of my presentation, the two major challenges with the existing second generation CARs, that is lack of persistence and antigen escape. So briefly, when we look at the molecular mechanism, which is associated with the lower persistence of second generation CARs, so we have now come up with 6 major pathways which plays an important role in leading to lack of persistence of these T cells. First, CAR T cell exhaustion. Second is tonic signaling. So people sometimes underestimate this tonic signaling. When we are working at NIH, we had to play with the design to make sure that there is an optimal expression of CAR on the surface of these T cells. It should not be too much, it should not be too less. So it has to be an optimal expression, because if the expression of the CAR molecule on the surface of cells is more than needed, then it leads to tonic signaling and these CARs become dysfunctional. And then third is activation induced cell death, where these CAR T cells also involve FAD, TRAD, cell death receptors, and that eventually leads to death of these cells. Trogocytosis is another emerging mechanism which is associated with low persistence of these cells. And genetic makeup of a particular individual. When we take out the cells from a particular individual, these patients have already undergone different treatments, chemotherapy and other, and there may be some genetic modifications which might have happened to their T cells that may also impair their long-term persistence. And of course, manufacturing is also critical, because it also makes a lot of difference how you are making your CARs. So what kind of lentiviral approach you are using, and all the different cell culture approaches. And then this is a new mechanism where we have worked on this tunneling nanotubes. So, and we have been observing this CAR molecules can also move from one cell to another through these tunneling nanotubes, and that also contributes to the persistence of lack of persistence of these cells. So now coming to the second challenge, these CAR T cells have CD19 when we're talking CD19 antigens. So the escape mechanisms majorly involves antigen downregulation at changes at post-transcriptional and translational levels, and then antigen loss when there are changes in the regulatory or the ORF of CD19, the whole antigen is lost from these B cells, and then lineage switching. There is the phenotypic change on the B cells, so they do not allow the CD19 to be expressed on their surface. So considering these two major challenges, one, lack of persistence, second, antigen escape, our lab has from last 4-5 years has been actively working to address these challenges. And what we found is that I'll be showing one part of the study. The cytokines that we use to activate these cells makes a huge difference. IL-2 is a traditional conventional cytokine which is used to activate these T cells, proliferate, proliferate and also helps in the differentiation of these cells. And it has been extensively used to make the CARs. Second, and recently people have also explored IL-7, IL-12, and IL-15 used in different combinations to help in the CAR T cell proliferation. And what we did is we used these 4 cytokines in different concentrations and in different combinations. What was interesting was that a combination of cytokines, when you give IL-2 for initial 48 to 72 hours, then followed by IL-7 plus IL-15 for the rest of the proliferation cycle, the CAR T cells showed much better proliferation. And then we also looked at the in vitro persistence, and we saw that a combination of IL-2 then followed by IL-7 plus IL-15 combination led to much better persistence of these cells. And then we also took all these different combinations, performed RNA transcriptome analysis of these CAR T cells, and then there were many genes which were differentially regulated between these conditions. And when we looked at the pathways, the major pathways which were differentially regulated between condition when we have IL-2, then followed by IL-7 and IL-15, mitochondrial metabolism was a prominent pathway which was getting differentially regulated, and exhaustion markers, stemness markers, then senescence markers, as well as autophagy markers. So after doing all the filtration and final comparison, what we came up with 3 key genes which showed much differential expression between all the conditions. Among them, ATP5J2, GIMAP1, and HNR-NPH2. These were the 3 genes which showed differential regulation. ATP5J2 showed lower expression. The other 2 were increase in expression. Then we evaluated these genes in vitro functionally. We designed lentiviral-based shRNAs to target these different genes. And what was interesting was when we targeted ATP5J2 and lowered its expression, the proliferation persistence of these CAR T cells was substantially increased than the mock control T cells. While GIMAP1 and HNR-NPH2 did not show any significant difference. Then we further went to see what this particular gene does. This ATP5J2 is a molecule which is involved in regulating ATP production from mitochondria and also regulates mitochondrial metabolism. And here you can see we performed, we used a lentiviral expressing mito-GFP, and we looked at the mitochondrial content of all these cells treated at different conditions. And we found that when we have shRNA targeting ATP5J2, the mitochondrial content was less. But when we overexpress this particular gene, there was increase in the accumulation in the number of mitochondria present in these T cells. And what was interesting was that the increase in the number of mitochondria was not correlating with the increase in the metabolic fitness of these cells, but it was actually hampering the health of these cells. So these mitochondria were not functional. They secreted a lot of ROS. Their membrane potential was lower. And then there was also decrease in the clearance of these mitochondria from the cells, that is through this mitophagy assay. So in conclusion, what these results tell us that when we use this unique combination of cytokines, which is IL-2, then giving a rest, and then followed by IL-7 and IL-15, there is enhanced metabolism of these cells, and which eventually lowers their ROS levels and also contributes to the mitophagy, so the dysfunctional mitochondria are not accumulating in these cells, and that eventually leads to much better persistence of these cells. So now coming to the second part of the challenge, which is how to overcome the antigen escape. We asked, can we simultaneously target multiple antigens on the and make unique CAR constructs? And we have made 35 different CAR constructs using different combinations. The antigens, SCFEs we worked on was CD19, CD20, and CD22, used either as monospecific or bispecific combination. And then we also tested different combinations of co-stimulatory molecules. And this work was carried out with help from Dr. Shiva Prakash at IGIB and Salogen Therapeutics, Dr. Gaurav Kharia, who provided the resources and funding for this particular work. So besides CAR Ts, we have also generated almost now 55 different, some of them are also armored CARs, 55 different vectors, lentiviral vectors expressing all the next generation CAR molecules. And this work is being carried out by PhD scholar Sufyan Ansari in the lab. So I'll not talk about, I'll not talk much about CAR NK cells, but I'll focus mostly on CAR T cells. So among the 35 constructs which I showed you, we have finally narrowed down into two potential constructs, which is CD20 followed by CD19 and ICOS4 and BB co-stimulated domains. All these constructs are driven by EF and alpha. And then this is the general design of these constructs verified by sequencing and other methods. So after we validated these constructs, we use this approach of magnetic isolation CD4 and CD8 cells. Then these cells were confirmed by looking at the surface expression of CD4, CD8 and negative marker like CD14. And after we confirmed the purity of these cells, they were transduced with the lentiviral vectors expressing these CARs. And we confirmed the expression of the CAR molecule on the surface of these T cells, as you can see here. And this work was carried out by Ariz Akhtar, who's PhD scholar in the lab. So now, once we validated these CARs, the expression of these CARs, we performed in vitro assay. So in the in vitro assay, we did use K562 cells, transduce it with different antigens which are expressed on the B cells, CD19, CD20, and CD22, to simulate this antigen escape of B cells. And what we found is when we have a bispecific CAR which express either CD20 or CD20, CD19, and we have cells which do not express CD19 but do express CD20, there is killing by the bispecific CARs. So, and similarly, when we have a condition where CD19 or CD20 both are lost and only CD22 is expressed on the surface of these cells, the other CAR construct, which is CD22 and CD19, did show the better anti-tumor activity. And we are also working on, have generated CRISPR-based knockouts of these cells, Raji cells, where we have no surface expression of these in the lymphoma cells. With either one antigen, two will be left over, will be only the CAR T cells, and all the lymphoma cells are being taken care of by the CAR T cells. So that is the time lapse imaging. And I'm not showing all the other experiments because of the time constraint. So I'll briefly talk about the preclinical study. We have procured these NSG transgenic animals from US, which took almost 2 years to get to India. And finally, we have validated these experiments, in vitro experiments in these transgenic animals. Compared with untransduced, we could see CD20, CD19 mice survived for a much longer period of time, and there was better tumor clearance. And we also did compare it with second generation CAR. All in second generation CARs, these are represented images. We have repeated this experiment a couple of times. And with second generation CARs, there was still some tumor left. But with the bispecific CD20-19 CARs, the tumor clearance was much better, and the survival of these mice was also better. So as of now, we have completed our basic research with all the indigenous CAR molecules. The lentivirus particles are also generated in-house. Preclinical studies have been completed. Now we have also approached regulatories. We have got some initial approvals, and we are also waiting for other approvals for conducting our phase 1 and phase 2 trials. And this, finally, I'll express my gratitude to Dr. Shiva Prakash at CSIR IGIB, with whom we did construct all these molecules, and Dr. Gaurav, Cellogen Therapeutics, who provided funding, and the lab members who have been actively working in this area, and the funding agencies, although we have yet to get any fundings from these funding agencies, but yeah, I'll acknowledge them. Thanks. I'll be happy to take any questions. So, Gaurav, you want to ask anything?
So, I mean, you just summarized the entire very comprehensive overview of I mean, I've seen this every day, so not many questions are there from my side, but I'm sure there'll be a number of questions from other audience, other sort of developers or the clinicians have their questions about the advantages of bispecific or third generation.
Absolutely.
and not only advantages, but challenges which can come up with these.
Exactly.
So what do you anticipate? I mean, yes, you feel that they are better in terms of proliferation and persistence. But if we talk about the other side, what could be the challenges which we need to foresee can come up with these bispecific and third generation constructs?
So the challenges can be like we are talking about simultaneously targeting two different cancer antigens, CD19 and CD20. So the off target may be slightly higher in this case because these antigens are also expressed by the normal cells. So that may be little higher. And also we have to take care of the CRS. So because we are talking about much more potent CARs.
So if I ask about that, although it will differ in the clinical setting as compared to the preclinical setting, but what does your tox analysis say in the preclinical
So
settings
as in preclinical studies, we have not seen any much when we compared with the conventional second generation CARs, the toxicity was not that high. But in patients, it may be a different scenario. But people across globe has already tested a couple of these bispecific CARs. So I think this is going to be manageable kind of in terms of CRS and other issues. The best part here is the challenge, which is antigen escape. So that can be taken care of.
That can be
because now if you have
mitigated.
patients where 20 to 30% of cases where you have antigen escape, you can provide them with a better alternate, right? Which will have a much better persistence.
No, I agree. I mean, this was the gist of the ASH this year, that the next ASH will be all about the bispecifics and third generation CARs.
Exactly.
So it definitely makes sense.
Also, there is one question.
Thank you, Dr. Tanvir, for a great presentation. It was really good to see all the research which is backed by academia and collaboration with industry. My question is that you have shown a battery of clones which you might have screened. How are they essentially different from each other? And could you, in your experience, either with some in vitro studies, could compare with the existing CARs which are in the market already? Some of your candidate clones, was there some amount of comparison done to the existing CARs which are there in the market?
Absolutely. So the first question is, what is the difference in terms of the design, right, in these CAR constructs as compared to the others? So the difference in the design is all the components to talk about, the co-stimulatory domains, right? The combination of these co-stimulatory domains and then the transmembrane domain. So there are transmembrane domains which are CD8 based, there are transmembrane domains which are CD28 based and ICOS based. So the design is different based on the combination of these signaling molecules, co-stimulating co-signaling molecules. And that's how we could get patent out of this, because the designs we have chosen were unique. And then coming back to the second question is, yes, we did compare these CAR designs with the existing ones. And there are in vivo studies also where we have done the comparison with the existing second generation CARs. So yes, the comparison was always done, both in vitro as well as for some of these constructs in preclinical models.
Thank you.
Yes, please.
Nice talk. So myself is Neeraj. Just very quick few questions. So like, because you showed a tandem CAR as well as a bicistronic CAR. Now question comes, which antigen take first? Because when you make a loop CAR, the exposure to the second antigen always become a problem, because maybe many a times it's overshadowed by the first one. If you take the bicistronic CAR, even the second CAR expression remains a problem.
Excellent. Yeah, that's an excellent question. I agree. So when you have a tandem CAR, both of them are expressed together on the surface. So the availability of the surface, synapse formation becomes limiting for one of these designs. And that is what people have also seen in different studies. And then people came up with the idea of bicistronic CAR, where you have both the antigens expressed by the same cell, but at different locations on the surface, right? So, but here in this case, it also makes a difference, the hinge which you are attaching with the CD19 and CD20. The length of the hinge determines the synapse. So if the hinge is of optimal length, then both of these molecules can make synapse together. But if the hinge is either smaller or longer, then these steric hindrances will come into the picture. So hinges are critical when we make these tandem cars.
Then another question related to the same. So when you talk about co-stimulatory domains, so many a times what we see that many of the construct, they use the same co-stimulatory domains. Some research coming up when you are trying to switch the co-stimulatory domains, especially 28 41BB. But the thing is how to avoid the tonic signaling, because of 28 it's very fast and already initiate tonic signaling. How your second 41BB is going to work?
That is another excellent question, how to avoid tonic signaling. This is what people, when they show the expression of these cars, they get excited that we have got per cell, we have got 100 molecules or 200 molecules. Then the shift in the facts, you will see a huge shift in the facts. Data plots. So regulating that amount of expression, and that this is what we have been working at NIH also. Regulating the number of CAR molecules per cell becomes important. The tonic signaling can be regulated by letting the optimal amount of CARs expressed on the surface. That's why people are also trying, and we have been also looking at different approaches of EF and alpha, where you can make some modification in the promoter to not let this molecule to be do not let many molecules be expressed on the same T cell. So that's how we can control the tonic signal.
Especially the work that you showed on the 35 systolic constructs, and you're selecting one or two of them. I'm not sure whether they're going into from the preclinical to clinical. I don't know whether that is going to happen. But on that note, I just wanted to, a slight note of caution is there are multiple ways of doing it. One is the bicistronic, then of course the tandem. You've got the co-administration and then the co-infusion, or when you say the co-transduction. True. So we do not know at the moment what is best, what is most economical. On the bicistronic or the tandem node is what you mentioned, the hinge size, because it may happen that it looks great, the science is great, but when it comes to the cancer cell, is it able to stick onto both antigens? Does it able to activate or kill the cancer cell off? So those are the various things. So theoretically, yes, and we look forward, as Dr. Gaurav mentioned, we look forward to the data on both 1922, where a lot of data is being generated, 1920 as well. But I think we have to just be a little bit more careful. 1 + 1 is not always 2. It may be 1 + 1, maybe 1.5. I think there's a lot of we need to watch out for. What we can say at the moment is the box standard second generation cars are good, and we know that, and some of it will be covered in one of the presentation. But we look forward to this. I think it's a way ahead.
No, I totally agree. So that's what we have data from last 15 years on second generation cars, and that's because of the challenge we saw. Because when you are going to give this car to the patient, it's not a cheap treatment. The patient should not have this kind of, the patients are now smart. They should not think of that there is a better version of the car, and these bispecific, bicistronic, and the one which was recently presented in ASH, where what you are talking about, where you can have two different vectors co-transduced, that's a third approach. Those two vectors transduced differently, then you can have some population of T cells which will express CD19, some population which will express CD20. So yes, which can have both. So that is the third approach. But from last 6 years almost now, we have data on the bispecifics, and phase 1 clinical trial has been promising. That is the reason, and we have been also working on these from last 5-6 years. What you are talking about when you have these two targeting the cancer cell, I have shown the data. Data is from other institutes as well. They have an optimal, better anti-tumor activity. The best part is when you have a CD19 negative cell, what you will do that. Both K562 data and then CRISPR knockout data that we have shows that when you have a CD19 negative cell, the bispecific CD20 or CD22 CAR works in those cells. So that is how it seems to be a better alternate approach. So when you have CD19 from last 15 years, you need to come up with something which is new. It would have been better if second generation CARs would have been working absolutely fine. But when 50% relapse, we need to innovate and come up with something which is right, just really new. Right?
So I think that's the journey of coming from first generation to second generation.
Exactly, evolving.
Right. So thank you very much, Dr. Tanveer, for a really excellent beginning of this program. And thank you very much. And our next presentation is again an interesting presentation on a molecule, Varnim Gene autoleucel, by Dr. Kamath. He would be speaking on data from this study, the Imagine study, which was conducted by the Bangalore group. And I think we again will get new insight into something, some other new things in CAR-T. Please.
Good afternoon. Before we go to Dr. Kamath's presentation, I have an announcement. We have 125 people logged in on the online portal, and we are receiving several questions. So this announcement is as a welcome for all of them who are attending virtually. And your questions will be taken up during the question and answer session after these talks. Thank you. And apart from this, we have this being streamed on LinkedIn, YouTube, and other platforms also. There, I do not have a comment right now, but at the end of the day, we will be able to announce that. Dr. Kamath, please.
So good afternoon. First, Dr. Pendarkar, thank you very much for inviting me and Immunil to this meeting. And it is fantastic to actually interact with the prime players in India as we stand in CAR-T domain. And many of the faces and of the people I've met today are very familiar, and it's very nice. And I hope that this continues and this interaction becomes a good player where we can exchange ideas. So my task today is to explain and rather discuss where we are in terms of Varnim Gene autoleucel, the Imagine study, and how Immunil is bringing CAR-T to India. So I'll break this session into firstly the unmet need. We had the panel discussion, and as Dr. Kharia mentioned, the unmet need in terms of our first CAR-T, which was approved by US FDA in 2017, TISA and Axicell, and it's now 2023, 6 years down the line. We may be a bit slow, but we're getting there. And of course, today's meeting is a testament to that point that we are now soon going to get into the clinic in terms of commercialization. I'm not going to go into details of this slide because this is an informed audience. Suffice to say that CAR-T cells essentially target the antigen on the cancer cell. I'm not going to go into details how they do it. But of course, it is very important that we mention on why we are doing it. We know about this poor prognosis for BELL and BNHL. These are the classic papers, both from the UCAL as well as the Scholar one, which shows that for relapse refractory leukemias and lymphomas, the outcome is very poor, dismal, 20% or less. And moving on from there, we know that two of the pioneer drugs that came out from US, one was the TISA Genluxil from UPenn group, Carl June's group, and Exicaptogen from the NCI, which was the CD28. So both of them are different, but they had the same binder, which is FMC63 targeting CD19. And we have seen the real world data, which shows that potentially there are cures in 4 out of 10 patients. So initially you have an outcome which is as high as 60 to 80%. And over a period of 5 years, even in the real world data, we have seen that 4 to 6 out of 10 are potentially cured. And nowhere in the treatment of hematological malignancies have we seen a treatment that is that effective in causing and resulting in cures in this patient population. So as we know, there are 4 approved CAR-T treatments in the world, specifically in US FDA and Europe. Of course, none in India at present. And 4 of these are targeting CD19. 3 of them are 4-1BB costimulatory domain. 2 of them, the Brexicaptogen and CD28 costimulatory domain. 2 other molecules are Abecma and Carvykti. I'm not going to touch on myeloma at the moment. Again, both of them are different. One is a murine, another is a camelid nanobody. What I do want you to focus is ARI-0001 from Spain, because that is where I'm going to discuss a little bit further. Of course, the details for unmet need, all the clinicians and as Samir mentioned, end users, but of course the end user is the patient and we are the facilitators. This is probably an underestimate, but this is what the figures are in ICMR, but we all know that the real numbers are much higher. So bringing CAR-T to India. Now, we all in this room are aware about the the triple jeopardies in terms of what happens. Firstly, the healthcare expenditure. If you compare the top countries in the world, I'm looking at US, Japan, India, and China, because India has overtaken UK in terms of GDP, in terms of the gross. However, the per capita income is low. So if you look at the healthcare expenditure, per capita income, and the cost of CAR-T, India is severely impacted. So hence, to meet the needs of access, availability, and affordability, it is a challenge to make sure the cost at which we can bring the CAR-T product to the country matches to the affordability of the patient population. And that challenge is higher for India compared to US, UK, or China. So I'm going to take you through Warnim Captagen, the Imagine story. Now, this is Warnim Captagen autoleucel is essentially IMN 003A, also ARI 0001, and it was born in Spain. And I'll take you through that a little bit more in details, but a little bit about Immunil. And I mentioned to Dr. Pendarkar, and Dr. Pendarkar gave me an insight of the pre-Immunil days as well. And these are our founders, that is Siddharth Mukherjee, Kiran Mazumdar Shaw, and Kush Parmar, and we are led by Arun Anand, who is the Chief Operating Officer. It is, as Dr. Pendarkar mentioned, Atmanirbhar Bharat. So this is essentially make in India, for India, and India plus. So we're not focused only on India, but going beyond India as well. So this is a glimpse of the facilities that Immunil has set up in Bangalore. Of course, you need to have an end-to-end chain. If you want to bring a product of this kind to the country, you need GMP compliant R&D labs, manufacturing facility, as well as the quality control labs. And I'm not sure whether there are any representatives from ICMR or the regulator here, but if they are fully aware, all these facilities do get inspected and approved so that they can meet the needs of the patient population. A little bit about the preclinical work. The preclinical work was done by our collaborator, Hospital Clinic Barcelona in Spain, led by Professor Juan Manel. And as you can see on the left side is the cytokine assays, which is showing the TNF alpha as well as interferon gamma release based on the lentivirus, which is a third generation lentivirus, but this is a second generation CAR. People do get confused. The lentivirus is the box standard, which has been used for many years, but the transgene is different. What is different is this is an innovator. It is not FMC63, it is A3B1 binder. This is the first A3B1 binder targeted CAR-T in the world. So it is a complete innovator molecule, and there are certain differences which I will mention in terms of the safety profile. And of course, the mouse model does show what I would like to mention to Tanvir, as you showed the mouse model, be careful, because when you inject the cell, the tumor cells in the mouse, if you allow the cells to persist and then you give the CAR-T cells, eventually in the mouse model, the CAR-T cells will disappear and the tumor may come back. It all depends on how long you follow up the mice as well, and at what day you sacrifice. I'm sure I'm going to hear more about it later, but this is there are some intricacies of the mouse model and how the experiment is done, at what time point you sacrifice the mouse. So this is a glimpse of the effectiveness of the A3B1 lentiviral construct for that is Warnim Captagen autoleucel. So first I'll show you the phase 1 data, and then I'll take you through the phase 2 data, that is the Imagine study. So if you look at those figures, and I'll try to get the laser is on. So 71.1%, which is as per you would expect from TISA or AXI. As I said earlier, 60 to 80% is the range in the world for the FDA approved products. But I would like you to point out two things. One is the ICANS, 0%. Another is the CRS, 4.3%. This is grade 3+. Now, these are the two safety features which stand out. And I'll tell you what is different about Warnim Captagen autoleucel. This is the first product in the world which has been given in a fractionated infusion protocol. Yep. What it means is that the total dosage is divided as 10%, 30%, and 60%. And we can give it over from day 0 to day +9 over a period of 10 days. So that is one. And this was the pioneering of the fractionation protocol came from UPenn, from Dr. Carl June. But the TISA team, the Novartis team, didn't take it into the trial because their phase 1 included single infusion protocol. And that wasn't it. Of course, I would like to hear more about it later. But it is difficult once you've gone through the phase 1 with a single infusion, then suddenly change your infusion protocol for phase 2. So what Immunil has done is our phase 2 is mirrored on the phase 1 experience of fractionated protocol. And we find that for India, where we do not want to face these toxicities, manage it more effectively, fractionated protocol is the way to go ahead. So that's number one, fractionated protocol, reducing the incidence of grade 3. But what is the reason for lower icons? We do not know the answer, let me be honest, but we believe that it may be something related to the A3B1 binder, because otherwise it is a 4-1BB. So it may be the uniqueness of the A3B1 binder that has reduced the results of the toxicity for neurotoxicity. And of course, the CRS is under control. So let me take you through the IMAGINE study. The IMAGINE study is essentially the study to determine the safety and efficacy of guanine captation autoleucel in patients with relapsed refractory B-cell malignancies. And of this cohort, we have equal number of B-ALL and B-NHL. Our principal investigator, Dr. Sunil Bhatt from NH, is in the audience. And of course, we also have a DSMB member in the audience. I'm not going to noise him out because obviously that is an important job and he reports to the regulator as well. So we have two important people from the trial in the audience, and some of this data has been presented at ASH. So what I'm presenting today is not the latest data because the latest data has to go to the regulator. What I'm presenting today as the data cutoff on the 28th of November, which was the data which was just presented at ASH. This infusion, as you can see, says day 0, 1, and 2, but we do allow infusions right up to day +9. And that is what will happen once, and with the blessings of the regulator, once it comes to the patients, that is what we will be going for in terms of commercialization. And of course, the primary endpoint is safety and overall response rate at day 90. All of you who are involved in the CAR-T process, this is a box standard, but nevertheless, if there are some people in the room, the apheresis product comes to Immunil facilities where it undergoes manufacturing, which includes the selection, activation, transduction, expansion. As per published data, you're all aware that this is based on a closed GMP system. And of course, process development may evolve with time. I cannot discuss process development here at the moment. The final product is cryopreserved in 3 different bags, and then it is shipped back to the patient's bedside for reinfusion. This is the key inclusion-exclusion criteria. This is published on the CTRI website. What is unique about this is to accommodate India's requirements, because as you all know, there is a huge attrition in the patient numbers in leukemia and lymphoma going beyond 2nd line, 3rd line, 4th line. If you look at the picture in the West, 3rd line, 4th line, 5th line, you've got a lot of patients. In India, there's a incremental drop in terms of eligible patients. So this trial allows the investigator to treat a patient after 1st line, to meet the needs of the patient population. And the MRD, the only thing is it has to be MRD positive for BLL, and for BNHL, it has to be PET positive disease. Of course, the standard exclusion criteria apply. If the patient has had previous CD19 targeting drugs such as blinatumomab or tafastamab, then they are excluded. CNS disease is excluded, but these are the common contradictions that we see in some of the registration studies. So what is the data that was presented at ASH? The overall response rate was 77.7% at day 90. The ORR at day 28 was 80%. PFS, duration of response, overall response was not reached. In other words, we haven't reached the 50% cutoff for median PFS, OS, and DOR. CRS was 70% grade 1. Grade 4 was 10%. We had 1 mortality due to CRS, and ICANS was grade 3 was 0. And overall tocilizumab use was 60%. This compares almost equally with the data that is presented. Remember, this is the initial cohort of 10 to 11 patients. The data, which is of the current data cutoff, will be going to the regulator, and the TRM is significantly dropped since then. Because as the end user, which is our clinicians will get better treating CRS and preparing for CRS. So the CRS incidence, we expect eventually to be in single digits and less. So this on the right side is, I want to point out the proportion of naive cells. Now, those who have done experiments in terms of the pharmacokinetics would know that the number of naive cells also is a indicator of how good your product is and how likely the product is going to expand. And it is pretty good. It's really good in the sense it's in the range of about 35%. Of course, what is shown is a mean and average. This is not that one patient's product. It is a combination of multiple products, and what we are shown is a mean. The figure on the bottom is where you show the B cell aplasia and the in vivo persistence. So of course, there is a correlation in terms of the B cell aplasia along with the in vivo persistence that we can encounter with these patients. These are examples. These are real patients from the trial. And you can see the day 90 responses, complete responses, and fascinating responses, as you can see with other patients elsewhere in the trials in the world. So those are the two main abstracts that was presented. Response peak persistence of 1M capitation autoleucel. First in India, industry CD19 directed CAR T cell therapy with fractionated infusions for patients with relapse refractory B cell malignancies. Early results IMAGINE study. The second abstract was more related to the clinical data, which is the early results from the study for these patients. So what are we looking at? What we want to aim of this statistical hypothesis for this current study was to show that we can improve from 20% to more than 60%. We have been quite modest. What we wanted to show was more than 60%. We are fairly confident that we will be hitting the same targets that we have seen in the West in the range of 70 to 80% in terms of responses. But what is more important, and this is where our pediatric colleagues, Dr. Khaira and Dr. Sunil Bhatt in the audience will know, is how it will impact on the future for ALL treatment. For example, I'm not saying this, but this is for example, you have, and this work is already starting, and I'll show you in the next slide, is in terms of your induction, 4-week course of steroids, tapering of 2 weeks, maybe an additional treatment with inotuzumab, maybe not, and then consolidation with CAR T. Your entire treatment is completed in 3 months. At the moment, your conventional treatment is 2 years for girls, 3 years for boys, unless of course you intern with the allogeneic transplant. So this potential algorithm is what is going to be completely disruptive in how we care for our patients with BLL. And we are fairly confident that the numbers that we're hitting for responses, 80%, 90% will be higher than that, given that we can use early use. And we know that using these technologies early on will help prevent some of the relapses. And sadly, I cannot show some of the more advanced data because obviously we have to show it to the regulator. But eventually, in the next few months, we should be able to share more data in terms of PFS and OS. So this is where I was mentioning, if you look at the Cassiope study, TISA cell, and they have been trying to use as early as in high risk end of consolidation treatment in MRD positive. So similarly, the IMAGINE study will offer TISA for MRD, offer 1M capitation for patients who are MRD positive post-induction. And looking at BNHL, we know about from a year ago, the data from the Belinda Zuma 7 and transform study. And of course, the whole algorithm is evolving because we know how poorly these patients do with the salvage, especially the second line post-rituximab era. The responses that we used to see with DHAP, HAP is long gone because rituximab somehow has edged over and we don't see those kind of salvage responses. So your second line is essentially 1M capitation autoleucel for patients with B-cell non-Hodgkin's lymphoma. And this is where we are. This is not the end of the story. This is the end of part 1. The story continues and hope Dr. Pindarkar, we or Emmanuel can come back and share with you the part 2 and the ongoing story. Thank you.
Thank you, Dr. Kamath. We'll just take we want to keep to the schedule because the basic issue is at 5:30 we have presentations from US. So we have to balance our time in such a way that we don't, at least that time we cannot change. If any quick question to Dr. Kamath, please. Yes.
So in any of those patients was there any case of antigen escape, CD19 antigen escape? And second part is, what was the proliferation of these CAR T cells? Did you follow up the proliferation, persistence, particularly persistence of these cells?
Right. So I'm sorry, the slide deck has been shut, but if you remember the table where I showed the 77.1%, and if you look at the bottom, there was a figure with the B cell aplasia, which was the red line touching the bottom, and the blue curves, which are up, that was the in vivo persistence. So that was the 1 captation autoleucel levels in the blood. So the persistence is, we have not reached the median yet. In other words, as the trial progresses, it is more than 90 days. I will not be able to tell you the exact median right now. It's evolving data. It's 3 months plus. And your other question was, have we seen CD19 negative escape? Not in this population. There is, but I'm not saying, because this data is the one that was presented to the regulator, but I'm not saying we do not have. So in this current cohort, we have had 2 CD19 positive relapses, but I cannot confirm or deny of CD19 negative. Yeah, thank you.
Thank you very much. And our next presentation is by Dr. Rahul Pawar on CAR T scenario in India, CAR T cell therapy in India. Dr. Rahul, please.
Can I share my slides?
Yeah.
Can you see the slides?
Yeah. We can see the slides.
Perfect. First of all, thank you, Dr. Pindakar, for giving this opportunity to us to present some of our work. And before going to my own work, I would really like to congratulate Tanveer and Shiva as well as Gaurav for excellent work they have been doing. Phenomenal work. So excellent, very, very big congratulations to all of you. And of course, there was a great talk by our Anil Kamath, and also congratulations to him. So I will also discuss some of our work which we are doing. And here, one of the major thing is that this CAR T, right from the product development till the clinical trial journey, all the work is done in India indigenously. So the product design end to end, this is really all the work done in the academia as well as now in the industry setting. Okay. These are my disclosures. I don't need to, I think, highlight that now to this audience that cancer is really rising even in LMIC, including India. Now we have more than 1 million cases each year in India, and India is also ranking second in the global cancer mortality rate. So there is a huge need in this global south, we call all the LMICs. When there's a huge need, especially if you see wherever the less dark is, that means although the cancer death annually per 100,000 people are less, but given our population is so huge, the number of deaths are happening very, very high. And here CAR T, which I think everybody already mentioned, the intent to cure platform, which is a living drug, is a personalized therapy at this moment. And if allogenic CAR comes by Tanveer and group, then we also can have off-the-shelf CAR. But at this moment, what we have is the autologous personalized therapy. And these are intent to cure platform because they can provide long-term cure. And they are giving very good responses. But also Anil mentioned that more than 70–80% overall response rate you see. So this platform is really an excellent platform. And I think a lot of good data now coming for the long-term cancer cure, even by UPenn group, as well as I think Dr. Brenjan's group. They all have shown excellent work showing that CAR T CD19 CAR works for long-term cancer cure. Right. And based on their pioneer work of Dr. Brenjan's, Dr. Carl Jones, their pioneer work led to all these approvals. And unfortunately, this drug is still not in many LMIC, including India. And their work also have this paradigm shift, the way now cancer is treated compared to the standard of care. Now you see the Axicell giving excellent advantages in this treatment of the cancer. And recent work by their groups also published that CAR-T is no more coming in second or third line or fourth line relapse, but can be given to patient, can benefit patients even early relapses. So even first line relapses, some of the patients can be eligible for the CAR-T. So now CAR-T can benefit more and more patients and can be given upfront in the treatment regimen. Right. And this platform so far shown remarkable success in hematological cancers, but I think now field is growing and it's an evolving field where CAR-T can be successful in some type of solid tumors. And very recent work by UPenn group showing that CAR-T or engineered T cells can even have a big impact in curing or in treating cardiac fibrosis like heart attack, which is another big problem in our country as well. So all this CAR-T work, as I try to explain that there is a need in the global south, but availability is in the global north. When you see this global map showing that all the CAR-T, in not only just CAR-T, majority of cell and gene therapy, it all exists in the global north, which is US, Canada, and now China is also picking up immensely in the cell and gene therapy space. Unfortunately, nothing in the Southeast Asia, nothing in the African continent, very little in South America. So where there is a need, these state-of-the-art or these intent-to-cure platforms are not available, and this is a concern, right? So our effort was to develop indigenous CAR, made in India CAR, because that's the only way we can make it affordable. If we import from any other country, license it from any other country, and then market it, it will not be affordable, or it will be affordable to only very, very few people. And that's the approach we taken differently, and I will show you the data. Okay. There are so many challenges in India. I will not go in many detail because Tanveer as well as Dr. Anil has pointed out some of these. But one thing I want to tell you, what is the most common thing in these cancers and relapse and refractory setting in B-cell malignancies is median survival is hardly 6 months in this part of the world. And what happens after relapse in Indian kind of settings, this may be very different from the developed world. And I think many of you are clinicians sitting there, you may also agree that this data, I took it a slide from Dr. Hasmukh Jain, who is the PI at Tata Memorial Hospital, Tata Cancer Hospital here in Mumbai. What he suggested that if you have a de novo DLB cell patient, 100 patients, 60 get cured, and almost 15 would be refractory to chemo and 25 will relapse. Out of these, again, 20 will respond, 20 would again get refractory. Within these 20, only 10, half of them will go for the bone marrow transplant, another half will not get this bone marrow transplant. Within this 10 patients, only 4 undergo remission and 6 again relapse. So overall, if you see, these are the 36, almost 40% patients who do not have a good option, treatment option, and their CAR-T can make a huge difference in our population. And based on this clinical need and also the huge burden on the patients, we wanted to have indigenous CAR, and the only way to make it is collaboration, right? And so we collaborated with clinicians, because I am being a scientist working in IIT Bombay, and we collaborated with Tata Hospital in Mumbai with Dr. Gaurav Narula, who is a pediatric hemato-oncologist. I'm sorry about this, slides keep changing. And Dr. Hasmukh Jain, who is the adult hemato-oncologist, medical oncologist, also in the same institute. And then we had a humongous help from NCI, Dr. Nirali, as well as Dr. Terry Fry when he was there at NIH. Both of them immensely helped us in clinical trial design and our clinical training. Dr. Steve Heifel and their group in the NCI, who does the clinical grade CAR-T manufacturing, they helped us in developing the processes, developing our infrastructure, and setting the standard that what CAR-T standard we should use so they are safe for the patients. And now we, along with a few of my colleagues, we founded this ImmunoAct 4 years back. for the taking CAR-T to many patients, in fact, beyond Tata Hospital in Mumbai, so that all the patients from all hospitals can get the CAR-T. Our innovation is, of course, in the technology as well as novel products. And we want to have with the objective that CAR-T should be at least less than $40,000 per patient. If that's the only way, or one of the way I would say, we will take to the many patients in India. Otherwise, they're very expensive. Again, many of our patients can't even afford it, and then dream will not be true. So last 10 years, it's been now 10 years, I have been working on this CAR-T project, and we designed many constructs of the CD19. They're all humanized CAR-T, and these patents are granted for some of the constructs. And one of them is now in the clinical trial. And long back, we did all the laboratory assays showing that these CAR-T, we can make it from the patients in the lab. They are killing the tumor cells selectively in an antigen-specific manner. They secrete cytokines, so they are functional. And we also perform, just like Tanveer, we have performed these animal studies where we have clearly showed in the left, what you see, these are the mice, which are tumor-bearing mice. These are the, I think, NALM6 cells, CD19 positive NALM6. And then when you treat these mice with the CD19 CAR-T, the tumor was almost gone, indicating that these cells are efficacious in the animal model. So in brief, you can say our CAR is a humanized CD19 CAR. It's a unique, patented. We tested in the lab as well as in the animal model. Patents are granted. And we were the first one in India who went to the clinic and treated patients with the CAR-T. And this work was published in already. And in fact, the cover image, what you see, this is the Indian CAR in the AACR journal. And I think then the most important, I think the significant milestone come when we treated our first patient of the lymphoma under the lead PI, the Dr. Hasmukh Jain and his team. So this was a phase 1 open label study with the 10 patients of the lymphomas using Scholar 1 criteria. Duration was 10 years, and sponsor was Tata Hospital, and research supports were given by ImmunoAct as well as by ICMR. I will not go in detail of how we screen the patients. Dr. Pindarkar said that we have to limit the presentation, but just to give you an idea that we have done this trial during the COVID time, so it took us long. And the second biggest challenge was the regulatory challenge to us, because we were the first one probably in India doing the CAR-T trial, and regulators asked us that we can only enroll our second patient when first patient is discharged. So when we finish our first patient, it takes a month, 2 months, sometimes 2.5 months, and then only we can have second patients enrolled. So that's why our phase 1 took quite some time, almost a year, but finally we finished it. There were like these 10 patients. And another point what I want to highlight to the clinicians, we have never shied away taking the patients who were even having bulky disease at the time of enrollment. And they all have, many have bulky, as well as they have extranodal disease, and we treated them. And we had basic characteristics, so like 8 male, 2 female, and these you can clearly see most of them were lymphomas, 3 were PMBCL as well. Many multi-line failures, like 5 of them were 1 or 2 line failure, or 3, 4, or 5, 6 prior lines of therapy failure. We manufactured the cells within 7 to 8 days. So the day of apheresis, if we consider day 0, we select and activate, transduce cells, remove the vector, expand the cells for 2 to 3 more days, and then cryopreserve and do the quality control. And we also infuse the cells in the split dosing, just like Dr. Anil Kamath in our lymphoma trial, at a 10% on the day 1, 30% on day 2, 60% on day 3. So this was for lymphoma, but for our leukemia trial, we also did only 1 dose instead of this split dosing. Then these are just some translational work showing what was the product profile. So we could make enough number of cells within 8 days. These cells were having CD4, CD8, and other naive central memory, and all different type of T cell subsets in the product. What was interesting so we treated patients in divided in 2 cohorts. One is less than 5 million per kg or greater than 5 million per kg. And what you see here, no matter what was the dose, even that higher dose, we had CRS only either grade 1 or grade 2 CRS. But there was no icons, not even a single. So we had 0% icons in our, especially the grade 3 icons here I meant. And these were almost 0 in our lymphoma trial. In the leukemia situation was slightly different. CRS, we did not have any grade 3 or grade 4 CRS in the lymphoma trial at any given dose. And I can tell you, we have gone even as high as 15 million cells per kg in the patients, but we did not see the CRS grade 3. Okay. So, so here you have the dosing I have mentioned here. So this is the column where I will just take the pointer used. But anyway, I can't see this thing. But if in all the patients, you can see we have CAR T either less or even high as 15, what you see here, 15, even 17. We did not have the grade 3 or grade 4 CRS, and we did not have grade 3 ICANS either. We had hematological toxicities which recovered after some time. Summary of this dose and adverse, of course, there has been hematological toxicities, but no ICANS of grade 3, only ICANS grade 2 in one patient, and the CRS was not grade 3, it was only grade 1 or grade 2 in about 60% of the patients who received greater than 5 million cells per kg. I will not go in detail of other these toxicities. If you have interest, we can discuss. But then we wanted to see early sign of this efficacy. And again, you see this patient has a disease and this was completely gone within 30 days. Similarly, another patient, you can clearly on the left-hand side, you see a bulky disease patient, and within 30 days, it was completely gone. The ID4, in next 2 months, we tried, but there was a minor disease left and it came back ultimately, and the patient is no more. So even if there's minor disease left, it relapses and then the patient passed away. If you see the overall in this 10 patient data, what you see for the greater than 5 million per kg, the 66% response, and the complete response is also 66% in the greater than 5 million per kg. If you see deep responses, so, until now, there are 4 patients, 3 patients are cancer-free, still surviving over more than a year now. The 1 patient, which was ID11, he was cancer-free, but died because of other disease. So these are still the patient surviving. Another patient, if you see the ID5, he ultimately, there's a PMBCL patient. So this is another very good example I want to share with you, that how we learn from the clinical data and then change something and then it starts working. So this was the ID5. ID5 had a lot of CARs in the blood. So even when we did 1 year almost CAR extension, we could find CAR T, but this person did not have the response. Disease was like progressing and the CAR was there in the blood. So I think Dr. Hasmukh then decided to give nivolumab to this patient at day 330, around 300 a day. And then patient undergone to the CR. So these are some of the cases. And then we also saw what few more patients, even ID12, who were progressing even on the, so the diamond is a PET scan date. So when he got the PET scan, disease was progressing, we reinfused the cell. And after reinfusion, we saw some CAR expansion, but it was not great. So even reinfusion sometimes doesn't help. That's another thing what we are learning. But when we give nivolumab, in this patient, it did not work. So 2 patients it worked, but other patients it did not work. So out of 3, so there's one ID14. Another example, you see that we have this another PMBCL patient. So somehow in PMBCL patient, CAR was there, but it will not work. But nivolumab, given nivolumab, 2 patients came under CR as well. So some of you were interested in how much the CAR-T persistence were there. So this is the profile where you can see the CAR copies per microgram DNA over the day 100. What you can see in the blue line, this is the mean CAR expansion in the patient, all these 10 patients. So mainly, if you see the responders have a lot of CAR, but in the blue, when you see there are few patients where CAR did not proliferate that much, and these were the ones who were non-responders. Okay. I will not go in detail that how the CRS was correlated with different kinetic or different cytokine. I think it's very well known that IL-6 is very much correlated with the CRS in the patient. So overall, if you see phase 1, primary objective, there were no treatment-related death in our all the patients. Cytokine release syndrome were either grade 1 or grade 2. And one of the major point I want to highlight for the clinical audience, that there was no ICU admission in the lymphoma protocol, which is very important in our settings when we are developing CAR-T. If we reduce the toxicity, we also reduce the financial toxicity to patient. Because once patient go into the ICU setting, they ultimately start spending a lot, which is a big problem. Toxicities, grade 1, grade 2 CRS, grade 2 were managed by the tocilizumab. Another trial, just briefly, I will mention this was the pediatric patient clinical trial with Dr. Gaurav Narula and his team at ACTRAC has done from the pediatric patients. They screened 34 patients, infused ultimately 6 in 2 different doses. Leukemia in the pediatric population, we only use 1 million cells per kg or 3 to 5 million cells per kg body weight. Here, these patients, again, these patients had as disease burden, you can imagine, median was 32% blast in the bone marrow. So again, we did not take only MRD positive disease. We took the very high disease burden. Even there was a patient who had 82% blast, as high as that possible probably. So these really were difficult to treat patients. Still, doctors took them in the protocol and they treated it. So that shows that Indian CAR can be efficacious and safe even when you treat at very high disease burden. So here is the data of the toxicity. So all 6 patients developed grade 1 CRS. Grade 2 was in 1 patient, and grade 3 also 1 patient developed grade 3 CRS. And this was that the patient, I think, who was a very high disease burden patient. And he was the one who developed grade 3 CRS as well as ICANS, and ultimately did not survive. And when you see responses in all 6 patients, we had MRD negative CR was in 3 patients, MRD positive CR was in 2 patients. So total overall response was in 5 patients, 83% response, overall response. Patients are still 2 patients is alive at 11 months as well as 6 months post CAR-T, and we could still see some CAR-T cell in these patients. So 2 patients under deep remission. And here, I think one of the person asked the patient, what is the CD19 negative relapse? We had, I think, 1 CD19 negative relapse as well as CD19 positive relapse in one patient. So now we are recruiting the phase 2 trial. We are supposed to recruit 50 patients in the adult, and all kind of B-cell malignancies are there. And we have already finished 11 patients enrollment, manufacturing of 11 patients. The good news is that based on our learning from last 18 patients we have done in our phase 1 trial, now we can manufacture the cells within 9 days. Our vein to vein time improved hugely, because if you have noticed in our phase 1 data, sometimes we have 3 or 4 months waiting from vein to vein, but now in the phase 2, within 27 days of enrollment, we are infusing patients. That means our system is in place now. Once patient get consented, enrolled, now we can do all apheresis, manufacturing, everything is on time. Within 27 days, we can infuse a patient. So overall, the key conclusions are this indigenous CAR T platform is a clinical stage and is designed to clinic, all done here. First CAR T therapy is safe as well as shows early sign of efficacy. I have not presented what the view what we have, how to take it beyond Mumbai, how to take it beyond Tata Hospital or the West Zone. We are proposing there's a regional decentralization model that will ensure that affordability as well as the access to all who might benefit from these intent to cure therapies. And lastly, I would like to thank all the mentors, all the regulators who are very vigilant about how the clinical trial is progressing, funding agencies, as well as our investors who now invested in us, and all the hardworking research team, clinical team, but most importantly to our patients who put their hopes and trust on us that Indian CAR will come one day, right? And finally, my team at IIT Bombay, as well as all the collaborators at Tata Hospital, NIH, Carl June, as well as Avery Posi, both are in our scientific advisory board of ImmunoAct. Dr. Satwa Nilapu, who did the CD19-C28 trial, he's also in our scientific advisory board, an entire ImmunoAct team which is committed to take the affordable CAR T to the Indian patients. And thank you all for listening to me. In case you have questions, I will be happy to answer.
Thank you very much, Dr. Rahul. And I think there are a lot of questions coming in from the internet, and the major questions are related to cost. Is it likely to come down? One interesting question is, do we still need a transplant after CAR T treatment or after CAR T relapse? Do we need the transplant before CAR T? So what should be the relationship of CAR T and a transplant in relapse refractory ALL, for example? So these are some of the questions. These questions, I'm sure, as we progress, they will be discussed because they are really common questions which will be coming out. They will be probably already being discussed in our next presentations. So, but at the same time, we'll definitely take up some more questions. We have actually crossed our time for the discussion we had. We were supposed to finish by 3:40. So I would request now to move to the next presentations so that, as I said, we are on time at 5:30. So, and Rahul, thank you very much for giving us the insight. And I'm sure we'll have some questions and we'll come back to you. So the next presentation goes to Dr. Sunil Bhatt, who has a good experience of clinical experience of using CAR T in Bangalore. And I'm sure his presentation will give us more insight.
A very good afternoon, and thank you very much, first of all, for having this important session. And it's been a pleasure to listen to all the previous speakers, especially from the lab aspects of it. It's a mixed audience. We have got colleagues from the lab, from the industry, a few clinicians, and I think this is going to be a purely clinical talk. What I'm going to be, I think there's a lot of overlap, but I think partly because of our ignorance, what's been the full agenda like. So I pardon me, there'll be a few things which would have been probably discussed already. I would try to kind of brush them pretty fast so that we are catching on the time. So I think this we have seen why we're discussing this very important topic today. And we have also discussed partly on the evolution of CAR. And one important thing I wanted, kind of highlight here is that, it's been 80s and in early 80s and 90s that a lot of work from Israel had basically been started on the use of on the CAR T, especially from the animal experiments. And then, of course, we saw then 2000s showing the work being done in the humans, and the first FDA approval in 2017. The core of CAR T therapy is this transduction, and we heard Tanveer and Rahul mentioning about it, and I think I'll skip this. And but for the clinicians, actually, I think this was discussed a few times in the last hour or so, and that's the generation of the CAR. And I think just made it a little simpler here. The first generation CAR had only the binding site and a stimulatory molecule, which is the CD3 zeta. But then the second generation CARs, if you see on the cartoon here, that there was a co-stimulatory molecule which is added, CD28 being the most and 41B being the most common one, and OX40 also in some of the CAR constructs. And the third generation, of course, having a combination of the co-stimulatory molecules, and that's important because the co-stimulatory molecules do help, which will show in the later slides, at how they are important, both of the persistence and the responses, and how that plays a role in the clinical aspects of managing these patients. So I think this again has been shown what are the different products which have been approved both in the ALL and the lymphoma field, and also some of the products which have been approved in myeloma field. And I think Anil discussed the unmet need. There are thousands of patients in India, actually. If we look at the data from the ICMR, which of course underrepresentation, there are thousands of patients, both adult and pediatric, both in ALL and NHL field, would require those are relapsed refractory, wouldn't have an option for them, and CAR-T form is one of the more important as well as only option sometimes to be able to manage them in our clinical practice. Now, getting on to the the nitty-gritties of ALL, and that is we have come across, if we look at chemotherapy only in relapsed refractory settings, in pediatric practice, for example, relapse leukemia is the fourth common malignancy. ALL is the commonest malignancy, and almost 10 to 15% of them will relapse, and that from the fourth common malignancy in children. And when we treat them, if we look at the intention to treat analysis, we would be able to cure maybe 30 to 40% of them, even after the relapse. And if we look at the graph here, the new molecules which have come up, the BiTE antibody like blinatumomab, and also the CD22 antibody, the inotuzumab, they do help in some of these disease conditions in relapsed refractory settings. But if you see the graphs really merge later on, so they give temporary benefits, they do temporary responses. But if you see that the graphs later on are merging, so chemotherapy versus monoclonal antibodies, BiTE antibodies, they actually do not have much advantage in the long-term survival. So that although these antibodies would be a important they are important tools for us to get the patient's MRD negativity and transplant, but by themselves they are not sufficient enough to give sustained responses. So how does in this now disease group the CAR-T kind of plays a role? So the first trial which gave actually approval for this for the Novartis product, and that was the Ileana trial, and which we have been kind of studying and quoting for many years now. So they enrolled around 92 patients, out of which 75 under infusion, and these were young pediatric and young adults. And what they showed is that with the use of this second generation CAR, almost 70% of the patients had a response at 6 months, and almost 50% of the patients had response at 12 months. So half of the patients actually at 1 year were disease free, and about 70% of them were disease free at 6 months. And the same thing was parallelly happening in the adult field, and that was the ZUMA-3 trial, which was again a CD19 trial. And what they showed in this disease group, in this study group also, was that almost 70 patients were enrolled in this trial as well, and they showed similar results in adults as well, almost 71% complete responses, and that response was durable at 1 year. So just to summarize these very important trials, Ileana trial, at EFS and OS at 12 years, we do have a long-term data as well now, which will be shown a little bit later on. EFS and OS of about 50% and 76% respectively, and the ZUMA-3 had 71% at what is 18.2 months, and this is actually another multi-center trial which had pediatric and adult patients all showed similar results. So in summary, these initial studies showed us that we could cure half of this very heavily treated difficult patient group with CD19 CAR, both in adult and pediatric settings. And that, of course, led to the approval of one of these products. Now, if we summarize now some of other CAR T products with different co-stimulatory molecules with different vectors, we see the responses here, which has been pretty standardized in most of the studies, has been CR negative rates, MRD negative rates, and they are in the range of around 80 to 90% at 1 month, and that kind of is around 70-75% at 3 months, and that has been pretty standard in the disease in the various studies. I'll leave this because I think there's a lymphoma talk after this. This is data on NHL. But one of the important things from the clinical point of view is that it's a multidisciplinary team approach. There's many people involved, whether it is screening the patients, whether it is treating them or a bridging therapy. Lab is a very, very important component of this whole treatment, and of course quality control, as well as looking after this patient during the treatment and follow up. So it's a village which takes to bring the CAR T therapy actually to the bedside of the patients. Which was also discussed by Anil briefly, was that there are various toxicities. Some of them we kind of are learning while we are treating these patients, and the two important ones of those are cytokine release syndrome and the ICANS. Also B-cell aplasia, because I think it is a silent immunodeficiency state underlying the patient. I think we are knowing them more and getting better and better. And I think that was the whole reason, probably, which Anil also highlighted, where we fixed to, we stuck to the hyperfractionation protocol. And that was also, I think, when Rahul was presenting his data, that, the patients who had high disease burden did have higher, CRS rates, and of course, leading to poor outcomes. So various toxicities, I won't know the details of that, but for the clinical, from the clinician's point of view, these are some things which you need to know from the different time points. CRS comes early, neurological a little bit later on, and neutropenia infections kind of persist through the month, immediate 1 month post CAR T, and persist for a few months as well. And there are different, it's like a transplant kind of diagram. We see this very common in transplant, but also in CAR T era, we also have some of these infections at different time points and need to be aware of those. Now, few other aspects of clinical aspects of the treatment, the CAR T in ALLs, I think was discussed by Tanveer. That was persistence of CAR T is very important in context of durable responses, and various factors will contribute to that. He has actually highlighted some of the factors related to the CAR T themselves, but there are some other factors which would of course play a role. One is what co-stimulated molecule has been used, and we know that CD143B persists longer than the CD28, although CD28 has a very robust and initial responses. But it's also very intriguing that sometimes and this has been shown for the CD19 relapses, the post relapses, that the CAR Ts don't persist. However, CD19 CAR negative relapses, there may be no correlation between CAR T persistence with compared to the relapse. The other aspects which have been shown to play a role in the persistence has been is the disease, the disease status at the time of CAR T infusion. If you've got a more disease burden at the time of infusion, you'll of course CAR T persistence is going to be longer, and that is kind of self-explanatory. However, that may also relate to higher toxicities. Lymphodepletion forms a very important element in persistence. So the standard lymphodepletion, fludarabine and cyclophosphamide, there are the various regimens, and that has been shown now that's an important element of CAR T administration. It's also been shown that the CAR Ts which are more humanized may persist probably longer. However, this has to be seen in real life scenarios as well. But the so-called murine CARs or the chimeric CARs probably would persist lesser as compared to the humanized CARs for obvious reasons of immune rejection. Now, one more point is about prior treatment of blinatumomab for clinicians. I think we all have access to this now. It's a CD19 BiTE antibody. What happens to the patients who received prior blinatumomab before getting CAR? Ilana trial didn't actually take patients who had prior blin exposure. Zuma 3 did have some of the patients who had prior blin exposure, but there's a study we need to probably take in consideration where we are talking about where the blin use has been under consideration. Now, we all know that patients do relapse even after CAR T, and one of the mechanisms of relapse is CD19 negative relapse. The patients, the cells stop expressing CD19, and how that can happen is something called as alternate splicing. The B cell just shed the CD19 from the surface, and there's tumor, and because the CAR Ts don't work on them, and that's tumor escape, and that will lead to a relapse. There are also something called as immune pressure, which can build and lead to CD19 negative relapse. And what happens here is that there may be occasional cell which may not be presenting CD19. All other cells are killed, and the CD19 which was not expressed on the surface would have a survival advantage, and that lead to full blown relapse. And there's also, which I think Tanvir also showed, there can be a lineage switch. An ALL cell can become an AML cell, shed off the CD19 from the surface, and it really doesn't work. And also mechanism of trogocytal cooperative killing. What happens here is that the CD19 is actually transferred by the smart B cell onto the CAR T itself, and the CAR T cells start killing each other, and the B cells are kind of happy and start proliferating again. So this is another mechanism of relapse, and so these are some of the mechanisms which we have to deal with in context of the patients who are not responding and they're lapsing even after the CAR T therapy. I think a question was asked probably on which Dr. Dinesh just mentioned, was a role of transplant in CAR T era. So what are the patients who do we need to transplant all the patients? Do we need to transplant none, or do we need to transplant some of them? So it's a it's $1 million question. I think everyone wants to answer. And I just show some data. This is the data, long-term data from the from Nirali Shah's group, which was actually CD19 CAR using CD28 co-stimulating molecule. What they showed this data set is that they had treated some 50 patients, and what they showed is that the patients who transplanted had an event-free survival of 62% after CAR T therapy, and the patients who are not transplanted, all of them relapsed. So in this study, all the patients who didn't have transplant relapsed, but this was a CD28 CAR. This is another. This is the Ileana trial long-term data. They had few patients who were transplanted, few patients were not transplanted. What they showed in this group is that in this data set was that there may be some role of transplant, was not very clear because not all the patients underwent transplant in Ileana trial. And there's another study where there was a 50 transplant eligible patients, and then they showed is that the patients who had transplant did better than the patient who did not have transplant, but again, it's a heterogeneous group. So to summarize this all now, there are some risk factors now we need to consider, which are both pre-infusion and the post-infusion. Pre-infusion risk factors are what the disease burden, what is the potential molecular makeup or the genetic cytogenetic risk factor, as well as the type of CAR which has been used. And the patients who are high risk, that means patients who will lose the persistence of CAR T very early, who lose their B cell very early, those who have a high disease burden and refractory disease pre-transplant, these are the ones who will probably require transplant post CAR T therapy, and the others, which are the low risk ones, probably we can leave them just with the CAR T therapy. So this is, I think, evolving field, and in the next few years, I think we'll get more and more better in understanding which are the type of patients which would require a transplant, and CAR T would be used as a bridge, and which are the ones where we probably can expect a long-term cure just with the CAR T therapy. So now, what are the? I think Tanvir just touched it briefly. I want to get into this: is how do we address the CD19 relapse in the B cell ALL era, B cell ALL arena? We do have CD22 dual, tandem, sidecar, tricar, and sequential infusion. So this is example of the dual CAR. There is two different CARs on the surface, tandem, side to side, 3 different CARs, and also something called a sidecar. We can have an option of co-infusion and sequential infusion. CD22 CAR data is out there, and it's about 73%, which is almost same as CD19 CAR, and same as 1 year leukemia-free survival, about 70% in the data which is out there. Dual CAR, there is some data already there. I won't get the details of that. And also for bispecific CAR T therapy, there is data. But as you know, as was just Anil was mentioning, that the data from all these dual CARs, tandem CARs is still, I think, evolving. We are getting around 70-75% long-term responses, but I think still to be seen how that's going to be unfolding. It's not resolving the problem completely. I think there's still a lot of learnings from there. There's a concept of the sequential CAR-T where you can use sequential infusion of first you do CD19 CAR and then do follow infusion of CD22 CAR, and that's I think one of the other mechanisms which can be used for addressing these lapses. Now just a word on the T cell CAR, because ALL is B cell and T cell. B cell we have evolved quite a bit, but T cell there are few problems, few challenges. One is T cells are not unique, markers are not unique to the T cell leukemias only. So we do have normal T cells expressing, and there is a T cell aplasia which can be life-threatening. There's also something called as fratricide, where the expression of the CAR T cell by the CAR T themselves, we eliminate each other. So there's they will kill each other, and that's nothing much gonna be be achieved in this. And autologous CAR from the peripheral blood may also contain probably malignant T cell lymphocytes, and that probably can also be a problem. So these are the challenges with T cell neoplasms or the T cell ALLs. There are various mechanisms to address it, but the most important one I think has been studied is use of suicide genes. So you do the CAR T, and then once they've done the job, they kill them. Second, of course, is limited percent of CAR. So you program the CAR in a way that they are active only for a short duration of time. And very important in the T cell CAR T arena is that we need to consolidate all these patients with a hematopoietic stem cell transplant for immune recovery after the T cells have been knocked off. So these are some of the mechanisms of allogenic CAR is off the shelf CAR. Again, challenge of graft versus host disease when somebody else's CARs are being used, and that can be again addressed by disruption of the TCR on the surface, which may prevent the GVHD. It's maybe an important strategy for our country, because you can have one CAR product that taken from a donor, and that can be used for multiple patients, that can bring the cost down, and that probably once it's available, it may address some of the cost issues. The last phase of my this thing was to kind of dwell on our experience. I think Anil has discussed some of it, so I don't want to get into the details of this. As he said, this was based on this phase 1 trial which was done from Spain, and we have in-licensed the vector from there, and this is our trial which is open at the moment and almost about to finish enrollment. I am going to stress only on the B cell component of it. That's the leukemia component. Dr. Sharath may be touching on the lymphoma component, and we used basically all relapsed refractory B cell ALL cases. As I said, Blinna was exclusion in this, but we also took patients who were MRD positive and not only with frank disease, and that's important, because those patients otherwise do not have any other option. They can't even proceed for the transplant. This is the response data which he showed, and I think I'll take 1 minute. Response which he showed, which was present in the ASH this year, and what we could show is that in the B-ALL cohort, at that point of time when we had about 72%. This is MRD negative at day 90. 72% response on B-ALL, pediatric cohort about 80%, adult cohort about 50%, but that's changed now with more patients being enrolled. Overall response at day 28, of course, in the B-ALL was about 86-87%. Very acceptable toxicities because of the vaccination. And this was a question I think asked by Tanvira, that was the persistence of these CAR-Ts. So we did, there is some data on the persistence of CAR-Ts, about 15 odd patients, which was analyzed at the time of this presentation at the ASH, and this is the summary of that. So the red ones, as Anil was suggesting, is the B cell aplasia, and these are the graphs for the persistence of the CAR-Ts, and we could see persistence of the CAR-Ts up to 6 months and beyond. But again, this is the data which is evolving. So to conclude, and this is the first in India phase 2 trial, and we manufactured the CAR-Ts on a closed GMP closed system. It's feasible and rapid. We were able to manufacture CAR-T between in 9 to 10 days time and infuse in our 12 to 14 days on an average. Median expansion after CAR-T has been 10 days, and the persistence has been up to 6 months and beyond. And interim responses which we showed here were deep and are obtained are deep. So, of course, CAR-T established in India requires investments, infrastructure, training, and regulatory approvals, and that's where we are here. And we are learning as we go along. With that, I thank you for your attention. I'll be able to take any questions if you have. Thank you very much.
Thank you very much, Dr. Sunil. And I would request Dr. Samir to come forward.
Thank you, Sunil, for this enlightening talk. I think we are running over time, so we will probably go to the next talk. The next talk is by Dr. Sharad Damodar from Bangalore, and he'll be talking on progress in cell therapies in lymphomas. Over to Sharad Damodar.
Thank you, sir. I'd like to start by thanking Dr. Pendarkar for actually organizing this wonderful meeting here, this session. Can you all see my screen?
Yes.
Yeah, okay, great. Okay, so I'm going to quickly run through the next 10 minutes because you're running short of time as to what's the progress which is happening in the area of lymphomas. And clearly, what the scenario is in the world and what's the work which is happening in India, which you've already seen from Dr. Purwar's presentation. He presented the lymphoma cohort from there. So there are review articles coming up from our own country. People have spoken about the pheresis procedure, which is quite simple and has already been discussed, I think, in the morning sessions. Essentially, you need a manufacturing unit, GMP facility. As of now, you've got transduction, you're doing cryopreservation most of the time, lymphodepletion, and then infusion, and we're monitoring for CRS and ICANS. People have already discussed the various generation of CARs. We've spoken about the first, second, third based on the co-stimulatory binder here. There's also the fourth generation CAR, which uses the NFAT as an additional enhancer of the signaling domain there. A lot of CAR-T work going on in diseases other than lymphoma, leukemia. There's a big list of them, a lot of interest even in that area, which will again soon be relevant in our own country when we go beyond discussing this. So my job for the next about 10-12 minutes is to quickly run through in the area of B-cell lymphomas, what is the scenario here when you're talking about second-line therapy, and where does CAR-T then come in the relapsed or the refractory setting? Clearly, the initial data came from the first 3 pivotal trials, which was the Zuma one, the Juliet, and Transform. We had Axicell, Tisacell, and Lisocell. People have spoken about the various differences among them based on the co-stimulatory domains there. Clearly, the retrovirus was used for the Axicell and the lentivirus for the other 2. So some differences there. But the gist of this slide is to look at overall response rates. And clearly, you can see an overall response rate of about 70-80% in the Axicell and Lisocell, which is a little lesser in the Tisacell. So clearly, you're already seeing differences between the various CAR-Ts in this initial data itself. And then if you look at progression-free survival, 36 months, 33 months, 40 months, overall survival at 24 months, again, 50, 40, 50. So 2 of them are matching each other, but the one is a little lesser. And that was kind of standing out there. And the need to thereby look at more data from here and also in a more randomized fashion. So this is the updated data now. Zuma 7, Belinda, and Transform. And I'll just kind of highlight the little differences here so that, we put things into perspective when we interpret this data. So Axicell gave a fixed dose of about 2 into 10 to the power 6 cells per kg. This was per kg dosing, whereas Tisacell gave a dose of 0.6 to 6 into 10 to the power 8 cells. And here again, in the Lisocell, again, it was 1 into 10 to the power 8 cells. So just as a particular range of cells versus the per kg dose here. Otherwise, most of them were similar. There was leukapheresis done at randomization here, at randomization here, and before it was before randomization here. One second. And bridging therapies, if you looked at in the first trial in Zuma, only steroids were allowed, whereas in the other 2, you had whatever chemotherapies also which would be allowed. The other difference was crossover, which was not allowed in Zuma, which was allowed in the other 2. So all this has implications when you look at final subsequent analysis of this data. Now, what was the actual numbers? So this little table shows you the actual numbers here. Neat data here, 180 in each arm in the Zuma, about 160 each in Belinda, and about 90 odd in the Transform. The GCB subtypes were about 60% in the Zuma study, whereas it was about 30-40% in the Belinda. A little difference there, here not reported. If you look at the refractory to the first line, about 70% across the 3 trials were refractory to first line of therapy. So you have a population which is in that initial refractory subset. What about the response rates here? So if you look at the overall response rate and CR rate, similar to what I already mentioned, you're again seeing that same difference here in the final reported data, which is the phase 3 data. 80% about overall response rates in the axi-cel when compared with standard of care, which is the autologous stem cell transplant, high-dose chemotherapy followed by that. 80 versus 50 and then 65 versus 32 here. Here it is kind of matching each other. You know, the CAR-T data and the standard of care data is almost similar. Whereas in the lyso-cel, again, you're getting that difference of 80% versus 48, 66 versus 39. So clearly there's something ongoing here, even depending on the type of CAR, maybe the subset of patients, whether you allow bridging therapy. So various things come into play when we need to analyze this data and thereby read into that. What about CRS? We all spoke about CRS. About 90% got it in the Zuma study, 60% here, 50% here, lesser here. Neurotoxicity also was higher here. All of them used at the full dose, not like the fractionation, which was discussed in the previous 2 talks. So 60% CR rates here, TOSI usage about 65% in the Zuma study, about 30% in the other 2 trials. If you looked at anemia more than grade 3, about 30-40% range. Thrombocytopenia, again, variable here in these various studies, closer to about 30-40%. Grade 3 neutropenia, about 70% and 50% in these 2 trials. So what they then recommended was that if they came up with this algorithm on how would you place CAR-T in today's world if it was available easily, this is what they suggested, that generally when you look at relapse rates in non-Hodgkin lymphoma, the first, the relapse within the first about 1 year, it's about 70% of patients. And the subsequent relapse is about 25-30%. So those are the ones which you could be chemosensitive because it's a longer time frame, whereas the ones who relapse within the first 1 year are more likely to be chemorefractory. And thereby you make these 2 little divisions here of these population of relapsed lymphoma patients. So the ones who have relapsed more than a year later will be responsive to chemo and thereby actually will be eligible for a transplant. 50% of them will actually then go on to have a transplant, and in transplant, you will get another 50% of them who will actually respond, 40-50%. So this algorithm was also projected earlier. So this is the gist of what happens when you have a transplant. So if you started with about 100 patients and about 25 of them were beyond 1 year relapse, and they had the standard of care, which is now, you'd be left with 5 or 6 patients who will actually have a cure at the end of all of this. Whereas if you had the access to CAR-T, which is in about 70% of patients, of that 70 patients, about 50 of them would be eligible for a CAR-T, not have comorbidities, which would be kind of, keeping them away from it, which would still be there in about 20-30%. And if you got the responses, what has been described in at least the 2 studies so far, and you get about 40% response rates, you're looking at least about 20-25 patients who would be eventually cured with the CAR-T cell therapy arm here. So that's the realistic way of putting the data here and looking at how we would actually approach this. May or may not completely apply to our setting, but clearly once the cost of CAR-T comes down, I'm sure we will have access to this. Now, my next 2-3 slides will quickly tell you about what all is there in the data here and how the Cochrane library has actually put together all this data and come up with some of these caveats which they've actually identified. So these are all statements from there. I'm just putting it down so that, it puts things in perspective. They've identified about 13 uncontrolled studies till date. There are about 38 odd ongoing studies, including the RCTs. The overall risk of bias was high for all studies, in particular due to incomplete follow-up and the absence of blinding. And the evidence is kind of low for all the outcomes. I will just read out the highlights here. Evidence is uncertain about the effect of CAR-T cell on overall survival, uncertain about quality of, CAR-T cell effect on quality of life. PRMs have not been uniformly reported. So have the adverse events, which have been not uniformly there on the SAE front. So they have a little bit of uncertainty there is what they say. There have been incomplete reports on the PFS, DFS overall survival. It's been a little gray on some of the studies. So putting the gist of it together, they're saying that there's a little bit of uncertainty which has to be clarified in more randomized control studies to give us definitive evidence of a clear-cut outcome here. And this was what they concluded. Available evidence on the benefits and harms of CAR-T therapy for people with relapsed refractory DLBCL is limited mainly because of the absence of comparative clinical trials, and we should be guarded in light of this with conclusions to be drawn from this. So that's the gist of what they said. Now we will quickly go on to my last few slides, which are on our own data, which has already been presented, which was presented at ASH. I think Anil alluded to that. So this was the data which was actually put up from the clinical side. And Anil has spoken about the co-stimulatory domain and the A3B1 binder and the business of using fractionated cell dosing, which has clearly resulted in lesser CRS and ICANS in this particular subset. The initial data, which was actually presented both in the combined subset of ALL and in the non-Hodgkin's data to look at response rates. And this was the initial, inclusion exclusion criteria, which have already been spoken about. I just wanted to mention about the target dose in the lymphomas cohort, which is 5 into 10 to the power 6 per kg versus 1 into 10 to the power 6 for the ALL cohort. The fractionation was the same, 10, 30% and 60% within that first 10 days from day 0. So this was the initial data which was actually presented for the ASH. We have not yet kind of published the rest of the data, where you had 16 people who were enrolled and it was 8 and 8. And 10 patients actually were dosed. The manufacturing time of 12 days, overall response rate at day 28 was 80% and 77% at day 90. So this is the actual data which was presented there. And this is the subset analysis. A lot of it is the ALL data. I will come to the lymphoma data in the subsequent slides. Persistent to CAR-T, which I think even Rahul spoke about, at day 28, 89%, at day 90, 50%. And this is what is panning out even later. This is the actual patient data. You can see there are about, out of the 11 here mentioned here, there are about 5 NHLs there. And I will come to that little response a little later. This was the results as far as the AEs which were reported. So we did have CRS of about 70% grade 1, only 10% of grade 3, overall of 70%. ICANS was just 10%. Neutropenia, almost grade 3 was 80%, overall 100%. Anemia and thrombocytopenia overall, again in the 100 and 90% range, but the grade 3 were definitely lesser. Median onset and duration of CRS by about day 6 lasted for 2 days. 60% of them got tocilizumab. There was one death, so which was a 10% TRM in this subset, which was reported. There are some other AEs which have also been reported as per data from all the other studies, and it compares with them. This is the lymphoma subset where you actually look at the PET images. The day 90 response rates in both these patients, you can see completely clearance of all this disease, even in the high tumor burden, which is what was demonstrated in the lymphoma cohort. This is the summary of all that data, which actually re-stresses the response rate at day 90 of about 77% and about 80% at day 80, with the TOSI usage and CRS of about 60%. So this was the conclusion, proving the safety and efficacy of this particular product which was used. There is more updated data which will come up with more patients being enrolled. I think there are close to about 10 additional NHL patients who've been enrolled. And the same data with the percentages which I've put up is actually panning out even in that cohort, which will get presented in the subsequent meetings. This was the first infusion which was done there. Now I'll quickly run through some data on what is the actual problem when we look at these studies. How many of them will actually go on to treatment when you look at our standard of care arm to put the perspective of what happens when you do a CAR T-cell randomized trial. So here in the first one, which is about 300 odd patients, each one randomized equally about 160 each, and about 98% of them could actually go on to the subsequent arm which they were supposed to get. About 102 in the standard of care, which was supposed to get the autologous transplant, actually discontinued without receiving a transplant. So at the end of the day, 155 went on to get CAR-T, about 52 only proceeded to the autologous stem cell transplant. So this is the reality in the world. Here, this study allowed crossover so they could actually come on to back to the CAR-T cell arm and get further therapy. But otherwise, in reality, this is what happens. So a lot of patients will not be eligible to undergo that. And thereby, obviously, the final cure rates there will be lesser. I will skip the demographic data slides. So in this, which was the second study, where they actually said that both of them are equal and concluded that there was no difference between the two when you compare Tisa-cell with standard of care. This is that data which was published. However, the conclusion from the Axi-cell data is totally in difference to this. Here they got a significant difference when it was compared with standard of care. And I will show you that data again, where about 180 patients were randomized to each arm. Again here, 88 did not continue subsequently because they could not be eligible for the salvage chemo. So only about half of them actually went on to get an autologous stem cell transplant. And eventually in them, once they actually, some of them required a second salvage to be eligible to for the high dose therapy. So 170 were compared with 60 at the end of the day. And that's how their efficacy analysis was done. And when this data was analyzed, then you could see the clear difference here, Axi-cell versus standard of care. Clearly the line separating out, hazard ratios again clearly in favor of the CAR T-cell therapy. And then overall survival and progression-free survival again, spanning out there. So that was the gist of it. I will end here so that I'm well in time to what I wanted to actually convey. There are some questions we could take it a little later if time permits, but otherwise that's the gist of what is actually the reality. But clearly with more and more centers taking it up and CAR-T being available, I think we've got a really good chance to make sure that our patients will have access to this, because it's actually a small subset of patients who actually eventually go on to high-dose therapy and an autologous stem cell transplant to achieve that cure rate. Thank you.
Thank you, Dr. Sharad. Thank you. We may have time for one question, if there's any. Yes, please.
Sharad, thank you. It's Anil. As audience knows, Dr. Sharad was the PI for adult patients and Dr. Sulin for pediatric patients. Also, we have Dr. Pankaj Malhotra and Dr. P. Raja, respectively. The question for you, Dr. Sharad, is you shared the data.
I think you can speak from here. Maybe he will hear it better.
Sorry, I'm not sure whether you heard the earlier part. The question I'm asking is, can you share your clinical experience in terms of treating the patients compared to the, let's say, autologous haplo and sibling allos in terms of managing CAR-T? Because you are one of those very few lucky physicians in the country who have got their hands wet treating patients with actual CAR-T. So in terms of your experience with Warnin, Captage, and autoleucel, and the take-home message from those your experiences, is that something you can share, please, Sharad?
Yeah, just to put it in a nutshell, Anil, to answer that, I think at this point in time, with this particular CAR and the fractionated dose, it's been easier than an autologous stem cell transplant. You know, so no complications of the haplo, CRS, ICANS, especially in the lymphoma subset. When I talk to them in the adult subset where we've done it, it's been very smooth sailing. And like I told you, I put up the data that overall, when we put all the data together, our TOSI usage is about 60%, which compares with all the rest of the thing, but hardly anything to worry about. So I think that's one of the good things which I want to highlight.
Thank you. Thank you, Sharad. We will now hand over to Dr. Dinesh Pindarkar.
First of all, thank you, Dr. Sharad, and thank you, Dr. Samir, for conducting the session and for really taking time out and coming here. So our next presentation is by Dr. Priyadarshini Chatterjee on CAR-T manufacturing, which are the challenges.
Hi, good afternoon, everyone. So the topic of my discussion today is not new to most of us here, I think, who have had the opportunity to work with CAR-T cells. So we know the challenges to a large extent. We know we've strategized and we have kind of tried to overcome in many situations. And the discussions on these challenges and the way forward kind of go on. So the presentation today is to kind of take these discussions forward and try and see what we could do or what we are trying to do as the field in terms of CAR-T, managing the CAR-T challenge of manufacturing. Manufacturing, one of the most important challenges, and that's the challenge that the first thing comes to mind when we talk about CAR-T is about the cost of the CAR-T. So the cost of the CAR-T essentially comes from its basic raw materials, which we know apart from many other things, many other accessory things. But this manufacturing part, which we talk about the consumables, is something where our hands are really very tight. And for countries like us, where we kind of have to import these consumables, many of these consumables, which are GMP grade consumables, this becomes even more of a challenge. The other challenge that we kind of know currently is that for most of the CAR-Ts, I'm not talking of all CAR-Ts, but most of the CAR-Ts, the manufacturing time that we talk about vein to vein is about 3 to 5 weeks. And that's essentially keeping not just the patient waiting, that's one of the major challenges, but it's also essentially about the cost running up, because you kind of have to keep your cells in culture for so long. And you kind of have to essentially meet whatever dose you've set out to meet. And that kind of tells you that your consumable cost, your cost in terms of your facility running, everything kind of comes up. So what can be done? So these are the problems that I'm setting out, and we are going to discuss in the next couple of slides as to what people are trying as a field, like I said, to kind of circumvent some of these problems. So when we come to the cost of manufacturing and essentially the manufacturing timelines, what I see in the field is a very, very interesting interested pursuit that people are having now is to kind of reduce the manufacturing time. And they kind of say that the patient's body itself can be the bioreactor for itself. This is a very, very interesting concept. And it's not just the big players in the field like a Novartis or a BMS doing it. There's so many academic labs that I see these days coming out with this concept. They say that you can, if you look at the literature that's published, you say you can manufacture it in 2 days. And in some cases, you can just bring it down to about 24 hours. Now, what do they do so magnificently different that we as most of the larger crowd have not been able to do is basically they have found out the basic science that kind of rules the CAR T. They say that it's not just about the dose that you give to the patient, it's also about the quality of that one CAR T cell or the few CAR T cells that you want to give to your patients. So when we talk about the quality, what do they essentially mean about the quality of the CAR T cell? I'm going to discuss that a little more. But just like I said, it's not just about the quantity, it's about how potent your T cells are. And when we come to the potency of the T cells, there's a lot of discussions going around as to, I think even Dr. Kamath mentioned, how to keep them more naive or more stem-like. We talk about stem cell memory. How can you keep your cells more stem-like in their nature so that when you give them to the patients, what happens is when they encounter the antigen, they have the power to proliferate. They have the power to essentially do what they're supposed to do, to proliferate, differentiate, and attack your cells. So what's happening is instead of making them ready to act in vitro, ex vivo, outside the body, you kind of give the cells the power and you give them into the patient. And then the body of the patient, the cells do what they're supposed to do, essentially differentiate and then attack the tumor cells. So this concept is becoming very, very numerous names out there, T-charge, next T, fast T. But this is something I think we kind of can look a little deeper and try and see what's the real biology behind it. And if improvisations can be made, can we really do that here in India as well? So just like I said, the timelines can be brought down, they say, from about 2 to 6 weeks of production to somewhere around, say, 24 or 36 hours of production. This is being ambitious, but then there's data that supports the ambition. So this is not undoable. Now, what this essentially shows us is, so if you look at the stem cell, if you look at the stem cell memory here, so this is data from Novartis, their T-charge platform. What they're trying to say here is if you compare the first bars, which are the apheresis product, as compared to the final product, they're kind of being able in this mechanism to produce cells which are more stem cell memory-like. They have more cells which are central memory-like. And these are the cells, I'll just talk about this, the more towards the left-hand side of the development, which is the early developmental stages, the cells are, as we all know, or many of us know, are endowed with the property of proliferation, lymphoid homing, and essentially better metabolism. So your cells are more powerful by themselves. So the numbers don't become as important in that case. You can probably shrink the numbers a little bit, shrink your manufacturing timelines a lot more, and essentially deliver the cells which are more potent in nature. So this is just elucidating the same thing. The other thing is that while we do that, while we kind of try to make cells in a very short time frame, the other important thing to see is the reagents that we use. Are they going to actually push our cells towards more differentiation, like more towards the effector memory and the effector cells, the terminally differentiated effector cells, or can you have mechanisms, you have biology which kind of pushes the cell even during manufacturing, not allowing them to come towards the last or the later phases of differentiation. And cytokines out there, this is nothing new in what I'm going to say. People kind of have done this along, but just to kind of emphasize that this is something that we can think along to try and come up with reagents which kind of will be more helpful in this kind of thought process, like using an IL-7 IL-15 combination versus an IL-2 in many cases have been shown to kind of achieve the same potency of the cells, but not really drive them towards exhaustion, not really drive them towards terminal differentiation. I know many people in the room here, they've been employing IL-7, IL-15 mediated activation and transduction platforms. The other thing that becomes very essential and very important here are other reagents that people kind of come up with, like retronectin, which also has been shown to do a similar thing, where kind of to try and maintain the naivety of the cell, the stem cell-like nature of the cells. So these are reagents which are out there and have been discussed and utilized. But my thought here is that we can always kind of think through for more of these mechanisms which kind of help us retain the cells in towards the left side of the quarter, which we talk about the naive and the stem cell memory. Now, what kind of stays as one question is, as we move outside our comfort zone, where we have had the experience of growing cells for like 2 weeks or 3 weeks in culture, have the cells which have a balance of effector cells as well to a large extent, and which is going to just function when you're giving it to the body. As opposed to that, now you're talking about cells which have to differentiate in vivo. Now, when you have this balance, one needs to actually strike and kind of come up with mechanisms where you know the dosage now that you want to kind of give your patients to, when you're talking about more stem cell-like cells, as opposed to a mixture of when you have effector cells as well to take care of the immediate functionality inside the body. So that is something that's kind of, it's being done because there are trials from Novartis. There's trial with the FAST-T, which the CAR T called the FAST-T CAR Ts, which have shown that there are doses which are possible to kind of achieve a good response rate. The other important thing which we all know, and there's nothing new to this, but this is something, and sadly that's not something that we can do much about, is the variability in the starting material. The variability in the starting material, and the other thing that I would want to emphasize is about this, the supply constraints. It's not just about the T cells that you get, which are variable in terms of their responsiveness, but it's also about the numbers. You often don't get enough T cells. And the other challenge that I've been hearing a lot about is the supply of the vectors, the GMP vectors. And I know there are many centers which have addressed this. This is a more relatively easier thing to kind of work on, because you just need to have more people kind of come and show interest and come into the GMP-based lentiviral manufacturing. But the difficulty, like I just said, stays on in terms of your, I'm going to discuss about one way out here where we have a variable starting material. Now, when we talk about these two, what people have also tried to do is that when we have variability in terms of taking pan T cells, like all different kind of T cells, is there a possibility of selecting out? People have done selection of specific T cell subsets. There are pros and cons to this, but then there are certain groups that kind of say that while your initial population is going to be huge mixed population of cells in terms of their active state, in terms of their numbers, their proportions, if you're starting with, say, a central memory T cell, and you kind of try and see whether that T cell is your starting material, and if it kind of gives you a little better homogeneity, then is there something worth trying? So this is kind of, as I said, there are pros and cons to this approach, but this is something that the field tries. The other thing, which is not really a manufacturing trouble, but it's more of a logistical challenge many times, is whether we can really work with the frozen starting material. And while these are very basic, difficulties that people have faced, and many of them they've kind of showed, many people have shown that actually when you talk about frozen and fresh PBMCs, in many cases, the initial starting problem is there where you have less viability when you're working with frozen T cells. But we in our lab have also established that actually over a period of 2 to 3 days, those cells can actually come out of their state of a cold shock, and they can pick up and they can do equally well. So it's not really a manufacturing challenge, but it's a logistical challenge related to manufacturing that can help us kind of get more flexibility in terms of logistics. So coming back to where we just left, the other thing that I think one of the common challenges, what I started out with, was the manufacturing failure essentially is because of our inability to a large extent to produce the dose. We just discussed about if we can dose it at numbers which are more potent than the quantity, if that kind of helps us. But cell expansion, even if we don't take what's there in terms of a fast T or a T charge, cell expansion is still something that needs a lot of attention. And I think a lot of discussions that go on is about how to improve the cell expansion capabilities. I'll discuss about some of the modes that people have tried for improving their cell expansion, while the other problem that stays on in terms of cell viability. In the CAR T cells that are being produced, many times that doesn't meet your specifications and you have to release an OOS product. When we talk about the cell viability here again, people have seen that your autologous plasma sometimes, because these are all autologous therapies that we are discussing. So autologous plasma in many cases works better than some of the commercial products. But having said that, cell viability continues to be a challenge for many cases. And this is again something when you're doing the expansion, if you're really exhausting them a lot, if you're making the cells effector-like, then that kind of challenge of your cell viability becomes even more. The other thing is because of transduction being a natural viral infection, it's kind of mimicking the natural viral infection, the T cells have a tendency to kind of destroy or kind of eliminate the foreign DNA, the double-stranded DNA in most of the cases. So what people have also tried these days is to use inhibitors that kind of will block the natural mechanism of these T cells and help you get or achieve better transduction. The next thing that we're going to discuss about briefly is about the scalability of the process. Now, this again is something that I personally feel is very, very critical, because while we are able to manufacture some CAR T cells, meet the needs which are there for some small number of cells, as and when we kind of address, because this is an autologous product, as and when we have to kind of address questions, when we have to address masses or large numbers, then we come into a lot of bottleneck. And this bottleneck is natural, because what happens is currently when we talk about the bioreactors that are available, there's so many commercial all-in-one bioreactors that are available, but each of these bioreactors are essentially for one patient. So what happens is that you really can't scale up at that level. So what people have tried to do is to kind of have what they call process treats, which is essentially to have a semi-automated mechanism. So you have bits and pieces of automation, which is merged together with a lot of manual processes. So what happens, each equipment does its job to the maximal. And then there are stages where you have bioreactors or human intervention, where you essentially can actually marry these two together to get a better productivity with your limited set of equipments that you have. So this process treats kind of are something that holds a lot of promise for the future, is what people think. The other important thing is when you really move into commercialization stages, what do you really do? So what is the thought currently is that you have multiple small manufacturing units, very small automated manufacturing units, along with some of these process treat units, where you kind of have some small bioreactors coupled to certain equipments that are there, which are commonly used by those bioreactors together. And if you have this merge coming in, that makes your commercialization or meeting of high doses or high numbers of productivity much more feasible than it would have been otherwise. But the challenge here is you make these procedures or processes. The important thing is, how do you do your QC tests? You have to have the QC tests also to be very automated and very regularized. So what people are focusing these days a lot more is to see, because many of these QC tests are essentially, we know it's flow cytometer based, it's qPCR based. So how do you do these samplings? And also when you do the sampling, you're taking out a part of your cell, especially when you're talking about cell numbers, which are very restrictive. What do you essentially do? So this is something which is interesting. People have talked about fluorimeter based detections, which can be a little bit faster than flow cytometer based discussions. But these are areas where we probably need to work up as scientists. So what the innovations can probably focus on to is not just the science, but what we're doing together with that is the engineering. I'll just take a minute more maybe. So one of the other things, and I think many of you would have discussed this earlier, is essential trying to see that if there's an answer for some of these questions, moving away from the autologous therapy and coming into the allogenic, not saying that the allogenic doesn't have its own challenges, it has its own bag of challenges, but many of the things that it can address is also not to be ignored. Cost being one of the major things, your timely production and your variability, lessening the variability being the other very important pros. So having said that, I would like to conclude here, and thanks a lot for inviting me for this talk. Thank you so much. Bye. Any questions, or should we just move?
Yeah, all right. This was. So, thank you very much. And my next presentation, we are just making some changes and trying to, because at 4:40 we had a presentation from the regulatory DCGI office, and we're just trying to connect.
Presentation, I have opened it. Share screen. Share screen, I'm sharing. कर रहा हूं मैंने, but it is not sharing. Select a tab to preview. No. Window, entire screen. Achha. No. No. How it is to share? Just a minute. I will call somebody.
So Dr. Basu is from DCGI. want to discuss the regulatory. Yes, we requested them to give some regulatory requirement for CAR-T therapies. Yes, sir, it's visible now.
Yeah, huh? It's now visible.
Yeah, it's okay, sir. Go ahead. Oh.
This is my PPT. Screen, I already opened it. Share button. I put that share button.
It was visible earlier, actually.
Desktop option. You see Chrome tab, window, and entire screen. Entire screen. Okay, entire screen. Then share. Share system audio. No, it is not happening. Which one? That share. Entire screen. Choose what to share. There are the three options: Chrome tab, window, and entire screen. Okay. Now in the below it is share system audio that I clicked. Then cancel box is blue lined. That is a blue screen with a upward arrow that I have already done. But why it is not clicking, I could not understand. Why it is not clicking? I'm clicking it, but it is not taking. The whole thing I should close.
अभी स्लाइड देखी थी एक बार. अच्छा वो अपनी वाली. क्या करें? कंप्यूटर का कंट्रोल ना करो. So एक काम करते हैं. We'll start the next presentation, and if required, I think let's start our next presentation, because otherwise, see, we don't want to miss the 5:30 target. That's really very important for all of us. So I think I would request. That. कैसे करना है? Okay. Voice. Can you hear us, sir? Sir, you will have to unmute yourself.
Now?
Yes, sir. Go ahead. We can see the slides.
And my face also.
Yeah.
Okay. Now, how to this slide moving?
It's again gone, actually.
Just a minute. Can you wait, please? I will take some assistance from.
I think I would request our next speaker to go ahead, because we really are short of time, because we really don't want to miss the next targets. So I would request Dr. Vasundhara to do a presentation on CGT platform by MedTherapy Biotech.
This one.
Good evening, everyone.
Just a minute, please wait.
Good evening, everyone. It's a pleasure to be here with the experts in onco and therapeutics, and me, Vasundhara Shukla, presenting here MedTherapy in front of you all, which is focused on cancer gene therapy, making it affordable and accessible globally.
Okay, no problem. So now it is visible, na?
Our mission to conquer cancer, it has been our global mission, because we want all eligible cancer patients to be able to afford and access cancer therapy. Currently, a cost of $500,000, it's unaffordable to patients in developing countries. And so we look forward to join hands together for this great global mission. It's all started in 2013 when a 6-year-old girl, Emily Whitehood, was diagnosed with blood cancer and was mentioned to live for a few months. She became the first patient to be administered CAR-T therapy developed by Novartis. Today, she is a 16-year-old girl cured of her cancer. I am proud to share that our founding members were part of the first CAR-T developed and approved for Novartis, Dr. Bikash Verma, Dr. Vladimir Slipushkin, and Dr. Daniel Diagno. Successful therapy as CAR-T is associated with some of the crucial challenges listed here. I'm going to touch base for manufacturing as a major component cost component, and the critical problem with manufacturing CAR-T therapy deals with long time and high cost. So being a focus as a manufacturing being a focus as a cost component, it's very important to give you a brief about our strategy. We are Boston and New Delhi based CAR-T therapy group founded by CAR-T experts from Harvard and former Novartis executives. We have world-class global team and next generation technology for CAR-T. The existing challenge, be it cost, manufacturing, and relapse in patients, MedTherapy is on our way to solutions by next generation cancer gene therapy products. Our technology and business model dramatically reduces manufacturing cost and time. This can't be achieved by a single entity. So a global collaborative team comprising our partners in US, UK, India, and other countries are involved in this effort. A full spectrum of integrated end-to-end solution will be provided by MedTherapy. So if you can see, and we have seen since the afternoon, that the complexity is there in CAR-T manufacturing, be it viral vector manufacturing and CAR-T manufacturing. According to that, we have seen that there has been an exorbitant cost as a major driver for the therapeutics, where here you can see the segregation of cost, which says that over a 40% of cost is on the manufacturing part, be it equipments, be it reagents, or be it overhead for manufacturing equipments there. Therefore, we have developed our proprietary next generation manufacturing process, whereby we have innovated each step, which you can see here. We have innovated each step, which will be dramatically reducing the cost of the therapy. We have developed the next generation viral vectors to lower down the cost of CAR T therapy, and the one person who is behind this was Dr. Vladimir Slipushkin, the pioneer in T-cell engineering. These are some of the projects ongoing with our partners. We are having a co-development on these projects, showing you the different stages here. We are coming up with integrated facility for developing a complete spectrum of end-to-end solution, being it our unit in Boston as well as in New Delhi. We are self-sufficient CAR-T company whereby we have full control of all the three components of CAR-T manufacturing process, be it vector, be it product, or be it our upcoming clinical trials here. Addressing these all global problems, this is very important for us to be globally connected with our partners in US, in other countries, be it Africa, be it UK, be it India. So with that, I conclude my talk on mentioning that we have our collaborators. We are thankful to our collaborators, our partners, the patients in India, UK, US, and Africa, and the rest of the world. MedTherapy here mentions that we will be the pioneer company for providing affordable and accessible CAR-T therapy worldwide. Thank you.
Thank you very much. So now we really have a capacity also to manufacture on a license basis. So maybe a lot of you who are really developing products, you can use this facility to manufacture. So if somebody has a construct, they can use that construct and deliver the product to you. So I think it's a good concept, and I'm sure this concept will be utilized more and more, especially by clinical trial agencies where the people may have a construct but may not have the manufacturing facility, the GMP manufacturing facility, which is really a big task.
Start my video. Just look at, do it.
So
Now
we
it's
are
okay.
going back to our, thank you very much. And we're going back to our lecture on regulatory.
The screen is visible now?
No, sir.
No, I am visible, but my share screen, share. This one.
Yeah.
How to share that? No, I think. One second.
Yeah.
Now it.
So other screen is opened up. I think we'll have to open the PPT.
So we can see your screen. You have to minimize this mail and open your PPT. Sir.
Yes,
You click there double.
Yeah.
Then share. Now it is available.
Yes. Sir.
Yes, it's available now.
Now I have to see my screen. No, that's slides.
Just make it full screen, sir.
Slides.
Yeah, slides option.
Let it be as it is. Okay, let's go ahead.
No, my share slides. This slide you keep. Okay, fine.
Okay.
Fine. I can move it this way. So now I am visible and audible. I hope.
Yes, sir. Absolutely.
Please respond it. Thank you. So I am from the regulatory perspective. I was earlier attached with CDSU headquarters, and I worked there for many products like blood products and recombinant DNA, etc. basically the stem cell products that came later stage only. And after that, gene manipulated product and many others, xenografts, etc. came. So, but now I am in AMTZ, Andhra Pradesh, Visakhapatnam. Am I audible and visible, my slides?
Yes, sir.
Yeah, I am worrying every time whether it is going or come. Thank you. So regulation of the medicines and medicinal products in India, that depends on the basic principle I want to share here, that the age-old Drugs and Cosmetics Act that was framed in 1940 before independence of India, and the rule that was also framed simultaneously in 1945. So those are the backbone, and under which we tried, our Ministry of Health also trying to fit even medical device and stem cell products both, because these are the two new emerging areas. Now, regulatory principle is by system of the things like licensing and permission and periodic inspection. And there is a sampling of products and testing of the labs. Now, these two, the colors are different from red color, sampling of the products and testing at labs. In this case, like stem cell or in the CAR T cell therapy, it's individualized, autologous. So in this case, how it is possible, there is a question and challenge. And let us talk about the other things like Drug and Cosmetics Act 40 and Drug and Cosmetic Rule 1945. Within that, Schedule Y was basically a guideline that was adopted around 1989, and that actually revolutionized the approval process of new drugs. And basically, new drugs are the molecular entities which are, or the recombinant technology-based products which were not approved, extensively used in a country in India, but maybe approved in USA or Europe. So there is a process that was adopted. I'm talking about the history of the in how to enter into this emerging field of the stem cell therapeutic or dendritic cell therapy, or the this CAR T cell therapy recently. Now, the new drug clinical trial is the new version of this Schedule Y, and many things has been adopted. By that time, clinical trial related, there were a lot of debates in Supreme Court and ethics committee and the patient's protections, and how the informed consent form, informed consent process, etc., to be adopted in the rule making process. That those things were taken care, and finally 2019 new drug clinical trial rule was adopted. So in that, the features are 13 chapters there, and schedules are there, and rules 107 and form 17 are there. The another rule 2017 we adopted, that is medical device rule. Now, the basic regulatory pathway, in fact, in all the countries around the world, is that company will generate data, company, or it may be an individual person's effort. So they will do the R&D studies, preclinical studies, and then the product, the new one, the innovated product that goes into the phase 1 and phase 2, phase 3 trial. All these data are submitted to the regulatory submission to the national regulatory agencies of various countries. In USA, it is US FDA. In UK, it is MHRA. And Visa in Brazil, or TGA in Australia, and India, Ministry of Health and Family Welfare, DGHS. And the small wing is CDSO within that. So there you have to submit, and they have their own procedure. Obviously, TGA's method of examination and evaluation is different from the US FDA and Europe. So, but anyway, there is an approval process. Now, in our case, CDSO, we give licenses, first permission and then license for import and license for manufacturing. But after that, the story doesn't end. The thing is that phase 4 studies has to be done, because during clinical trial, phase 3 clinical trial, only limited number of subjects are taken care and taken as a subject in the trial, and that too with all blood parameters, urine parameters, cardiovascular status, everything is taken as if it is good, and then they are taken. So when the product is marketed, approved in India, the whole country, there are questions that how it will behave in the geriatric patients or children, because during clinical trial they were not the subject. So phase 4 studies are also important, and active surveillance studies are also important. Active surveillance study is a continuous monitoring of the product in the country. Now, CAR T cell therapy, as we all know, and many, I think, speakers have already discussed, chimera. Chimera is the Greek word, the incongruous monster, a god. So chimera, from chimera it has come chimeric antigen receptor T cell therapy. T cell is therefore it is an immunological treatment of the basically now it is approved for the cancers. Patients, and around the world there are a lot of research are going on. Patients T cells, a type of immune cells, are changed in labs so that they bind actively into the cancer cells, and that leads to the destruction. By morphological modification of T cell, its binding ability is increased basically. So now it is approved in USA for the adult B cell NHL and childhood CML, chronic myeloid leukemia. But that too, I had a doubt that why it is so that after the unsuccessful treatment of the with the standard treatments of cancer drugs. Now I am going to talk about the legal provisions and what are the regulations presently available to us. That is MCI Code of Ethics regulations and clinical trial requirements. Screening committees are present there, and licensing procedure, application review system, and guidance for industry. How much time can you please tell me? If it is, I should not delay it. Am I audible? Hello?
Yeah, yes, sir.
How much time is allotted to me? It may be around 15 minutes more.
No, sir, 10 minutes.
10 minutes, okay. I will proceed for immediately. So legal provisions is cell biological products are cell biology based products are now considered as a drugs. Drug is defined in the Drug and Cosmetics Act in a section that is 3B1 and 1 to 4. So under that definition, it fits. Now, ICMR published the national guideline for stem cell research that was in 2017. I think many speakers have already discussed about that. And that guideline entails the restriction and prohibition or permission of the research along with the minimal and substantial and major manipulations. Further, the indications which are not requiring any prior approval, that is also given in that guideline. And that guidelines, Annexure 3 is the area where, like stem cell, one of the example is stem cell transplantation in certain blood disorders that is exempted from clinical trial. Now, conduct rules of the Medical Council of India and for doctors is also governing this treatment of the using stem cells. The MCI codes, certain codes are here that no exaggeration or nor the minimization of the gravity of the patient's condition in clause 2.3. Then clause 3.2, this is, I'm talking about MCI, although it is not in our jurisdiction, but I just thought that this can be picked up in these slides. So maybe many speakers have already discussed about that. Consultation for the patient's benefit. Benefit is the utmost importance, benefit of the patients. So physician, as far as possible, they should be frank with the patient and their attendants, telling the whereabouts of these therapies and the situations. Now, fees and other charges to be displayed on the board of the doctor's chamber and hospitals. This is also a MCI clause. And prescription should also make clear if the physician himself dispense any medicines. And clause 6.1, physician shall not make use of the subject or patient's name or in conjunction with his own name for advertising in such therapies. Another important point is that all cell-based treatment should not be taken as a standard practices under the MCI regulation. That's a very important point here. Standard practices, which is already known, established over the years, doctors are practicing tamoxifen or docetaxel. Those things are the cancer products. And what would be the dosage and what would be the dosage schedule? Everything is standardized here. But in cell-based treatments, one should not consider that or should not be biased or over enthusiasm that CAR T cell therapy will perform well with a belief, 100% belief. That is the MCI's regulation is good point here. Now, definition of the new drug, as I told earlier, that drug which is not extensively used in India, but might have been approved and used, let's say 2 years or 3 years in Japan, Australia, or New Zealand, and USA, UK. Also, that has not been recognized as effective and safe by the DCGI's approval mechanism process. So for the proposed claim is important. The statement of claims indication is very important. Now, in case of the new drug, what documents, and for the CAR T cell therapy, of course, the CMC data, preclinical and clinical data on safety and efficacy data has to be submitted to the DCGI. Now, that data is evaluated by a committee, that is CBBT DEC, actually cell biology based therapeutic drug evaluation committee, but this committee has now been shortened. The name has been changed into IND committee for stem cell production, human cell products been done. But earlier in 2012, there was a press release, I will show you in the next slide, that this committee was formed to as an umbrella award for taking care of the all human cell biology based products. Now the screening committees are for import marketing authorization of the stem cell products. This committee, which was named as CBBTDEC, now named as IND committee, they are responsible. Some 40 to 50 experts pool is there, and it is taken care by taken from the microbiologist background or pharmacologist or immunologist, even the cardiologists are taken in that. And ICMR, everybody knows that National Apex Committee for the Stem Cell Research and Therapy, NAC-SCRT is there. And NAC-SCRT's homepage is this one. I think in this field, whoever is the stakeholders, they must be knowing. And a whole lot of experts and doctors, Dr. Anurag Agarwal, chairperson from CSIR Institute of Genomics, New Delhi. His photo is there. And function of the NAC-SCRT is basically not regulatory, but it is partially like general oversight of the field of the stem cell research and therapy in India, and formulation of the policy related to, and review of the controversial areas and ethically sensitive research proposals to be looked after by this committee, NAC-SCRT. And operations at the CDSU, how do we do it? We do it actually, basically we have done everything on Sugam portal, but this stem cell therapeutics or cell-based therapeutic or CAR T cell therapeutics, these are still taken by hard copy papers. I mean, paper-based documents. We are still taking. So you can take the acknowledgement copy of the submission of the at the DUC receipt section in the CDSU headquarters. And for a follow-up action, you can talk to PRO with a phone number and email IDs are there. And you can also directly straightway write to DCGI by email ID dci@nic.in. So agenda items for the committee, IND committee for cell-based product is circulated to the stakeholders, and minutes of the meeting comes up in CDSU web page. So for the indigenous manufacturer, there are certain forms, and 1, 2, 3, 4. This is the flowchart. Like indigenous manufacturer, a startup who is doing CAR T cell therapy, he has to take first the test license. Test license is basically a license for manufacturing limited quantity of the product or limited number of the patients, and that form will be given by DCGI. Now with the data generation, you have to come back to DCGI. Then DCGI will, I mean the IND committee will examine the CT protocol in depth, and then DCGI, they will advise DCGI to grant permission, clinical trial, to the applicant. So CT permission is the second phase. And the third step is the market authorization. If somebody wants to take the market authorization, then they have to apply in CT23 and they will get to CT21. So form number, it can be available anywhere, but basically there are forms through which DCGI operates. And to obtain manufacturing license, this is a large scale manufacturing, form 28. But at this stage, nobody has come in the CAR T cell therapy. Still it is going on. I like in CMC Vellore, Dr. Vikrant Matthew, he was granted the phase 1 and phase 2 trial, still going on there. So, and also Tata Cancer Hospital, there is another trial was approved, authorized by DCGI. Now for importers, any kind of import, the same thing, 1, 2, 3, 4 steps. One is the test license, take limited quantity, do studies, generate data, come back to DCGI again with a clinical trial proposal, I mean protocol with investigators list, etc. Take the clinical trial permission from DCGI, and then come back again with the larger data of the clinical trial and the test license data, preclinical data. And now you ask for the market authorization, and that is in CT20 by applying in CT18. And then there are two things here, the manufacturing, no question of manufacturing. Here is two more forms has to be taken. Was in import registration, another is import license by applying in the specific format. Now there was a PIB's Press Information Bureau's press release in 2012 for creation of the stem cell division in CDSU. That was in April 2012. This is the view of the PIB's government of India in 2012. That time Ghulam Nabi Azad Sahab was the health minister. At below end, it is written Ghulam Nabi Azad. So PIB's press release was in 2012 for creation of the stem cell division in CDSCO. So there are many things, therapeutic products derived from the stem cell, human gene manipulation, xenotransplantation technology, CBBT that has deliberated on the need for strengthening the regulatory agency DCGI by establishing separate wing. And accordingly, it was framed in 2012. So almost 10 years back. Now, for an ND, new drug, with including biologicals, discovered in India, you have to start with phase 1. So in case of the CAR T cell therapy, you have to start with phase 1. If IND, I mean, from other countries, it is approved, then you can start with phase 3. And if it is an arm of the Indian arm is there in global trial, then that data can be considered. But when the approval stage is coming, there are certain conditions are given, like PMA study you have to do. You have to submit 6 monthly PSUR reports, periodic safety update report, what is happening with the therapy, CAR T cell therapy, therapeutics. And so this is but I have certain points to raise here, that like the other drugs, pharmaceutical or blood products or biological other drugs, there is a batch concept and there is a certificate of analysis concept, and there is a parameter, specification, standardization of the product, those things. How a individualized CAR T cell therapy, T cells are basically the bioengineered products, augmented receptor binding. There may be 2 or 3 heads receptor binding that can attach with the cancerous cell and kill it, and still many fine tuning is going on. We don't know today what is talking about these 2 receptors. Maybe after 6 months there will be another subtypes will come up and that will be more activated. So the things are going in that way. So how the standardization of the product to be done, this is a challenge, biggest challenge. So thank you very much for giving me this opportunity. I am privileged to talk to you, the August gathering of our oncologists and doctors, I believe. Thanks a lot.
Sir, thank you very much for enlightening us on the regulatory issues related to CAR T cells. So this gathering is not only oncologists and hematologists, this gathering is all industry manufacturing today CAR T cells in India. So 6 companies and their scientists are sitting here and just deliberating on how we can take forward CAR T cells. So companies like, like Dr. Reddy's, Intas, there are many, I would not like to name all of them, but then they are all here sitting and trying to develop. So Immunact, Immunil, everybody is here. So basically, we want to understand how fast can we improve the process, and everybody is really very keen that we were just discussing in the morning that during COVID, DCGI did a great job by manufacturing the product, allowing it to happen, doing the trial, and actually in just 1.5 years, really giving vaccine to every individual of the country. So we covered 1 billion patients, literally, or individuals in 1.5 years. We investigated the trial, we made the drug, we approved the drug, we approved the trials, and we finished the usage. So we really expect DCGI that this speed would also be allocated to CAR T facility manufacturing and gene therapy, so that we can actually bring the product to the public as early as possible. Thank you very much. We'd like to thank you again.
The truth is that cannot be equated actually. Vaccine inactivation of the viral body is a established technology by which Bharat Biotech has developed. But CAR T cell therapy, it is like autologous. One patient's T cell, I am taking it, manipulating in the laboratory, gene insertion is there going on. Some proteins is emerging out of the cell and that is binding. So as I told, that batch release certificate, standardization and certificate. So how to go about the in-process quality checks in multi-steps. Those are the important things. So a little bit may be different, but we of course will try.
Thank you very much, sir. Thank you very much.
So should I log off?
Yes, sir. Thank you very much, sir.
Thank you. Thank you very much.
So our next presentation, the presenter from Harvard is already ready. But if we want, we can have a 5 minutes break and just get the tea, please. But we really assemble within 5 minutes because she is already there. So.
The Indian food is better here. Thank you for doing this.
My pleasure. I'm sorry I'm on Zoom. I couldn't arrange the flights and all.
Sure, we'll give you another crack at coming to India. You've been to India before?
Never.
Ah. So now Dinesh is really going to make it a job to get you to come here.
I have to apologize. I didn't know about the panel discussion, so I actually have a conflict at that time.
That's not a problem. I mean, we're delighted that you can talk. And I don't know if you saw much of the earlier presentations, but it's really quite impressive. I think, Dinesh, when's the first time you invited me to India? 2012, and they were light years away from where they are right now, which is really impressive. They have CROs that make cells. They have companies that are doing phase 1, phase 2, and looking to do phase 3 trials. Really impressive.
Faster than what you can do in Boston.
Well, or in the United States, actually, I think.
So I'm gonna speak. Thank you so much for inviting me, and I hope someday I'll attend this kind of meeting in person and see everything that's going on in India in CAR T cells. It's really a pleasure to be here. I'm going to talk about some of our recent work trying to understand the mechanisms that underlie CAR T cell therapies that drive CAR T cells. These are my disclosures. So when we think about T cells and how they work, I try to break it down into understanding what the driver mechanisms are when T cells become activated, when they encounter the antigen, with the idea that there may be opportunities to increase potency and responses and decrease toxicity if we can separate out these different functions and then perhaps manipulate them further in the laboratory. So as you've already heard, chimeric antigen receptors recognize antigen in the form of as a surface marker using a single chain variable fragment or some protein binding domain. And when the T cell engages with the antigen, the first job that we want, at least in the setting of cancer, is for that tumor cell to die. So we want the T cell to induce tumor lysis or trigger apoptosis in the target cell. As part of T cell activation, they will also produce cytokines that will in turn recruit other bystander immune cells, including macrophages and potentially other immune cells to carry on a further immune response and antigen presentation and so on. The T cell itself will also proliferate after having encountered antigen, and they will form a population of cells, and then a fraction of those will persist and perhaps form long-term memory. And so I would argue that at least in the setting of cancer, we definitely need the tumor lysis and apoptosis triggers. I would say it's unclear whether it's useful to produce cytokines or recruit bystanders. And we also know now, based on multiple clinical trials, that proliferation of the T cell is an important aspect of how these mediate clinical responses. And the degree of persistence and memory formation is at best debatable. It seems like in some diseases, it's important for the T cells to stay around for 6 months or longer. And in others, the T cells can do the job, ablate tumor. And if they're gone by 3 months, as in like in large cell lymphoma, you don't need to have prolonged T cell persistence. So let's break it down a little bit further. One of the things that we did initially is to start with our clinical patients. So I arrived in Boston about 8 years ago, before the first CAR T cell products were approved. And working in the Boston area, we were able to pool some of our experience from patients getting FDA-approved commercial CAR T cell products to then study those mechanisms from patient samples. So we took 32 patients who were treated with either Tisa-cel, which is the 4-1BB containing CAR, or axicell, which is a CD28 zeta-containing CAR. And we divided those patients into responders and non-responders. So here the T-cell patients are in blue, the axicell are in orange. And we defined response in this cohort as the patients who were still in a complete remission at 6 months. So that's not the same way that responses are defined in clinical trials. But what we really wanted to understand is what are the characteristics of the T-cell product and the post-infusion products in the patients who had essentially cures of their disease or long-term remissions of their disease. And we decided to take a single-cell RNA-seq approach to be able to identify differences both between the CARs and between responders and non-responders in our cohort. And so we performed single-cell RNA sequencing of 105 pre- and post-treatment peripheral blood mononuclear cells and infusion product samples. And then we also performed TCR sequencing to track the temporal evolution of the different kinds of T cells from our patient cohort. What we first recognized is that the transcript of the CAR was most readily detectable in the infusion product and 7 days after infusion. And this was true for both T-cell and for axicell. And so this gave us an opportunity to really look at not only the transcriptional profile of T cells in general in the patient, in addition to the rest of the PBMC population, but it also gave us the ability to sort of gate on the CAR positive cells that had a transcript based on their single cell RNA sequencing. And so if we look at the big data overview, this is all done computationally, which was led by Nick Hardvala and Gadi Getz at the Broad Institute. When we sequence 602,000 PBMC and infusion products, you can get sort of a big picture view of the distinct transcriptional phenotypes that exist among all of these different kinds of cell types. So if you look at all of them, you can see that the monocytes cluster separately from the CD8 T cells and the CD4 T cells and the NK cells. And you can see that the infusion product T cells, which are mostly CAR positive, actually cluster separately altogether. So that doesn't look like anything that's normally circulating in the body. I will also say that the CAR positive cells at day 7 cluster separately from the CAR negative cells, which are here in gray. So even after infusion, the CAR positive cells cluster separately on a big UMAP plot. So what did we learn biologically? Well, I'm not going to show you all of the data from this paper, which is quite dense, but the two major points that we learned biologically are that specifically in the T-cell product, when the CAR has a 4-1BB signaling domain, that the patients who were responders had a massive expansion of their CD8 positive CAR cells. This was not true for axicell. So if we look at the responding patients for T-cell, the ones that had more than 50% of their CAR positives being CD8s were the ones who were actually responding for T-cell. But for axicell, this was the same, whether they were CD8 high or CD8 low, it didn't really matter in terms of response. And the extent of the CD8 response was actually associated with the 6-month response in T-cell, which you can look here in the sort of highway plot of how the CD8s and the CD4s track after T-cell infusion in a responder versus a non-responder. You can see that when you have this persistence and growth of the CD8 positive CAR positive cells, which is the green part down here, those were the patients who were responders. And this turned out to be sort of mechanistically true in that we had a single patient experience, which was pretty unusual, but supported this hypothesis that the CD8 positive cells, CAR positive cells, were really driving the response in T-cell. So we had a patient who received T-cell for their large cell lymphoma. You can see their PET scans here across the top. And they initially had a response, but then relapsed within 6 months. And so according to our definition, this patient had not really had a curative response, not a durable response. And the patient actually had a second dose of their initial product that was already frozen. And so we were able to administer that product to the patient. And usually this doesn't work. Usually when you give a second dose, they actually don't respond better. But in this particular case, the patient had a prolonged durable response after the second infusion. When we looked at single cell RNA-seq of the product, the first product and the second product, they were actually completely overlapping because it was the same product. But what happened in the patient after infusion was remarkably different. After the second infusion, the patient had a significant expansion of their CD8 positive CTL CAR positive response, whereas that did not happen in the first time around. And by single cell RNA-seq, we saw that there was this remarkable expansion of the CD8 positive CAR cells, both by single cell RNA-seq and by flow cytometry. Oops, sorry. And so that really sort of suggests that the CD8 expansion was really what is driving the CAR T cell response in TISA-cell treated patients. This was, again, not true for axi-cell. The second thing that we found is that we saw that there was a significant increase in the number of CAR positive regulatory T cells in the infusion products of the patients who were non-responders. So this was true for both axi-cell and TISA-cell, but it was statistically true only for axi-cell, where the non-responders had higher frequencies of CAR positive Tregs, and this was only found in the infusion product. This wasn't detectable in the post-infusion products. But what we did is we also decided to go back and use some of our mouse modeling to see if we could recapitulate this in mouse models. So we took immune deficient mice and grafted them with lymphoma, and then gave those mice a 95%, and on the left, it's a CAR that has a CD28 signaling domain in orange, and then on the right with the blue is the 4-1BB based CARs. But we gave those mice 95% conventional CAR positive T cells, but then we added in isolated, sorted, transduced Treg cells from the same donor. So when those mice had 5% CAR Tregs that were purposely infused into those mice, we saw that the mice had initial responses, but also had this delayed relapse. So after around day 30 or so, the mice started to relapse at much higher frequencies when there was 5% CAR Tregs. And this was actually true whether the mice had 28 zeta CARs or BB zeta CARs. So although in the patients, we found that there were more CAR Tregs in the axi-cell treated patients who were non-responders, in the mice, we could replicate this finding whether the CAR Tregs had a 28 zeta or a BB zeta signaling domain. So I'm going to change gears a little bit and go back to some of our more basic sort of forward translation, where I've been talking to you about reverse translation and using the data from the patients to understand mechanisms. Now we're going to go back into the lab and try to sort of screen for potential mechanisms of resistance, particularly in solid tumors where CAR T cells have not yet had tremendous efficacy, although I'm going to have to eat my words a little bit, because just today's New England Journal of Medicine, or I guess it's last Thursday, the one that arrived in my mailbox today, as the first responses, as far as I can tell, in a larger study in neuroblastoma with GD2 CARs. So that's very exciting. I haven't even finished reading the paper yet. But as a rule, we've had several different CAR T products that have been tested in patients with solid tumors, and we don't yet understand what those mechanisms of resistance are. And so we decided to take a CRISPR screen approach to try to understand if there were any genes that were that, when knocked out or mutated, would confer resistance to basic CAR T cell mediated killing. So how does the tumor cell actually die, and what genes are necessary in the tumor in order for it to be susceptible to CAR T cells? So this work was led by Becca Larson, a graduate student in the lab at the time. She's now defended her PhD, moved on. But what she did was she took a brain tumor cell line, U87 glioblastoma cell line, transduced it with the Brunello lentiviral guide RNA library. And the Brunello library is a library of lentiviral vectors that encodes every single, it encodes 80,000 guide RNAs, and each guide represents a single gene in the genome, and there's redundancy in the system. So there's 20,000 genes in the human genome. There are 4 guides per gene. So altogether, there's 80,000 guides in this library. And she transduced the glioblastoma cell line, selected it for puromycin, which is part of this selection just to encode the guide RNA. So now there's a library of brain tumor cells, each one of which has a single gene knocked out in it based on the guide. And so we then divided this into an experimental arm and control arms. The control arms are to make sure that you have adequate guide representation. So every guide is represented in the library, and that we confirmed that by sequencing. We also performed a control arm where just knocking out some genes may increase the proliferation of the brain tumor cells. So we wanted to normalize for that, because what we really wanted to know is which genes, when knocked out, would confer resistance to CAR T cell killing. And so the experiment was to take this library of tumor cells, expose them to CAR T cells, in this case targeting epidermal growth factor receptor, or EGFR, using a CAR with a BB zeta signaling domain. We expose those tumor cells to the CAR T cells for 18 hours and then let them recover after the exposure for 48 hours. And so the glioblastoma cells that were resistant to CAR-mediated killing should be then increased in terms of their representation in sequencing of that entire group of cells. And so then we identified the top hits. So which genes, when knocked out, were highest represented after exposure to CAR T cells. The top hit was EGFR, which makes sense because this is the gene that encodes the target. And so if there's no target, there's a lot of resistance to CAR-mediated killing. But the rest of the genes that were in the top hit were actually all in the same pathway. And that was interferon gamma receptor signaling. So interferon gamma receptor 1 and 2, which is a heterodimer, and then its downstream signaling molecules, JAK2 and STAT1. And so this seemed to be the primary genes that conferred resistance to CAR-mediated killing. This was done in a screen format. So then the next thing is to validate it in a series of single gene hits, which we did both in vitro and in vivo. And just for the sake of time, I'm going to walk you through some of the in vivo testing. So we took a second glioblastoma cell line, different than the one we'd started with. This is U251, injected it intracranially into mice and treated those mice with wild-type EGFR CAR T cells. And what you can see is that when the tumors are wild-type and you treat them with EGFR CARs, you don't cure the tumor, but you get a difference in terms of both the progression of the tumor as shown by bioluminescent imaging across the top, and also you see an improvement in survival when you use EGFR targeted CARs compared to untransduced T cells. When you knock out EGFR out of the tumor line and then you inject the mice, now there's really no difference between giving the mice EGFR CARs or just untransduced T cells, and you see complete overlap of both the tumor progression and the survival curves. And when you knock out interferon gamma receptor, again, you see complete overlap. So it looks a lot like EGFR knockout or target knockout. So these tumors are resistant to CAR-mediated killing. Now, this was true not only for glioblastoma and the EGFR CAR combination, it was also true for glioblastoma with a different CAR targeting IL-13 receptor alpha-2. It was also true for pancreatic cancer using EGFR CAR or mesothelin CARs. So two different antigens, and this was true across not only these two solid tumors that I'm showing you here, it was also true for breast cancer, ovarian cancer, lung cancer. Across all the solid tumors, we found this effect that if we knocked out interferon gamma receptor or the signaling pathways, that these would confer resistance to CAR-mediated killing to a wide variety of different CAR target and CAR target antigens. What was interesting is that this was also different for liquid tumors. So when we knock interferon gamma receptor out of leukemia and then treat it with CD19 CARs, you see that there's actually no difference in terms of responsiveness of the tumor. Again, in the top graphs, we have wild type, the target knockout in the middle graphs for CD19 or BCMA for multiple myeloma. You see those curves come together. When you do interferon gamma receptor knockouts, those curves actually completely overlap with wild type. So those tumors had no improved survival in front of a CD19 or a BCMA targeted CAR. We then took the converse approach, where instead of knocking interferon gamma receptor out of the tumor, we knocked interferon gamma production out of the CAR T cells. And we did that by including guide RNAs to interferon gamma upstream of the CAR vector. And we made CAR T cells that had specifically the TCR knocked out and also interferon gamma knocked out in the experimental arm. And you can see that when exposed to PMA and ionomycin, these T cells can make other cytokines just fine, like GM-CSF, IL-2, and TNF-alpha, but they don't make interferon gamma. And what we found is that when we took these interferon gamma knockout CARs and we treated mice bearing either leukemia, which I'm showing you here, or lymphoma, which is in the paper, we see that there is no production of human interferon gamma in the blood of these mice. That's that middle panel in the top. But there is no difference at all in terms of tumor progression as shown by bioluminescent imaging. Very little change in engraftment of the CAR T cells and no change in survival of the mice. And so we really did not require interferon gamma production by the CAR T cells in order to mediate anti-tumor responses in leukemia, lymphoma, or multiple myeloma. So we then wanted to understand mechanistically what's different between the liquid tumors and the solid tumors. And the approach that we took is a transcriptional one where we took mCherry transduced CAR T cells, so they were red, and incubated them with green GFP transduced tumor cells, which were green, co-cultured them for a short amount of time, and then sorted them and lysed them separately for transcriptional analysis. And so we asked, how does the T cell walk away from this interaction, and how does the tumor cell walk away from this interaction? So the CAR T cells, when they were exposed to interferon gamma receptor knockout tumors, they looked like they had done the job. They had looked exactly as if they clustered transcriptionally, exactly as if they were wild type tumors. Whereas when they had encountered a tumor that was target knockout, they clustered differently. It was as if they hadn't seen the tumor at all. But when they saw interferon gamma receptor knockout, they thought that they had seen a wild type tumor and killed it and moved away. The tumor side, though, looked different, and it looked different for solid tumors than liquid tumors. And this is where, with solid tumors, we see that when the interferon gamma receptor is knocked out, it behaves as if there's no target. It clusters similar to EGFR knockout. But in a liquid tumor, after incubation with CD19 CAR, this is NALM6, the wild type tumor actually clusters with interferon gamma receptor knockout tumors, whereas the 19 knockout, so the target knockout, clusters separately. So what is this difference if we look a little bit closer at the actual genes and pathways involved? So what we found is that after exposure to CAR T cells, solid tumors have this massive upregulation of genes involved in adhesion and migration. And when you knock out interferon gamma receptor or the target, that upregulation of these adhesion pathways is much diminished. Whereas in liquid tumors, there was very little upregulation of immune cell adhesion and migration after exposure to CAR T cells. There was a little bit, but it was just not very much. And what we could also see is that when there's interferon gamma receptor knockout in the tumor cells, the binding avidity of the CAR T cells to the tumor cells is much diminished. So this is an experiment of acoustic force microscopy where you ramp up acoustic force over time, and you can measure how quickly the CAR T cells come off the tumor cells. And so when you have a wild type tumor, and by wild type, I mean it's interferon gamma receptor positive and it has a target on it, you can see that the CAR T cells will actually stick to the tumor pretty well. And at 500 piconewtons of force, there's about an 8-fold change in how strongly the T cells will stick. But when you have no target, the T cells don't really stick. That's the light gray line. And when you have interferon gamma receptor knockout, you see those two, this is with two different guides. You see that those CAR T cells are no longer as able to stick to the tumor cells as they do when it's wild type for interferon gamma receptor. And you can do statistics on it when you pick a specific point like 500 piconewtons to be able to measure the acoustic force. So if we look even a little bit deeper, not just at adhesion pathway gene scoring and avidity, which is a functional measure of adhesion and avidity, what we found is that there was a single gene that seemed to be particularly notable with regard to its adhesion phenotype, and that was ICAM1, which looked like it was differentially regulated in interferon gamma receptor wild type versus knockout tumor cells after exposure to CAR T cells. And this was true not only at the transcriptional level, which is shown in the volcano plot on the left, but also true at the protein level when we looked specifically for ICAM regulation after exposure to CAR T cells by flow cytometry. So when we look at the mean fluorescence intensity of ICAM, which is the graph on the left, you see that after exposure to CAR T cells, when the tumor has intact interferon gamma receptor, the ICAM expression goes very high, increases log-fold change, whereas when there's no target or no interferon gamma receptor, there's much less upregulation of ICAM1. It still goes up, but not by the level that it does when interferon gamma receptor signaling is intact. So then mechanistically, we wanted to understand whether ICAM was actually directly related to this adhesion pathway and also to the susceptibility to tumor cell lysis. And so what we did was we took blocking antibodies to ICAM and co-cultured U87 cells on the left versus NALM6 cells on the right with CAR T cells. And what you can see is that indeed, if you block ICAM, you get a change in the susceptibility of the tumor cells to CAR-mediated killing, which is this difference in panel F in the solid tumor. However, when you add blocking antibodies to ICAM in liquid tumors like NALM6, there's actually no difference in the killing. It's completely intact. If you knock out, if you block the binding partner of ICAM1, on the T cell side, so that's the binding partner, which is a heterodimer of CD11A and CD18, you can actually replicate that as well. So if you block either CD11A or CD18 in a solid tumor co-culture with CAR T cells, you see the separation of the curves where you have a decrease in the susceptibility of the tumor cells to CAR-mediated killing. These are all killing assays. But when you block LFA1 on the T cell side in the NALM6 liquid tumor, then you see that those curves overlap. So there's very little effect of CD11A blockade and a small but statistically significant effect of LFA1 blockade in the liquid tumor. And so we realized that this difference in the binding was really important, and it seems to be primarily mediated, but not solely mediated by ICAM1 and its binding partner LFA1. And then we did the converse experiment where we took the interferon gamma receptor knockout tumor cells and then overexpressed ICAM1 to see if we could then restore both the binding avidity and the susceptibility to CAR-mediated killing. And what you can see here on the binding curves is that indeed knocking in an interferon gamma receptor knockout situation, if you overexpress ICAM1, you increase the avidity of the binding, so the curves actually move up. And then in terms of lysis or susceptibility to CAR T-cell mediated killing, you see that the interferon gamma receptor knockouts is the red line at the top, overexpress ICAM1, and they come down to looking like they are wild type for interferon gamma receptor. So overexpression of ICAM1 restores both avidity and the susceptibility to CAR-mediated lysis when the interferon gamma receptor knockout tumor cell line. So what drives CAR T-cell resistance in liquid versus solid tumors? Summary points is that at least with commercial CARs and lymphoma, one mechanism of resistance is transduction and infusion of CAR Tregs. This is what we found. Of course, others have found CD19 loss, mutations in apoptosis, and other mechanisms of resistance. We've also learned that targeting liquid tumors is not dependent on the interferon gamma receptor pathway, and perhaps presents an opportunity to decrease cytokine-related toxicities by blocking or knocking out interferon gamma production, which we're now exploring. We also have learned that loss of interferon gamma receptor signaling in glioblastoma and other solid tumors is sufficient to drive resistance to CAR-mediated killing, and that part of that mechanism involves a critical role that interferon gamma receptor signaling plays in enhancing the solid tumor CAR T-cell interactions. It turns out that in liquid tumors, this adhesion is also important. It's just mediated by a different pathway. So I just wanted to thank the people who did the work. Becca Larson and Stephanie Bailey did the interferon gamma knockout experiments. Nick Hardvala and Gadi Getz did the single cell RNA sequencing with us in collaboration with Kathy Wu. And I didn't talk about our funded work with Kite Pharma today, so sorry, that's left over as an acknowledgement, and thank our funding sources and you for your attention. Thank you.
Thank you for an excellent talk, Dr. Moss. If there are any questions, you can.
Hello. Yeah. Hello, Dr. Moss. This is Pawan. Thank you for a wonderful presentation. It's truly nice to see some work related to interferon gamma. My question is in the context when interferon gamma is being used as one of the very important QC or release criteria for a CAR T cell. How, in the light of your research, will the future interferon gamma QC will be done?
And that's a great question. Thank you so much. I think, part of the reason it was used is because it's measurable, quantifiable, and, fast. If you needed to measure T cell proliferation or single cell RNA sequencing, we would never release a product in time. So I think for now, I think it's useful. It is a quick and easy way of measuring some T cell activity for the purpose of releasing a product. I think if we ever had interferon gamma knockout CAR Ts as a product, then we would need to come up with an alternative release assay. I do think that sometimes we have to develop these assays before we fully understand the biology of how the product works. But I think that's just what happens in medicine. I mean, I think we used drugs before completely understanding their pathways in many other settings. And so I would not advocate for blocking all, release assays based on, that's not the real potency assay, because I think we need to move forward, and it's fit for purpose. It's enough to just demonstrate that there is an antigen-specific T cell response.
Thank you.
Hello, doctor. It's Neeraj here. It's a very intriguing data, but just wondering how a very small population of Treg can inhibit the CAR T functioning. Is there any possible mechanism behind it? And second, related to that if we really want to get that out, then because it's very difficult to get them out from the T cell population. So what will be the path forward?
Thank you for the great question. So we've actually talked to, I'm sorry, first about the mechanism. Tregs work in multiple different mechanisms. It's hard to understand, even for me, how such a small population of Tregs has this effect. I think it must be very early on in terms of their ability to suppress the other CAR T cells in the product. They don't seem to expand in the patient. So at least not in our data, we don't see this massive upregulation or expansion of the Tregs. So they're probably putting the brakes on earlier on. And so you're not getting as high of an expansion initially or an area under the curve. I do think it's possible to remove them using like CD25 depletion up front. I don't know how efficient it is. And if you remove, if you go from 5% to 1%, if that's good enough. And I suspect that it would be clinically meaningful to attempt to remove the the Tregs before starting the transduction process. In the US, when it's all centralized manufacturing, it's very difficult because the process is locked down, and the sort of level of effort and money that it would take to change that process is cost prohibitive. When you have decentralized manufacturing or distributed manufacturing, and you have a little bit more opportunity to change your manufacturing process. From what I didn't hear all of the symposium today because of the time zone, but what I heard earlier, it sounds like India may have the ability to do just that. So I don't know exactly how it all works, but I could imagine actually testing that in patients in the context of a clinical trial, of having different manufacturing with or without Treg depletion, and really informing the field as to, how worthwhile is it? Do you see that durable responses go from 40% to 80%? If so, then, that would be an incredible benefit.
Thank you, doctor.
Yes, Dr. Randeep.
Hi, thanks for a wonderful talk. Are there any trials of combining checkpoint inhibitors with CAR T cells, mainly for the lymphomas?
Yes, thank you for the question. There are several trials that have been tested combining anti-PD-1 therapies with CAR T cells. I actually haven't kept up with all of them initially. I think it was very exciting. There were a couple of reports where there was like refueling of the CAR, and there was a re-expansion of the CAR T cells. But it turns out to be relatively minor, and overall it hasn't been the sort of breakthrough that some of us thought that it might be. I don't know if other checkpoints have been tested as extensively. Like, I'm not sure that I've seen an IPI combination or a LAG-3 combination, but I think it's being tested.
Any more questions? Okay. So thank you, Dr. Moss. I think we'll move ahead with the session.
Thank you very much.
So our next speaker is Dr. Eric Smith. He's the Director of Translational Research Immune Effector Cell Therapies at Dana-Farber Cancer Institute. We have been talking since morning about lymphomas and leukemias. Now we should move on to myelomas. And I think he'll be talking about non-BCMA CAR T cell therapies for myeloma. So I welcome Dr. Smith.
Hello, great to be here. Bring my slides up. Great, it's really an honor to be invited. So again, thank you And I'm delighted to address this Congress and look forward to the discussion after the talks. These are my disclosures. So CAR T-cell therapies, as you've no doubt heard over the course of this conference, have really been transformative for B-cell malignancies, particularly B-cell leukemia and lymphomas. And myeloma is in some ways no exception, and in some ways the exception that breaks the rule. It's no exception that the responses that we see are quite frequent and deep. Over 90% of patients with heavily pretreated myeloma are responding to the cilta-cel product. However, these responses are often not durable, and the vast majority of patients with myeloma will relapse. This is in contrast to the B-cell malignancies, where approximately 40 to 50% of patients appear to have truly durable responses and likely cure. And when patients relapse after CAR T-cell therapy in myeloma, their prognosis is quite poor. Most patients do go on to get another line or more of therapy. However, overall, the median PFS, as was shown by my former colleagues at Sloan Kettering and Mount Sinai, is only 3.5 months. This is somewhat better if the next line of therapy is another T-cell redirection agent, but still nowhere near as efficacious as the initial CAR T-cell therapy in this heavily pretreated clinical setting. So in our group, we study what are the mechanisms of relapse and how we can engineer around these. And this includes certainly with CD19 genetic lesions with BCMA targeting. There are some examples of genetic loss of BCMA, although far fewer. It's believed that there is a BCMA low antigen reservoir that may predict relapse, and even if that relapse is again BCMA positive. So we believe that another target was needed in order to address these BCMA low escape mechanisms, and we identified GPRC5D. This is work that I did in Ranier's lab, and I see Ranier in the front row there, so looking forward to talking with him later. This is data from the Cancer Cell Line Encyclopedia. There's 1,000 different cell lines here, and we're looking at the mRNA levels of GPRC5D. And this tall red bar, 30 different myeloma cell lines. And so this was really the first piece of data that clued us in that GPRC5D may be very specifically expressed by malignancies derived from plasma cells and not from other normal tissues. And in fact, when we do look at primary normal tissue and from publicly available transcriptome data, you can appreciate this huge delta between all of the different normal tissues. And in the top right up here, this is myeloma bone marrow. So myeloma bone marrow, we see the GPRC5D transcript much more highly than any other normal tissue. And these outliers here in the middle are from skin. And so when we identified GPRC5D as a potential target for immunotherapy of myeloma, of course, we went to PubMed and saw while there was a paper here or there incidentally finding GPRC5D correlated with myeloma, we also saw that GPRC5D was expressed in a subset of hair follicles across different species. So very little otherwise was known about GPRC5D. It's still to this day an orphan receptor, meaning no known ligand or physiologic function. And the potential for on-target off-tumor toxicity was primarily, as I mentioned, in the hair follicle and hard keratinizing tissue. So we were encouraged by that, and certainly not the heart, lungs, kidney, liver. So we thought that this was an attractive target that was worthwhile pushing forward as a potential immunotherapy approach. We went on to conduct antibody discovery with a human scFv phage display library from collaborators Eureka Therapeutics, and ultimately identified 7 scFv hits, and these bound to different extracellular loops on the 7 transmembrane receptor. And from these 7 hits, ultimately in collaboration with Juno Therapeutics, ultimately ended up contributing to the translation of this product. We developed a library of 42 different chimeric antigen receptors. This is from varying the orientation of the heavy and light chains, also looking at different spacer lengths. And I won't go into this data now, but we found from our earlier BCMA work and this GPRC5D work that there were two central assays that really helped us predict what we might see in vivo and of course, hopefully and eventually in the patients. And those are a proliferation after repeat antigen challenge and lack of tonic signaling. And through comparison of those assays, as well as others, we identified a lead GPRC5D targeted CAR. And skipping ahead, we used that lead GPRC5D targeted CAR to investigate this model of BCMA antigen loss mediated relapse. So here we've CRISPR'd out BCMA from a myeloma cell line that, like mostly all myeloma cell lines, endogenously express both BCMA and GPRC5D. And then we have firefly luciferase specifically in that BCMA knockout myeloma cells. And the mice get injected with a pool of the unmanipulated wild type cell line that expresses both antigens and the GPRC5D only expressing cell where BCMA has been knocked out. And then we treated the mice with either irrelevantly targeted CD19 CARs, our negative control, or two groups of mice were individually treated with the BCMA targeted CAR that we developed and went on to become Orva-cel, also being brought forward by Juno and BMS. And saw that, not surprisingly, the BCMA knockout population continued to progress. We then retreated the mice either with a second dose of the BCMA CAR or with the GPRC5D CAR, and demonstrated that the GPRC5D targeted CAR could salvage the mice in that situation. When we look at the myeloma cells from the marrows of these mice at the end of the experiment, we see very clearly that it's the knockout population only that's expanding and causing the disease in the BCMA treated group, and the mice where we target GPRC5D can do well long-term. So why is this important clinically? We've had patients, a number of myeloma patients that have been treated with BCMA CARs, patients who have had 8, 9, 10 prior lines of therapy, really very few options, get a BCMA CAR, in this case, the Orva-cel CAR stemming from our earlier BCMA work. And this patient has a really great response initially, completely eliminating their free kappa light chain and M spike, and does well for a number of years. But then, as you saw earlier on, as with most patients on all the trials with BCMA targeted CAR T cell therapies, begins to relapse. And you also saw that in this setting, patients have a median PFS of about 3.5 months from their next line of therapies. So we're very fortunate for this patient and others in that situation to have a trial now with these GPRC5D targeted CARs, particularly one of the first trials that targeted patients relapsing in the BCMA setting, or even had those patients be eligible for study. This is a standard dose escalation trial with fludarabine and cyclophosphamide lymphodepletion, autologous manufacturing at Memorial Sloan Kettering. And this trial resulted in recent publication. Shyam Malankody was the PI, and led, of course, in close collaboration with my colleagues at Sloan Kettering, Vernier included. And these patients, as I mentioned, were very heavily pretreated. They had high-risk features, including high-risk cytogenetics and extramedullary plasmacytomas, which we know are more challenging to control, including with cellular immunotherapy. And again, had half of the patients approximately had prior BCMA therapies, 59% had any prior BCMA therapies. It's a total of 10 patients, and 8 of those 10 patients had prior BCMA CAR T cell therapies. And so we were keenly interested on how patients with prior BCMA CAR T cell therapy would respond to this novel targeted therapy. As with any first-in-human trial, the selection of the starting dose is one that is really not taken lightly. It's always the balance between offering a patient a clinical trial that we assume is safe but may have minimal chance of efficacy, versus one where you take an unnecessary risk with potential toxicity. So in this case, we started at 25 million cells. For comparison, the FDA-approved dose of ide-cel is 450 million cells. But we had seen some efficacy at 50 million cells with the Orbicel product. Ide-cel is not active at all at 50 million cells, but given our experience seeing some efficacy at 50 million cells, we decided that the right thing to do is to have that and start this very conservative dose at 25 million cells. And this first patient had a large amount of extramedullary myeloma, and we were extremely pleased to see the response on PET scan 4 weeks post follow-up of the rapid eradication of this bulky tumor, even at this very low dose. So overall, patients did quite well. 70% of patients overall in the study had a response. And this was irrelevant if patients had prior BCMA therapies or not. 70% of patients overall responded. And several patients had quite durable responses, particularly at the 50 million dose, with 2 patients that continue to respond at this report with greater than a year of follow-up, and many of the patients in the highest dose level as well continuing to respond approaching a year. In terms of toxicities, there was the expected low-grade cytokine release syndrome. 88% of patients had some sort of cytokine release syndrome. There was one patient that had a severe cytokine release syndrome. The same patient had ICANS and macrophage activation syndrome. This was all transient and resolved in this patient. We saw the expected cytopenias, likely significantly contributed to the lymphodepletion chemotherapy. And then in terms of on-target off-tumor toxicities, we were paying close attention to any changes in nails or hair or dysgeusia, change in taste that's been associated with the GPRC5D targeted bispecific. And we saw some of these changes, but all grade 1 and transient changes. The dysgeusia in particular is in contrast with the data seen on the bispecific, which of course is continuous administration and may have a different bioavailability, where the dysgeusia and some of the skin and nail changes have been much more problematic. In terms of infections, there were, of course, a few, but overall, far less infections and severe infections than is being reported, particularly with the BCMA-targeted T-cell engagers. And then we did see, to our surprise, a cerebellar-like disorder. This is characterized by dizziness and altered gait. 2 patients, both at the highest dose level, that had grade 3 cerebellar disorder. And so this was only at the highest dose level. I'm not seen at any of the lower dose levels, but of course, this was something of concern and that we looked into. So we looked at the Allen Brain Atlas, which is a publicly available resource that has autopsy information from 6 individual donors, where throughout the brain, many biopsies have been taken, transcriptomics have been looked at. And so this has been done before for BCMA by the Mount Sinai group. BCMA targeted CAR T-cell therapy, particularly the cilta-cel product, results in a Parkinsonian-like side effect in a small subset of patients. And when looking at the Allen Brain Atlas, the investigators did find that there was differentially high expression of BCMA in the areas of the brain that might correlate with this Parkinsonian pathology. And so we took the same approach for GPRC5D, looking at this atlas, and we did see this one area. So in this case, there are 2 probes for GPRC5D. There's only one in the atlas for BCMA. We did see this one area, the inferior olivary nucleus, that it did have higher expression than in the rest of the brain. To be clear, this is still extremely low expression. In fact, when you read the guidelines of the Allen Brain Atlas, there are a total of 60 different biopsies in this area of the brain across the 6 patients, and very few, and just 1 or 2, actually would cross the threshold of what the Allen Brain Atlas said should be considered signal from noise. Albeit, while the rest of the brain is essentially silent for expression of GPRC5D, if you look differentially, there does appear to be a signal here in the inferior olivary nucleus, and that area of the brain would explain, a lesion in that area would explain potentially the cerebellar toxicity that we saw in 2 of the patients at the highest dose level. And then this other gene, POU for F1, just for comparison, this is the gene that's most highly expressed in the inferior olivary nucleus. So back to our patient who was relapsing after BCMA targeted CAR T-cell therapy. This patient was enrolled on the study and received a dose of a second autologous product, this time targeting GPRC5D, and again responded and continues to respond. So we're super excited to be able to offer this therapy to patients based on our work. Is now a multi-center study, including that's open at Dana-Farber. And similarly, the patients on this study had high-risk cytogenetics, preponderance of extramedullary plasmacytomas, and about half of the patients that had prior BCMA-directed therapy. Again, similar, we saw CRS, mostly low grade. In this case, they've dose escalated, not up to the 450 million dose level yet, where we saw the cerebellar toxicity. And as in our study at lower doses, they did not see cerebellar toxicity. There was 2 transient low grade, more typical ICANS events. And again, similar to our results in terms of the skin toxicity and the dysgeusia, there was a minority of the patients that experienced this, all transient and low grade. In terms of the responses, in this case, there was overall an 89.5 response rate, dose escalating and up from 25 million to 300 million cells, and impressively almost a 50% complete response rate. In terms of the patients that had prior BCMA CAR T-cell therapy versus those that had not, we see similar complete response rates, about 50%. In the no prior BCMA, there was 10 out of 10 patients that responded. In the cohort that received prior BCMA, it was 7 out of 9. So if that's meaningful or if that's just small numbers, I think yet to be determined, but we're very encouraged by the approximately 50% complete response rate, regardless of prior BCMA therapy. But the real question is, are we pushing the curve to the right, or are we actually raising a tail on the curve and curing some of these patients? And probably with our sequential CAR T-cell therapies, we're pushing the tail, we're pushing the curve, and we hypothesize that if we could put pressure on multiple antigens at the same time with CAR T-cell therapies up front, that this could lead to cure, at least in some of the patients. And so there is a trial that, again, working very closely with my colleagues at Memorial Sloan Kettering, contributed to leading the investigational new drug application and getting into the clinic. It's now enrolling with a carpool approach, so making 2 cell therapies, one targeting BCMA and one targeting GPRC5D at the same time, and giving them back to the patient simultaneously. And this is an advantage of a stepwise escalation, particularly important if there was a different maximally tolerated dose between the 2 cell therapies. So this will be important to learn from. And then again, using these same 2 therapies, the same 2 CARs, we're also interested in other dual targeted approaches, thinking about bicistronic vectors and single stalk dual targeted vectors. I already mentioned to you a lot of the data with our MCAR-H109 GPRC5D targeted CAR. Just to very quickly mention our BCMA data. So this has now gone on to multiple trials, multi-center trials. The first multi-center trial presented at ASCO 2020 showed a 92% response rate, with many patients completely clearing their myeloma paraprotein. And then patients that, particularly at the higher dose, that had ongoing responses. And there are multiple trials with this vector moving forward. Most recently reported at ASH 2022, just a few months ago, was this trial with BMS's NEXT-T rapid manufacturing, where they treated 55 patients, had very little CRS or ICANS, and an overall response rate of 98%. With this rapid manufacturing, they saw more central memory like cells, which may be one of the reasons that at these very low doses between 20 and 80 million, as opposed to up to 600 million, as I showed you on the prior slide, they're seeing this 98% overall response rate. So again, moving on from a carpool approach to studying other approaches that could be used as a single vector, a single manufacturing step. Carlos, who worked with Rainier and I at Sloan Kettering, published this work where he showed that not only is this feasible, but that there were some biologic advantages of having both CARs, or at least both targeting moieties on the same T cell. And I won't have time to go through all this data now, but just to show you at a very high level, the schematic that we explored, we compared a carpool approach to a bicistronic approach where the same cells are expressing two CARs, to a tandem single stalk approach where the same CAR is expressing binders to the two different targets. We tried to leave the orbicell construct constant for the maximal translatability. We also compared where we could either dual 4-1BB co-stimulation or the combination of 4-1BB and CD28 co-stimulation, and then had a control where we deleted the signaling domains. And again, I won't be able to go through this data, but just to say that we did find expressing both CARs or both binders on the same CAR to be superior to the carpool approach, both biologically in our in vivo models and obviously for ease of manufacturing and cost. So just to conclude some of the ongoing work that we have at Dana-Farber now, we've transitioned to looking at single domain binders, so VH-only binders, which are smaller, easier to package, and particularly for dual targeted approaches have the advantage of lack of concern about any cross reaction between the binders. And to do that, we immunize mice that have been knocked out for the murine variable light and variable heavy domains and knocked in for the human VH only domain. And so, of course, humans don't make VH only binders, right? In contrast to llamas and alpacas that do. But we have hundreds of millions of years of B cell biology that has evolved. And when that is the only binder, the B cells will undergo somatic hypermutation of these binders and make some really great single domain nanobodies. And part of the power of doing this is we use a technology called Beacon Berkeley Lights, where you can assess thousands of individual B cells in these nanopens. And the individual B or plasma cells will start secreting these nanobodies or antibodies if they were more typical B cells. And through a flow-like assay on the microfluidics chip, you can identify the single B or plasma cells that are making productive and specific antibodies to the antigen that you flow in. And then these can be captured and sequenced. And then CAR libraries can be made and analyzed to identify really optimal approaches. And this is work being headed in our group by Anasuya, who came to us after 6 years leading antibody discovery at Sanofi and is building a platform to really expand the throughputness of this approach for our group and others. And then Kartika in the lab really did the proof of concept here for making another BCMA binder that could really be a platform for these dual targeted approaches or other next-gen approaches in the lab. And just to show you some of the data here, and then I'll wrap up. We looked at killing. So this is individual wells from a 96 well plate where the CAR T cells are in red or orange here, and the target cells are in green. When we delete the signaling domains, we've done on the right, by 2 days after, we see the target cells there. The T cells without signaling have mostly gone away. But when we co-culture with an active CAR, including these VH-only binders, we see that the CAR T cells cluster up and proliferate and eradicate the tumor. And regardless of which of these, the clinical benchmark or VH binders, this assay can perform quite well, and sort of a low bar assay. But when we take this into the mice, we've identified, compared to a clinical benchmark, a single VH-only containing CAR that really can, if you look, these CAR T cells are administered to mice on day 14. First time we look by day 20, the clinical benchmark, and this optimal binder 1046 has really eradicated the disease. And then by day 69, we see the mice treated with the clinical benchmark start progressing but the BCMA 1046 VH only CAR can continue to keep these mice in remission past day 100. So in conclusion, current second generation CAR T-cell therapies for relapsed refractory blood cancers, while certainly game-changing for this heavily pretreated patient population, are susceptible to a variety of pitfalls that can be addressed with engineering approaches. Hopefully I've demonstrated to you that GPRC5D is an attractive target for the immunotherapy of myeloma, including with CAR T-cell therapies in patients that have relapsed post-BCMA therapy. Upfront dual targeting approaches, including GPRC5D and BCMA, will continue to make their way into the clinic, and our engineering strategies that we develop in the lab do have enormous implications on the results that we see for patients. So with that, I want to acknowledge my current group in the lab at Dana-Farber and collaborators in the Boston community, including Marcellus here, Omar Nadim, who's leading the GPRC5D clinical trial at our site, and an special gratitude for all those that I trained with and worked alongside at MSKCC, especially Rainier, Shammel, and Cody, who is led a lot of the clinical work that you heard about, and Carlos, who did the dual targeted work in the group. And thanks to my funding sources and to all of you for your attention. I'm happy to answer questions.
Patients. And can you tell me your experience about treatment of plasma cell leukemias for with CAR T cells? We know these are like really a tough population to treat with your normal therapies. Even transplant is not we know the median survival is 9 to 12 months after the diagnosis.
Yeah, I missed the beginning of the question, but I just caught the end there about plasma cell leukemias. And no, certainly are patients with a huge unmet need. We don't have a ton of experience treating patients with plasma cell leukemia as one. There are very few of those patients, but they've also been, I think, largely excluded from a lot of these trials. But I do think as we now have the FDA approved therapies, it's going to be really important to see how those patients respond. Of course, they'll have more aggressive disease, but there's no reason why they wouldn't have initial responses. So we'll see as that data matures.
And do you think for such difficult patients like plasma cell leukemias or your extramedullary plasmacytoma, if we utilize CAR T cell therapies earlier in the therapy, so would it make a difference?
No, that's an outstanding question, and certainly one that the field is thinking about. So there are quite a number of trials now with ide-cel and cilta-cel that are starting to read out in the earlier disease setting, and we're super enthusiastic to see the results of those trials, right, for a variety of reasons. They may work better, and one, the disease is less proliferative, and particularly for the extramedullary plasmacytomas, while some patients present with those earlier in their course, most of those develop after multiple lines of treatment and evolution of disease. So we expect better biology, less proliferation, and also the immune system is less perturbed earlier in disease. So we really are enthusiastic about that. In fact, at Dana-Farber, we even have a trial now with T-cell engagers and soon with CAR T-cell therapies, even in high-risk smoldering myeloma, really thinking that may be an opportunity for cure.
Okay. So there is another question from Dr. Kulwant Singh. So he's asking, do you think targeting both BCMA and GPRC5D will have high level of toxicity if you are dual targeting?
Yeah, no, I mean, we've done this sequentially already, and now we're starting to do it together. These targets, for the most part, are really expressed on plasma cells and myeloma cells, and I think it will be tolerated well, and hopefully there will be patients where these targets, at least one of them is expressed on every last myeloma cell, and this may be an option for cure. So we're really looking forward to both see of course, the safety signal and the efficacy signal, but I don't think there's any priori reason to be concerned about worse toxicity.
Thank you. Any other questions? Now I'll invite Dr. Garima to introduce the next speaker.
I'm very pleased to welcome Dr. Renard Brengens for a special event. Born in Netherlands and raised in Buffalo, Dr. Brengens completed a master's degree and dual MD PhD degree at University of Buffalo. So he was a faculty at Sloan Kettering Cancer for 2 decades and been known for his work in CAR-T. Currently, he's a deputy director at Roosevelt Park Comprehensive Cancer Center and a chair Department of Medicine. Welcome, sir, for your next talk.
Thank you so much. I have to say that I'm extremely pleased to see how well the CAR T cell field is doing here in India. I think Dinesh invited me first 12 years ago, 11 years ago, and how far it has come. And I know that I think you guys are far too modest about where you are. I think it's a numbers game, and I think it's encouraging to see that in India you're seeing the same success with this technology, and unfortunately also suffer with the same problems, be it cost, timing, et cetera, et cetera. So to me, it's a tremendous pleasure to be back here and to see how far things have come in the last 10 years. How do I don't know if it's. All right, so I will talk today about, just to point out, those 2 prior speakers, both of which were at Sloan Kettering. Marcella was there, did a lot of her training, worked in Michelle Sadeling's lab, and Eric, I'm extremely proud of Eric, worked in my lab for many years, developed those 2 CAR T cells for multiple myeloma. He has a fascination with multiple myeloma. I don't quite understand, but that's to each their own. And they've both gone on to obviously very prominent positions and continue to do great work. And so I want to thank both of them for taking the time to talk. So I'm going to talk about another aspect of CAR T cells, in particular, how we can move CAR T cells from blood cancers to solid tumors. And we coined the term armored CARs to describe this approach. I don't much care for the term trucks, which was mentioned a couple of times, but I think that this is where we are. So again, I'll quickly go through these initial slides. The concept of taking a antibody binding domain, fusing it to a T cell receptor signaling domain to create a chimeric antigen receptor that can then be packaged into, most commonly into a lenti or gamma retrovirus, and that can be introduced into and expressed by a T cell, generating a chimeric antigen receptor. This is work that was done probably before many of you were even in high school. This goes back 20 years ago. This was the first time, as you can see here, where we were able to demonstrate, first of all, the generation of expression of CAR T cells targeted to CD19. This was a first generation CAR we called 19-zeta-1. We could demonstrate specific lysis. We could find ways to expand these cells. We utilized IL-15 long before it became part of industry standards for expanding T cells. And again, in this survival plot, really, we were the first to demonstrate in vivo that CAR T cells could function up until that point. And this was published in 2003. Up until that point, most of the data that was published on CAR T cells was just able to demonstrate that in vitro that these engineered T cells could lyse targeted tumor cells. We quickly came on a problem when we tried eradicating NALM6 cells with that same first generation T cell. We could always enhance survival, but we could never actually get long-term survival of these mice. And we back then postulated that it was due to a lack of in vivo co-stimulation. And we tested that by engineering the tumor cells to express the co-stimulatory CD80 molecule. And then we repeated the experiment. And while the survival curves weren't as high as we got with the Raji tumors, which I showed you previously, you could still see that there's a tail on the curve and we could fully eradicate disease. And this is really what prompted us when we started to do our first clinical trial in 2006 to go with a second generation CAR. And so that's, I believe actually the expression second generation CAR came from this review paper. What we did initially is we looked at CD28 as a co-stimulatory signal. So we thought, and this was work done initially by John Maher in Michelle Sadellane's lab. So we created the first second generation CAR, or actually Dr. Finney did several years earlier, but in a T cell. And we call that 28 zeta. 1928 zeta was the construct. Folks have looked at third generation, so multiple co-stimulatory signaling domains. But really what is currently available, commercially available, is either 4-1BB or CD28 containing second generation CAR T cells. We could have an entire symposium debating which is better, the 28 or the 4-1BB. I think that clinically, at least, if we look at the outcomes with either the Kite product or the Novartis product, the outcomes are very much the same. We've generated CARs with both, with 28 targeting CD19, and certainly 4-1BB is what Eric used targeting BCMA and GPRC5D. So we took a second generation CAR, and if you see here, we again treated now with wild type NALM6 tumors, and you can see that with the second generation CAR versus the first generation CAR, we get about 50% long-term survival. And so that's what prompted us to move forward to a clinical trial. We were actually by 1 week the first to publish outcomes in relapsed refractory B-cell ALL. This was a trial that Marco Davila, when he was a fellow, initiated at Sloan Kettering. And as you can see here with the red box, the first 5 patients that we treated, 2 of which had significant disease burden at the time of treatment, all of them obtained an MRD negative, either by deep sequencing or by flow cytometric analysis, and MRD negative stature, which is completely completely unexpected. And as you can see here, given the fact that the standard of care was to transplant, if you could get a patient back into remission, 4 of the 5 patients here were transplanted, several of which are still alive today. Overall, in this trial, we treated 52 patients. This was, I guess we started it as a phase 1, then we called it a phase 1/2 trial, because we just kept enrolling more patients. And we couldn't stop enrolling patients, in part because there was no commercially available product for these patients. And this was the only trial that was open for adults with B-cell ALL. And as you can see, overall, we got a CR rate of 85%. And MRD negative rate in those 85% in 2 out of 3 patients. As you can see, there is a tail on the survival curve, but as you can also see from this survival curve, is that the relapse rate, despite a very high initial response rate, the relapse rate is really quite high. And pretreatment disease burden, which is intuitive, those patients tended to do better than those that had significant amounts of disease, in this case, greater than 5% blasts in the marrow at the time of treatment. And so, and then again, in the pediatric patients, this is the work that was initiated at UPenn. That was the first pivotal trial that, and this data again demonstrates a high initial response rate, but unfortunately still a significant number of patients that relapse. In fact, Carl June was able to demonstrate in the first 2 patients, the first being Emily Whitehead, the second patient unfortunately did relapse, in this case with a CD19 negative clone. This did lead to FDA approval, the first FDA approval of a CAR T-cell product, and now I think they're up to 7. But it also highlights some of the problems that we face in the field in the hematologic setting. And so we have a multitude of questions. What's the etiology of differential responses to CAR T-cell therapy? What is the role, for example, of bulky disease or high disease burden and CAR T-cell anti-tumor efficacy? And then more relevant to solid tumors is what is the role of the immune suppressive? I call it hostile, but it's really not hostile, I suppose. Tumor microenvironment and CAR T-cell function. How can we build a better T-cell, and can we extrapolate this technology to solid tumors? And so just to highlight this, a lot of the work in my lab in the last 10 years is really focused on solid tumors and having the ability to translate our promising CAR T-cell technology from blood cancers into solid tumor malignancies, which obviously are far more common and once metastatic, almost universally deadly. So what are we talking about when we're talking about the tumor microenvironment? Well, cancer cells scaffold themselves with elements of the immune system designed to inhibit the ability of immune effector cells to conduct their function. And this is unfortunately not a complete list, but the first thing, as we have seen today on multiple talks, there's tumor antigen escape. That microenvironment also, the tumors as well as cells that scaffold the tumor secrete inhibitory cytokines such as TGF-beta, IL-10. They express immune checkpoints such as PD-L1, PD-L2. There are regulatory T cells within the tumor microenvironment. There are myeloid-derived suppressor cells, tumor-associated macrophages, and more recently, what's gotten a significant amount of attention, cancer-associated fibroblasts, which themselves also release reactive oxygen species and other mechanisms by which they can inhibit CAR T cells. So you have an entire army here of elements that can inhibit the function of our CAR T cells. And again, Marcella did an excellent job of talking about the relevance of Tregs. We had done work years ago that could demonstrate that a single Treg could overpower 8 CAR T cell effector cells. And again, that might have been an underestimate on our part, because I think Marcella's data shows that it's 1 to 20 can impact the outcome of CAR T cell therapies. So we decided, well, what we can't ignore the gorilla in the room here, and so we started working on developing the next generation of CAR T cells. And the phrase that I use is we have a Model T Ford, and really what we need is a Ford Mustang. I actually have that car. I have it with a soft top, and of course it's a manual, because you wouldn't buy a car like that with an automatic. That was my midlife crisis, in case anybody wants to know. So we came up with a different concept. I think that was born out of initially looking at the ability of IL-12 to overcome Treg mediated resistance. And this was work that was done years ago by a medical student in the lab who noted that if you add IL-12 to a Treg effector T cell tumor cell mix, that the IL-12 would enable these CAR T cells that would otherwise be suppressed to have cytotoxic function and maintain cytotoxic function. So we came up with this armored CAR T cell approach, and the name was one of my postdocs one of my technicians that later left the lab and went to industry, called me a year later, Jan Nickham, and he said, I got the name for you. Call them armored CAR T cells. And so that is a bit more of a aggressive name, but the idea there is that it's any CAR T cell that is additionally engineered to have enhanced function, be it through expressions of co-stimulatory ligands, for example, the ability to secrete cytokines, or the ability to secrete single fragment length antibodies that can have biologic function as well. And so I'd like to show you some of that data and then finish up with a few slides on a story that we just published, I think last week. about how all of that fits together. So Mauro Avanzi was a postdoc in the lab and started looking at the cytokine IL-18. This was after we had done a significant amount of work with IL-12 and actually did a clinical trial with CAR T cells targeted to MUC16 and ovarian cancer that secreted IL-12. But we looked at IL-18 in part because it seemed to have a very favorable pro-inflammatory profile, and studies done previously in clinical trials where IL-18 was infused directly into patients, it may not have had great anti-tumor effect, but it was extremely well tolerated, and we couldn't say the same for IL-12. So we came up with a model where we have transgenic mice that express human CD19 and on the B cells. So we could use B cell aplasias as a biomarker to see how long our CAR T cells stick around in these mice. And we did not give preconditioning chemotherapy. And Dinesh and I had a discussion about it last night about the role or the need to give conditioning chemotherapy prior to CAR T cell infusion. So in this model, we actually didn't do that. We wanted to see if these CAR T cells would be able to function even in the absence of conditioning chemotherapy. And you can see here, if you have T cells that secrete IL-18 but don't recognize the tumor, there's no survival benefit. If you have T cells that just express the CAR, there's absolutely no survival benefit. But if the CAR T cell that recognizes the tumor also secretes IL-18, you almost get 100% long-term survival. And obviously this is classic synergy, and we were very excited about it. We wanted to figure out what the mechanism was. And so the first thing we looked at is do these CAR T cells persist longer than ones that are not armored? And as you can see here, the blue dots represent at different timelines the detectable CAR T cells in the blood of these mice. And you can see that by flow cytometric analysis. And then when we look out further, when you can no longer detect when the CAR T cells are at a very low level, we still wanted to know if they were still around. And you can see that here on your right, is that these mice up to 150 days, when we stop looking, have persistent B cell aplasias. And when we then look at the marrow, we could actually, by PCR analysis, find the CAR T cells back. So they have significantly enhanced persistence, these IL-18 secreting CAR T cells. We then further asked, if we re-challenge these mice, are these CAR T cells still functional? And you can see about 50% of the mice were resistant to tumor re-challenge as opposed to the controls. And then one of the critical things that we then also wanted to ask is, what happens, what does the IL-18 do? Is it just an autocrine effect, or does it have a paracrine effect? Does the IL-18, is that able to elicit an endogenous anti-tumor response that's not dependent on target antigen expression? And we did that in a couple of ways. We demonstrated that. The first is that we took tumor cells that did or did not have CD19 on the surface, and we wanted to see if we could eradicate all disease. So this is a model where from the get-go, you would expect to have antigen escape, and yet we still found that 25% of the mice cleared all tumor, both antigen positive and antigen negative. We then wanted to demonstrate that this was due to recruitment of endogenous T cells, and that's shown at the bottom, where you can see that by ELISpot analysis, there was actually significant increase in T cells that recognized target antigen other than CD19 on the surface of the tumor cell. So this is now a CAR T cell that not only has enhanced persistence, but also has the ability to recruit endogenous anti-tumor effectors that could also help avoid antigen escape, which we know we're going to see in the context of solid tumors. Another approach that we looked at that stemmed from work on CD40 ligand, this was done by Nicholas Kuhn, who at the time was a graduate student in the lab. He's since moved on. And the idea there is, if we get CAR T cells that only transiently express CD40 ligand, and if we get it to constitutively express this co-stimulatory ligand, we postulated that there could be a direct interaction with the tumor cell if the tumor cell expresses CD40, but probably more relevantly, it would interact with dendritic cells, induce maturation of those dendritic cells, get epitope spreading. So again, recruit antigen. And then also critically, those dendritic cells would secrete IL-12, which again is a dangerous, but a nice pro-inflammatory cytokine. So Nick decided to look at that, and we looked at it. These are all syngeneic immune competent models. And these are all models, again, where the mice were treated with the absence of conditioning chemotherapy. With CD40 positive A20, which is a lymphoma cell line, you could see that the armored CARs have significantly enhanced survival. And even when we looked at an ALL cell line that's syngeneic and CD40 negative, we could again demonstrate enhanced survival with the armored CAR approach. And when we then looked to see what can explain this, we looked at the spleen, and we could demonstrate that the dendritic cells in the spleen underwent maturation. We could also demonstrate that these maturing dendritic cells now by intracellular staining secrete IL-12, and we could, and again, consistent with what we were seeing. And when we then looked, well, what's happening at the site of the tumor, and we looked at the T cells, whether they be CAR positive or CAR negative, and looking at activation markers, you could see that these CD40 ligand armored CAR T cells are able to induce activation not only of the CAR positive T cells, but also of CAR negative T cells, which again is consistent with this epitope spreading and enhanced anti-tumor efficacy. And again, Nick could demonstrate this by ELISpot analysis, where even when we and could demonstrate that this was in fact specific to TCR mediated killing, because if you take the tumor and you knock out the MHC molecule, then you could see that anti-tumor effect is lost. So again, we could demonstrate significant recruitment of endogenous T cells targeted to the tumor cell, which again is a way of avoiding antigen escape. And to demonstrate that, in fact, this is what's happening, we then re-challenged these mice with a target antigen negative tumor. So we knocked out CD19 out of the A20 lymphoma cells as well as out of the ALL cells. And yet these mice that had survived that were initially treated were long-term survivors when they were re-challenged with antigen negative. So this isn't due to persistence of the CAR T cells. You could see that these mice basically have been vaccinated against their own, against the tumor cells, as you can see here with 90% long-term survival. And then a third approach that we looked at for armoring, and this was done, Dr. Rafiq, who's now at Emory, who was a postdoc in the lab, came up with the idea that we engineer the CAR T cells to secrete checkpoint blocking single fragment like antibodies. And the rationale there is that, first of all, it overcomes the concern, PD-L1, PD-L2 mediated inhibition. But secondly, more critically, at least in our mind, is that it's going to be targeted delivery. So these T cells are going to give this payload at the site of the tumor, and we would potentially be able to avoid systemic toxicities that's seen with these types of therapies. So again, 2 drugs for the price of 1. And we initially looked at that. We selected one human scFv, E27. And as you can see here, when we repeatedly stimulated our T cells on fibroblasts that expressed high levels of PD-L1, as you can see here. So 3 T3s with PD-L1, there's no proliferation or very little proliferation of the T cells, as you would expect. But when you do the same experiment with T cells that secrete this scFv, this blocking antibody, you could see there's restoration of normal proliferation. And the same is true when you do co-culture with tumors that express high levels of PD-L1. You can see here that in the upper left quadrant, those are the tumor cells, lower right quadrant, those are the T cells, and you can see that the T cells have very little efficacy. However, when those T cells secrete this scFv that blocks PD-L1, you see restoration of killing. The tumor cells are gone and the T cells happily proliferate, even despite the context of the fact that there are high levels of PD-L1. We could demonstrate this in a couple of mouse models. To your left is the Raji tumor cells that have been engineered to express high levels of PD-L1, and to your right, you can see NALM6 tumors that express high levels of PD-L1. More importantly, we were able to demonstrate that this is efficacious in the context of solid tumor malignancies. Here we looked at CAR T cells that recognize MUC16. We engineered them to secrete that the PD-1 blocking scFv. And as you can see here in a orthotopic model, when we inject these tumor cells intraperitoneally and then inject the CAR T cells intraperitoneally, you can see there's enhanced survival due in part to the fact that due in part to immune checkpoint blockade. We then wanted to demonstrate that the secretion of that scFv could also rescue other cells, so that it was not just autocrine, but also paracrine. So we repeated the experiment at the suggestion of one of the reviewers. And here we had CAR T cells that recognized tumor but didn't secrete the scFv, co-injected with CAR T cells that don't recognize the tumor but do secrete the scFv, and see if we could get a bystander effect. And that's exactly what we saw in this experiment, as you can see here. So there's not just an autocrine effect where the CAR T cell itself is protected, but there's a paracrine effect wherein this scFv can rescue other endogenous cells, for example, in the clinical setting, tumor infiltrating lymphocytes or TILs. We then compared it to giving systemic antibody that blocks PD-1. This was again, this is now in a fully murine setting, where we have immune competent mice that have ovarian cancers. We treated them with MUC16 targeted CAR T cells, murine CAR T cells, that either with antibody that blocks PD-1 or with T cells that secrete a PD-1 blocker themselves. And you can see that the survival curves are really quite superimposable. And we could demonstrate that these engineered armored CAR T cells have enhanced persistence. We could still detect them by PCR 120 days out after infusion in long-term surviving mice. And again, we're hoping to move that to the clinical setting. So now, taking all of this together and keeping in mind that we're really focused on getting on targets in solid tumors. This was work done by Janneke Jaspers, who was a postdoc in my lab, has since moved back to the Netherlands. And she was looking at this target DLL3, which is actually expressed on a lot of neuroendocrine-derived tumors, and specifically small cell lung cancer, which I'm sure the survival rates are no better here than they are in the United States. There's very little that we have to offer these patients if we can't irradiate or resect the tumor. Usually we irradiate. And so one of the targets that is expressed on that surface is DLL3. It's also expressed in other neuroendocrine tumors, but it's very little expressed on normal tissues, and so it's an attractive target. It's been looked at as a antibody drug conjugate in clinical trials. There was toxicity seen, but probably related to the payload, not the target so much. And so we worked with this company, Stemcentrx, who had these antibody binders. And Janneke looked at a variety of these binders. And again, it illustrates that not all scFvs are created equal. And you could see that we had binders to different domains of DLL3. And Janneke did a bunch of experiments first to see what their killing ability, and then to see if the killing was specific, as you can see here. And then we looked at cytokines and proliferation as a marker for which of these binders to move forward with. And several of the binders that were of particular interest was 18125 and 16.8, which was, again, their nomenclature, not mine. And those are the two that we looked further. And when we start looking at both systemic disease as well as orthotopic lung-based disease, we did the comparison between the two winners from our previous, from our in vitro analyses, 16.8 and then 16.126. And as you can see here, the 16.8 is was the winner, demonstrated the best in vivo anti-tumor efficacy. And again, we then, we could demonstrate that there was enhanced survival. We wanted to see if we could improve that. In this case, in the syngeneic immune competent model, we gave cyclophosphamide conditioning, and as you can see, the cyclophosphamide conditioning markedly improved outcome. Then we moved on to looking at comparing armoring with IL-12 or with IL-18. And as you can see here, the IL-18 secreting CAR T cells demonstrated clearly the best anti-tumor efficacy. And as you can see here, even a combination of cyclophosphamide conditioning chemotherapy with IL-18 secreting DLL targeted CAR T cells came out on top. And then finally, we again looked at immune checkpoint blockade. So not all of these armors have the same pathway. And so we repeated those experiments and then just added whole antibody. We didn't, Janneke didn't have the time to look at our scFv, but you could see anti-PD-L1 or anti-PD-1 antibody, depending whether it's a syngeneic mouse model or whether it's this xenotransplant model. And as you can see here in the xenotransplant model, the antibody does a good job, rather the CAR T cell with IL-18 does a good job. Adding anti-PD-1 further enhances the anti-tumor efficacy. And that's actually the construct that we're going to move forward to the clinic. And I think that is my last slide. And I apologize if I went over. It's usually bad form. But again, as many of you are aware here, certainly now, there are promising outcomes targeting CD19 and BCMA. That's a proof of principle. The question that many in the industry are now asking is, can we get this to work in solid tumors? I would propose that doing a clinical trial, just going after a solid tumor target with a T cell that's engineered with the CAR alone is probably not going to be successful, maybe in rare instances. But to really address many of these limitations that we have with this technology, additional genetic engineering, I would argue, is be one way that you could improve outcomes. And that example, I've given you 3 examples of ways to armor CAR T cells. I would also point out that we haven't even tried putting more than one of these armors in a CAR T cell. So the ceiling is still pretty high. And one of the frustrating things that I think you guys have, but we have that in the United States as well, is that we've come up with all these ways of treating mice. Ultimately, that's not what we do it for. We do it to treat humans. The cost, even in the United States, is very prohibitive. And there's really no funding mechanism to be able to run these clinical trials. And so you hope for institutional support to be able to run the trials. Alternatively, you hope for industry sponsors and licensing of your technology to industry. The downside to that, of course, is that you lose control over your own technology. And so that's both beneficial. Juno Therapeutics funded, was able to pay for 52 patients treated on our ALL trial. They're also funding all of our trials in multiple myeloma that Eric helped develop. But going into new targets, et cetera, it gets complicated. And again, the field could move ahead so much more rapidly if we had funding to be able to test this out in the clinical setting. But like I said, we're certainly hoping to do that. And we'll be doing that at Roswell Park in the very foreseeable future. This is clearly an old slide. You can see Eric's still on this in this picture. I need to get a new slide since I moved to Buffalo, but that's the placeholder that I have for now. And I'd be happy to take any questions. Yeah.
You showed us various models of armored cars. Is there any one which you would prefer to take to the clinic given an opportunity? Which one? Because you can't really take all of them. So which is your top one of all those different strategies you suggested?
Oh, we could try to take all of them. We're very excited about IL-18. We're running a CD19 with IL-18 trial at that we started doing that at Sloan Kettering. And I think that is up and running. And then we're transferring 2 trials, one with IL-12 and one with CD40 ligand to Buffalo, and we'll be running those. The fortunate thing is that we have historical comparators because we have a whole cohort of patients that we treated just with 1928 Zeta. And so this will allow us to make kind of a historic but relevant comparison to see if they're better. My gestalt is that it's going to allow us to treat at lower doses. One of the complexities of this, of course, and why I kept emphasizing that we were able to eradicate disease in mice without prior conditioning chemotherapy, is that the whole rationale is that you're recruiting the endogenous immune system. If you give a big slug of fludarabine and cyclophosphamide, you're going to wipe out the cells that your CAR T cells are designed to recruit. And so what we came up with, and we'll see how it goes, is a zipper design where we give the CAR T cells no conditioning chemotherapy. If we get no response, then we give conditioning chemotherapy, then CAR T cells, et cetera. Carl June also is working with IL-18 that was reported, and they had one patient that did not give conditioning chemotherapy to with CD19 targeted CARs and went into a complete remission, which again, that's I think what we would predict.
Would you be worried about the T regulatory cells in that case?
Look, we know that IL-12 overcomes inhibition by Tregs. We haven't formally demonstrated that the same is true with IL-18, but our sense is that IL-18 works in multiple ways. So we haven't formally demonstrated Tregs, but what we can demonstrate is, for example, those tumor-associated macrophages that you see in solid tumors, they go from a suppressive M2 phenotype to a proliferative or pro-inflammatory M1 phenotype. The other things that we're doing, looking at that targets, is we have targets for cancer-associated fibroblasts. For whatever reason, cancer-associated fibroblasts, they're inhibitory. They provide a barrier to protect the tumor from the immune system. But they also curiously express a number of target antigens that you only see expressed not on normal fibroblasts, but on cancer-associated fibroblasts. And so that might be an Achilles heel. So we're actually moving forward with that too, we're very excited about.
But going beyond T, going beyond T for solid tumors, there's a lot of talk about macrophages.
Yeah, I think that's, look, I mean, we get this all the time, and there was somebody spoke about NK cells, and also the discussions about doing this in the allogeneic setting, which is great. I mean, if you can get it to work in the allogeneic setting, that's phenomenal. You can make a gajillion, and you can give the CAR T cells the day after you diagnose the patient. The downside is that immunologically there's a lot of barriers with the allogeneic setting, and none of which have been overcome to date. When we talk about NK cells, you have to give multiple infusions because the NK cell is not designed to undergo logarithmic expansion like T cells. If my argument would be this: get it to work, get a solid tumor to disappear with an autologous CAR T cell product. And once you do that, then you can start exploring, because if you add the variable that you're doing in the allogeneic setting, you'll never know whether your success or failure is due to the context. And macrophages, I mean, we're actually playing around with macrophages in the lab, but macrophages are not designed to kill tumor cells. They can, but they're not designed as such. And they don't proliferate barely at all. That's hard enough. And so, I think both with NK cells, gamma delta T cells, all these other cells, it's fine. Other people want to work on it, but I'm not spending any time. I'm taking the best T cell, the most potent T cell, and I'm trying to engineer it to eradicate tumor. If we get that done, then you can play around with NK. They say, NK cells are less toxic. Yeah, but they're also less effective. And the rule in my lab always was, if someone's giving a talk and the first sentence is that the treatment was well tolerated, you can go home, because we used to have, when we started doing our CAR T cell patients, and we were the first to recognize cytokine release syndrome. And patients would literally get depressed if they didn't have to go down to the ICU because they felt it wasn't working. And the patients that did go to the ICU were given meds that when they got back, they don't remember being in the ICU. So, but yeah, we really had to counsel our patients because they were really hoping to go to the ICU because that would mean that it was working.
So I've got two. So I've got two questions from the net. One, Dr. Sunita Olampally. How is the CAR T cell therapy response in naive, no chemo given patients?
I think Eric and I would both love to find the answer to that. Especially, this is especially true when we talk about, these hematologic disorders where prior chemotherapies are hitting down the lymphocytes. Most of the chemotherapies, other than antibodies, chemotherapies kill off T cells, and so T cells have to recover. Eric, do you have any? I mean, we would love to see it in the upfront setting.
Yeah, I mean, I think I talked a little bit about this to a question after my talk, but those trials are ongoing. And so we'll see. I mean, there's also an idea that you could collect T cells early and just store them, even if it's not the next line of therapy, and then the T cells may have better fitness. But no, I think that for all the reasons you mentioned, Rainier, it's very encouraging. And if this becomes, standard for frontline or first relapse in some of these B-cell malignancies, I think that's going to be great for patients.
And I think especially just to add to that, there are other trials that are going on in ALL with those patients that we know that have bad prognostic markers. We know they're not going to fare well. I believe they give induction chemotherapy and then go straight to CAR T cells.
Very interesting question. Could you foresee the future of cellular therapy as a therapeutic option for eradicating cancer stem cells? This is by Pramod Kumar.
It's a therapeutic for
cancer stem cells.
For cancer stem cells.
Yeah.
I think, and I'll give Eric a chance to answer as well. We are looking at, certainly with AML, the last bastion for transplanters. We are looking at targets that are expressed on the cancer stem cells, and so depending on what targets you're going after, the cancer stem cell targets are the ones that are being focused on by several groups, including ours. I think we are looking at CD371, for example, for AML, which is thought to be a stem cell, cancer stem cell marker.
So there's somebody on the net, I think. Rahul, do you want to ask a question?
No, no, I don't want to ask.
Okay. Sorry.
I'm happy to chime in on the cancer stem cell question. There's no reason why the CAR T cell wouldn't effectively eradicate cancer stem cells. It's more of a question about the individual diseases and the states of what we know about cancer stem cells and the diseases. AML, as Rainier mentioned, we probably know more than for other hematologic malignancies, and the work that Rainier is doing and others, I think, is highly promising. The challenge is differentiating the cancer stem cells from the normal stem cells in that disease. In B-cell leukemias and lymphomas, we're already curing half of the patients. So in that case, every last B-cell, malignant B-cell is being eradicated. There's another half the patients, of course, that we need to think about, and maybe that's something to consider. In myeloma, it's much more controversial if such a cancer stem cell exists or not, and what its immunophenotype would be, but clearly we're not curing patients. So there is a cell we're missing. If it's a true less differentiated like stem cell, it's something that we're looking at. But there are other alternative hypotheses as well in terms of, could it be somehow immunologically hidden by its local microenvironment?
So there is one more interesting question, that the question is, how many percentage of leukemic patients post CAR T therapy would require a bone marrow transplant? Would still require a bone marrow transplant?
I'll take that one.
Yeah.
The answer is we don't know. And especially, if you're MRD positive, clearly you need to go to transplant. If you're MRD negative, we looked at this retrospectively when we treated 52 patients, and that's not a scientific way of doing it. The study was not designed to answer that question, but we wanted to peek in because we were curious, and we looked at the number of patients that did go on to transplant versus the ones that didn't, and the survival was the same in both. Again, I'm not proposing that answers the question, but it certainly, we don't see a dramatic difference when we look back retrospectively. I think the argument for some, for kids, et cetera, not to transplant can be soundly made, especially if you achieve an MRD negative response.
So there is one more question. Please go ahead.
Oh, sorry, if I can, I would split hairs a little bit there, because I think there's a clinical answer and a scientific answer, right? Clinically, as Rainier pointed out, these patients retrospectively do equally well, but of course in the transplant arm, there's going to be some transplant related mortality from infections and the like. So in the end, the two groups do okay, but if you look at the patients that at least get through the early period, there may be a higher cure rate from the transplant, and scientifically why this may be is because we really want to get to a lot of what Rainier was talking about in his talk, trying to get to this epitope spread where we're able to eliminate some of the target negative cells, or going back to the stem cell idea, if they have a different immunophenotype, that's going to be essential for curing solid tumors, I think, where the antigen heterogeneity is such an issue, and will contribute. So if we can increase the epitope spread killing with some of our therapies, then that may even further overcome the need to consider transplant. But a lot of the really long-term survivors, even from the UPenn group, have had transplant.
Yeah, again, the converse argument is the ones that didn't get transplanted were probably older patients and less fit patients as well.
Thank you. Any other question? Yes.
They're usually, when you get them into remission, it's usually the first time they're in remission for a very long time. So they actually, once they, a month out, these patients tend to be doing extremely well. And so they're in better shape to get the transplant. I think Eric and I can both agree. I mean, years ago, I told the chief of bone marrow transplant at Sloan Kettering when I was still a fellow, I told him with a smile on my face that, I saw it as my job to put them out of business, and he thought it was funny. But I think we're getting there. I mean, keep in mind, if you want to look at the future, the early debate was 28 versus 141BB. I don't think that if 5 years or 10 years from now, the current CAR T cells will no longer be on the market. There will be something newer, better. This is going to be, I guess, a lot like the iPhones. Is that every few years is going to be a next generation, and hopefully those next generations will be more and more informed, and that we conduct studies so that we can see. The hope is that 5 or 10 years from now, we look back at the commercially available CAR T cells and say, "Wow, that was really pretty primitive." We'll see.
Yeah, I couldn't agree more with that. That's the goal, and we're already seeing it in some of these earlier trials. They're randomized trials, especially auto transplant versus CAR T cells. So that's a great goal to put the transplanters out of business for sure.
And then we're going to take on the radiation therapists, radiation oncologists as well. So and finally, the surgeons. Yes.
So my question is more of a fundamental, basic question. If you try to compare the microenvironment or the physiological microenvironment that we achieve after giving chemotherapy versus targeted therapy versus CAR T cells, which of these individuals, or even we can take immunotherapy, because PD-L1 and CAR T cells are more or less coming together. So which state achieved after remission? Example, you gave examples of patients having achieved remission or MRD negativity. Which of these patients would have the most physiological body milieu after having achieved MRD negativity. Most of our questions, for example, whether transplant is required post CAR T cell MRD negativity, why can't we go for solid tumors? Means I believe that our answer lies in this question. Means which of these provides the most physiological? For example, it is simply that we should not, when we say that we are not able to have CAR T cell therapy for solid tumors, it should principally be wrong. We should be able to target, for example, if there we are targeting some CD19 or 20, here we should be able to target HER2 new or something like that.
Well, I mean, a solid tumor, as opposed to liquid tumors, scaffolds itself. So when and target antigen expression is heterogeneous at best for any target that you go after. I mean, we know that out of hand. So
this heterogeneity applies for hematological malignancies also.
yeah, but hematologic malignancies do not have as profound. If you look in the bone marrow, you don't see that type of tumor microenvironment. You simply don't, and you don't have cancer-associated fibroblasts that can block penetration into solid tumors. You have that with solid tumors. You don't have that with liquid tumors. I'm not aware in leukemia that you have cancer-associated fibroblasts. When you look at a bone marrow that's infiltrated with leukemia, those are predominantly leukemic cells with some stromal cells put in there. When you look at the expression of CD19, BCMA, if you look at GPRC5D, all those targets are almost universally expressed on the tumor cells, 99.9% if you do flow cytometric analysis. On the other hand, if you do immunohistochemistry staining of a tumor, and you look at one corner of the tumor, you'll see HER2 expression, and at the other side you won't, because the stability, the genomic stability of solid tumors is far less than what we see in hematologic malignancies. And so I agree with you that on the surface of it, if you can engineer an immune cell to kill a tumor, to recognize and kill a tumor cell, it should work. But we need to be cognizant that the biology of a solid tumor, that the
tumor microenvironment basically.
It's the microenvironment, it's the structure, it's the heterogeneity of target antigen expression. Eric, do you want to add anything to that?
Yeah, I think that's exactly right, and this is, of course, where we all want to go. But there are these challenges we have. So we don't have the clinical efficacy in solid tumors. The antigen heterogeneity that you mentioned, one of the differences, the targets that we go after in hematologic malignancies are expressed on the normal cells. These are lineage markers, and it's okay that we can eliminate the normal B and plasma cells. Patients tolerate that fine, but we can't go after the lineage markers for the colon epithelium. It would just be unacceptable toxicity. So finding the right balance of antigens where they're on the vast majority of the malignant cells, but not on the normal cells which they came from, is an additional challenge. And that's being addressed at our group and others with logic gating. But it is, that's still problem A, and problem B, as we're going into more details about, is the microenvironment and the exclusion of some of the T cells from these tumors.
So my first part of the question, in which case are we going to have the most physiological milieu after therapy?
What do you mean by physiological?
For example, we believe that post CAR T cell therapy, there is a prolonged remission. In some cases, we may not even require transplant. So do we believe that it is the state where most of the cancer cells have been eliminated? Or
and if a CAR T cell is properly designed, as opposed to chemotherapy, which is far less targeted, you would hope that if it works ideally, you retain your normal physiology in the rest of the body, as opposed to when you give systemic chemotherapies. But I don't know until you do a comparison. My guess is that if you had to do a straight up comparison about standard chemotherapy for ALL versus CAR T cells, my guess is that CAR T cells would ultimately win.
Thank you. Sir.
Thank you very much, Dr. Eric. Thank you for giving us a lot of time. Thank you, Renier Brenjens. Dr. Brenjens, he took a long flight from New York to come here and just for this presentation, but his presence is important. This is his fourth visit to India, his first visit, as we said, the first ever meeting on CAR T cells we did in 2012. It was known as adaptive immunotherapy. So we are after CAR T cells, and we are happy that within 10 years we have reached somewhere. There are so many of you today sitting here trying to manufacture at some phase or so. So I'm extremely grateful to you for really generating an interest in us. And thank you very much.
My pleasure. Next time I'll get Eric to come as well.
Yeah, definitely. And I would really like, we had a small panel which we have discussed all these issues. So I would like to end the session here. And at the end, I would like to actually thank all of you who have taken time to come here, to spend time literally whole day and sit with a subject which is not that unique for a clinical practice and not even unique for a research because it's so detailed a subject. And I'm extremely grateful for encouraging us to organize this meeting. It was not easy. When we spoke to her, you said, yes, go ahead, go ahead, go ahead. And that's how we decided to make it. And grateful to Dr. Gaurav, Tanveer, Shiva, who really pushed this meeting in the beginning, Dr. Kamath, and companies like you who really helped us to keep the momentum. And really, I'm grateful to young oncologists, hematologists who have come here and given us really an audience to bring this topic up. I'm sure this would generate some interest. There had been about 200 people on the Zoom. And besides that, this was telecasted live on LinkedIn and YouTube. So I'm sure there must have been many more views. And with this whole national participation, I think this was a great meeting, which really pushed the idea of CAR T cells as an advocacy event. And I, once again, thank you very much. Thank you. We go out for a dinner. Dinner is ready? Yes.